Okay. Well, I'm excited. I'm just trying to get the focus there, but... All right. Thank you. We're just looking out for a rock then that looks like the right size. Are we live? We are. Hi, everyone. Welcome to Dive 420, which is the second dive of our expedition, where we are headed to the equator to look at corals in the Phoenix Archipelago. But today we're still in international waters at the high seas, looking at an ABNJ seamount, areas beyond national jurisdiction, on a high seas seamount that has never been mapped, never been explored. So we mapped it last night and this morning, and we just got the map, the completed map about an hour and a half ago, and then just turned it around and turned it into this dive. So this is never before seen seafloor, and we are out exploring. My name is Randy Rogen. I'm here with Anamare Tanaba. Go ahead, say something.
Hello everyone. My name is Anamare Tanaba. I'm an undergraduate from the BYU-H.
Yep, and Anamare is from Kiribati, which we had hoped to get to, but their borders are still closed due to COVID, so at this point we are so sad. But we will figure it out, and we are going to go to the United States, to the Pacific Remote Islands Marine National Monument, Helen and Baker Unit, and spend some more time in international waters, and it's going to be a great cruise. We are looking for a number of things today. We hope you will help us identify creatures and help us find some of the things on our list. So one of the things that we're trying to do is identify and look for corals and sponges. We'll be trying to identify new species, trying to look for range expansions, trying to just document and characterize the diversity of the community that's here. Steve, tell me if you want me to stop for any of these corals in particular. That's okay. Okay, keep going. The fish button. We are interested in corallivory, so organisms that eat live corals and live sponges, so spongivores and corallivores. We are interested in coral associates, so trying to understand who lives on which corals and why, and whether or not those relationships are have high fidelity, or whether or not they're a little bit more flexible in different parts of the ocean. We're looking for biogeographic breaks. We're looking at deep sea bacteria, and we're thinking about the origins of ancient immune systems, specifically basal metazoans like sponges and corals, and trying to understand whether or not their metabolism changes in response to various immune threats. So we are. Ooh, there's a little star. Let's take a little look at that. Actually, take a little snap zoom. So we're looking for all of those things, and we're also, always have an eye towards the unknown. And so we'll see what we find. Ooh, and this is my first time on the camera in a while. Am I fighting you for it? Is that what's going on? Oh yeah, you guys took it. Yeah. It's right there.
Nice to start.
Perfect. And actually, while you're down here, it looks like there's a lot of good rocks. Mm-hmm. So one of our first orders of business whenever we land on a seamount is we are collecting a rock for the USGS, the U.S. Geological Survey, because we are trying to, among other things, age these seamounts, try to figure out how old they are. They are all volcanic in origin. They've been here for a long time, and so by looking at the composition of the crust and the mineralogy of the rock itself, our collaborators are able to give us some insight into the geological history of these underwater mountains. The feature we're diving today is pretty cool. It actually looks just like some of these little brittle stars that you're looking at right now on the rock. It has a really cool shape with these long ridges. We are diving, I don't know if James can put it up, but we are diving on the edge of one of these ridges, one of these arms, if you will, of the sea star. And can I zoom in? Okay. And we're trying to get a handle on, get our bearings just as we have just come down. Great. Oh, and I see corals up to the right. This is going to be a good spot. I'm excited. Oop. Nope, not mine. I don't have control. Now do I have control? Okay. The ROV pilots and I are trying to figure out who's got the camera. Treated to a little show. Oops. We have Steve Askovich in the room with us here. He is a postdoc at BU in my lab. We're both at Boston University, and I'm so excited to have Steve. He's new because Steve is a deep sea coral taxonomic expert, and a lot of the stuff that we're seeing is easy to identify to family, pretty okay to get to genus, and practically impossible to get to species without Steve. So we're really excited to have his taxonomic expertise in the lab. And when Steve says that's probably a new species, he's almost always right. So it's really great to have Steve in the room with us. He'll be able to tell us a little bit about what's new, or in other cases, what's old in the deep sea coral world, right?
So true.
So true. This is Anamare's second deep sea dive.
Yes, and I'm very excited for this second dive
Maybe you
to
could
see more things.
maybe you could tell people in your own language too a little bit about what it's like.
Okay, ngayon kanapin ni Mary on kay ayong kay the second dive ay karawak the sea mount. Guys ang tawroy na nari nori bayay ka anong mga rin inanon tayo di research ay bugin karawagin te kakay kita kami tabayan na karawag. Kamra pa.
I have no idea what you said, but maybe you could tell us in English too. I don't know. Maybe not.
Yeah.
Or something else in English. It's pretty exciting to be in the control room for the first time, or your 25th time, or even your probably 250th time. I always
There's a lot of
there's a lot of stuff going on. I know this is exciting. There are 35 screens in this room and 39 people on ship, and all 39 people and all 35 screens are necessary in order to make this exploration happen as we broadcast to you live from 1,870 meters below the surface of the Pacific Ocean. Ooh, nice.
Can I white balance on that?
Yeah, please do. So the ROV pilots are going to white balance on the star. I love it. Do you know what kind of star this is?
I think it's a terastrid.
A what?
Terastrid.
Terastrid.
PT.
Yeah.
Is there anything that I can help with? I'm
Good
just
to go? You want to take over and look
Yeah.
around and walk?
I do. Okay. But I want to take some notes. Okay, so it is most likely a terastrid sea star it's got some sediment particles on it, which is cool. What about? I'm just looking around for a rock before we get really into the corals because I feel like the terrain is going to change pretty quick. What do you think?
Looks pretty angular here.
Yeah, it does. Oops. None of these look really dislodgeable, huh? No. Oh, well,
There's
let's
some up top there. Those little small ones. But
maybe.
you want a round one, right?
I want a round one, as round as we can find.
As a ball.
But let's go take a look at some of these corals. We've got a really cool one right in front of us, and then maybe we'll just keep our eyes open. All right. Ooh, we've got a sea cucumber, urchins, lots of ophoroids, and what a beautiful whip coral here. Just trying to focus here. I can't quite see it. Oh yeah. Some kind of Antipitherian, black coral, most likely Stichopathes. I don't see any associates. But let's get this into focus. There we go. Oops, nice. Just take a quick look at these urchins. You see how they, Anamara, you see how they have these big long spines? So they can, that's how they walk so they don't get stuck in the mud. They can just kind of like tripod themselves up around everything. All right, that's very cool. And we've got a cucumber over here. And then I saw a coral up to the right. So let's go take a look at that one. I think it's over.
That one, that's
Yeah,
right
both of
there?
those. Yeah.
Okay.
What is that thing?
What is what thing? The purple thing? That was a sea cucumber.
Oh, okay.
Yeah. So, yeah, we'll take a look while they're flying. They're gonna get mad. But you see, it's right there.
Mm-hmm.
Yep. Sorry, guys. I'll try to stay zoomed out.
Look at that Plexaurid on
Yeah, we're gonna take a look at this plexaurid. It's got an ophoroid in the middle, so it's got another one of these brittle stars kind of curled around all of its arms. You see that? All right. We are 1,870 meters. I know our overlays aren't coming up yet, so we'll try to keep you guys posted. We're going to take a look at this really cool plexaurid. Oh, looks like the ophoroid's waving to say hello. You see that? It's got an arm. It's
Yeah.
waving out. There we go.
Is that a gold coral?
It's a coral. It's no, it's not. It's not a chrysogorgid though.
Oh, wow.
Take a look at that. Isn't that gorgeous?
Yeah.
So, you want what? Can you send Tim a note and ask him if he wants the oath? Beautiful. So I'm really interested in looking at the coral's behavioral response to having these associates on them. You can see how the polyps are tucked in, but this ophoroid is not eating this coral. It's instead just hanging out using the coral for structure, and they're both catching marine snow that passes by. And we'll try to get you some fish zooms. I see ya. Oops, I was still figuring out how to get the controls where I want them. Okay, I think we'll take a little sample of this, if that's okay.
We're just trying to figure out how deep we need to go.
Sample
to
of
get
that?
Filter. Yeah.
Yes.
Yep.
Come around this side.
Samples for me.
Okay, we're gonna try to get a piece of the ophoroid and a piece of the coral. So you can get a
Okay.
piece of the arm.
Where would this be going?
This is gonna be going into one of the quivers, but not an RNA later quiver. Yep. We are. You guys, hang on. You guys. Is that a Walteria? Maybe. It looks like it.
Like the long one? No?
Yeah, it's a big sponge. It's an old one. It's a cool spot.
Good drop-in location.
Yeah, who called that one?
You did.
I did. Thanks, Dougie. We've got some. See, there's all kinds of stuff. There's a shrimp over here. There's a little anemone. Yeah. So we're really interested in these corals because these plexaurids are really hard to categorize, and so our on-ship taxonomist is really interested in trying to get this group resolved taxonomically, and so it'll be really interesting to take a look at this. And our coral associate guru, Dr. Tim Schenk from the Woods Hole Oceanographic Institution, is really interested in the associates that live on these corals. It's pretty interesting, actually. When Tim starts looking at these, he often can tell that there is an associate that only lives in one type of coral. So sometimes, it's funny, the ophoroid biologist can feed the coral biologist by saying, hey, I know that this ophoroid doesn't usually hang out on this coral, so I bet this is a new species of coral. So it's kind of fun you can kind of puzzle piece it backwards that way. Okay. That's a good shot. So as someone's pointing out in the chat, we are indeed the first humans ever to see this seamount, ever to see this spot on the seamount, ever to see this coral, this rock. It is all new to us, but it is, what's nice about it is
Ooh,
it's
it's waving.
it is waving. It is still somehow familiar because this is, similar to many of the other seamounts that we see in this area, but it's those similarities and those differences that allow us to get the context, the biogeographic context that we need in order to really start to dial in on what each seamount looks like. We are looking at a fairly cool feature. It looks like one of these brittle stars, and we're on one tiny piece of one tiny bit of a really long ridge. And you could spend forever exploring one of these seamounts and still not see it all. So this is going to be a heavy collection dive. We have a lot of things that we are going to look at in the lab, probably at 2 a.m. for us, ship time tonight. But rest assured, the collections that we'll be taking, we're
Look at
taking
that.
exactly what we need. Oh, it's so cute. Swimming away. And we're taking from such a small part of this seamount that there are undoubtedly countless more corals, oafs, sponges, and other things that live all the way around. So, as we collect and as we explore, it's always interesting and important to keep in mind that this is just one small part, and all of these collections are critical records that help us to understand and contextualize everything that we're seeing here in the deep sea. All right, we're just trying to set up here for a collection, and we've got a lot of little things to look at on this rock too. So we picked this particular spot because one of the questions that Brian Kennedy, who is one of the grad students in my lab who's on ship, is interested in asking is in thinking about how various corals and sponges change with depth and latitude and feature, and this fills a latitudinal gap in our overall study, and it's just the perfect latitude for us. And this feature, I can give you a snap zoom, but there's a whole lot of stuff. We'll just wait. Yeah. Literally waiting for the snow to clear, the marine snow that is. You said you've never seen snow, but you're like, you get to see marine snow before you see actual snow. What do you think about that? Right?
Since this is my very first time to see the marine snow, and I never seen the snow in real life, so I was thinking like probably it might be the same. I have no idea.
It's pretty cold down here too.
Okay.
Okay, let's get some focus on this thing, and then I'll try to get you a better lateral view, Steve. At some point. Here we go. All right. It's a tough angle to get you the shot you want. Is this
Those
gonna
are
be
pretty
sufficient?
creatures. What are those things? I'm
Which?
not- Like the
red?
animals, like
The
they
red.
stick to the.
Yeah, it's one big animal. Hang on, I'm trying to get focus. Ooh, here we go. There we go. See, here's the body, here's the mouth, and all the rest of these are arms wrapped
Oh,
around this
yes.
coral. So we're not going to take the whole animal, we're just going to take a tiny piece of one of the arms and a piece of one of the corals. And then the coral will grow back, and the sea star will actually be able to grow its arms back too, which is pretty amazing. But,
Ready for sample.
What?
That doesn't mean he's gonna like it.
yeah, we're ready.
You ready?
Yeah.
Can I take control of the camera?
Do it.
I promise actual snow isn't as gross as this.
Huh?
I was laughing at
Whoever
that
thinks
comment.
actual snow isn't as gross as this has not seen snow after it's
I
been sitting
promise
for a couple
actual
days in
snow
a
is
city.
not gross.
So you want the piece to have.
A piece of the oaf
piece.
and a piece of the coral. So maybe it might be easier to grab this piece of here. I don't know. Wherever you are, here might be easier. It's not a very big coral. I'm looking at the lasers down here, right? So that's how I know it's not that huge.
Just any little piece, right?
Any little piece, as long as it's got oaf and coral, we're good. So here, Anamata, you can see here there's more sea cucumbers in the back, right?
Yeah.
Yeah, so there's this funny, like, purple translucent color. They're really beautiful.
So they have punches of colors, like can be purple, can be whatever.
I know, it's amazing, right? So there's all these animals live in the dark,
Mm-hmm.
and there's never a light shine on them. This is, I promise you, the first time they've ever had light shine on them before. And but yeah, there's still all this color in the deep sea, and it's one of the big mysteries out there is why there's still so much pigmentation and still so much color in the
Exactly.
deep sea, right? Some things are just host pigments. Some things are, we know a little bit about what's going on. It's just more to do with their evolutionary history. But a lot of times you just see this brilliant color, and we don't really fully understand it yet.
So you have it.
Yep. Yep, I am. Steve, you good?
Yeah, yeah.
Perfect.
One update. It's based on the way that the polyps have been retracting. It's actually an Acanthogorgia, not
Oh.
a so it's a different family
It is an
entirely. But
Acanthogorgia.
it's an interesting observation from a coral perspective. I'm not sure about the associates.
Yeah, for sure. So it looks like it's Acanthogorgia. Oof.
It's really,
Oof, oof.
spongy.
It is spongy.
Don't wanna rip the whole thing off.
Poor
Nice job.
leg. Poor leg.
Put it off the top.
Just a haircut.
I can see the other sea cucumber on the other side as well.
Let's see. Yeah, exactly. So this sample is a really cool one. We're really excited to look at it tonight. This sample is going to be used to look at the basal metabolism of this coral.
What quiver is that?
It's going into quiver
16.
16. So we're going to figure out the metabolism of this coral. So for those of you who are wondering, how fast it's going to take to regrow and how much food it can take, it all has a lot to do with the metabolic, and sort of energetic. Sorry, I'm getting distracted watching this go in. Sort of the energy and the metabolism of these corals and how they do what they do in the deep sea. It also has a lot to do with food availability. And really, people haven't looked so much at the
Perfect time to do it.
metabolic respiration of deep sea corals before. And so we'll take a look at this one and get a sense of its basal metabolism tonight. In a couple hours, we'll know. That'll be pretty cool. And this ophiroid and this coral both will be worked up taxonomically and will really contribute a lot to our understanding of the biogeography of these species in these spaces.
Anything else you want to look at?
Nope, I think we're good, unless you see a good rock.
Do you have the basket map there? I can fill it in.
Yeah, why don't you fill it in? Sorry, I did not do that. I am the worst.
I'm
I didn't-
sorry. Is there anything that I can help with?
Okay. Yeah, you can, if you have a pen, or you have a checklist and a basket map. So it's the quiver 16. Okay. And that is, what's the sample number on this?
021.
FK21021. It's an Acanthogorgid. I'll show you what you're doing. Plus oath.
Just 61, which is that new table of the
And we're going to, so we're going to start, this is called the basket map. This matches what's on the ROV. And we're going to keep track of everything that's here so that we can, it's easy for us when it comes up to know exactly what's where.
Okay.
You want to
Just let
keep
me
track
know
of
and
that, that's great.
I'll write it down.
Perfect. Yeah. So for those of you wondering about the conservation context, we're in high seas waters, which are areas beyond national jurisdiction, or ABNJ waters, and these seamounts are not owned by anyone. And thinking about their conservation or their protection is really hard because there's the mechanism in place to protect the ocean mostly falls within national borders. And so trying to understand what's here helps to really justify the concept of conservation and protection, because before you go down and map it and look at a place, it's really easy to say, there's probably nothing there. But once we know what's there and we can really talk about what we expect to find on different seamounts because of their features and their depths and their latitudes, it helps us to get a handle on which spaces really should be protected and which spaces might play pivotal roles in the ecology or the evolution or the connectivity from one seamount to another. So all of this is part of a larger picture of trying to understand the ecology of the deep sea and also hopefully protect some of it. These rocks are looking pretty good. Find one?
What about the one like almost in the center?
I like this one. Is it too little?
Yeah, that's what I was thinking.
Yeah.
I don't know the size of that.
So we're trying to get a rock so we can age the seamount in collaboration with the United States Geological Survey, or USGS. And again, it's part of the story and history of trying to figure out the geologic history of the space. I see Brian has just walked in.
Do you want relief, or do you want us to go on? We don't care.
I don't know. I'm kind of having fun. I haven't been up here yet. But you want to come back in like 10?
Just report them, or Stockhouse, the Stockhouse, and I'm just standing by an
Oh,
island.
yeah.
Okay. If you're antsy, come on up. Do you want to? Do you want to stay up here, or do you want to go? It. So Alexis Weinig and Brian Kennedy are supposed to be up here right now. They are very patiently giving us a few more minutes for Anamade and I, since the dive just started. But we have different watch schedules. Alexis is a postdoc at USGS. Brian Kennedy is a PhD student at BU in my lab. And we have the world's best team. It's so fun. So let's see, while we've got this moment where we're setting up to collect a rock, we're going to check on the comments and see what you guys are talking about in the chat and see if we can reply. Let's see. So somebody's asking about pressure, and yes, there's no way you could dive out here without being protected by a submersible. So we actually this robot, Sebastian, is an unmanned. Huh?
It's a juice sample. Once we identify a loose rock.
Yep.
Now looking at
There it is.
Do you think the one to the right is a little bit better than?
Yes.
You want this one rather than
is
this
the one
one?
that you were looking
I think this one.
Either.
Do you think-
Either one of these are good.
What? Which one do you prefer?
I think I like this one better, don't you, Steve?
Yep. Yeah. Let's image it first.
Let's image it first before you take it.
Well, once we know it's loose, right?
Yeah. Do you guys want to try and see if it's loose? I think we are imaging it pretty. Let's see. Let's give it a scan. I think it's loose.
I don't want to hit sample in situ unless it.
Right, until they pick it up. All right. It's a good rock scan. You happy with this rock scan, Steve? Okay. I think we're good. Let's take it. So on this ROV, we actually have a ton of cups made by students from around the world, actually, that are doing this pressure experiment to show what happens to a cup when it is brought down to the deep sea. Ooh,
Ooh,
That's
it's
brittle.
Yellow.
it's brittle.
There you go. Wait. Can I ask you a question?
Yeah, go ahead.
Why does the stone was like the rock has a yellow thing inside?
Because it's got a crust and there's some iron in it, and it's got that like yellowish reddish look to it. So it's ferromanganese, and how much crust is on there sort of helps tell us how old it is. I don't know, Steve.
Oh.
It's good. I mean, let's take it, but yeah.
Take it anyway.
Yeah.
Take it it's all crust.
That's a
It's
good
all-
image.
It's all modified crust. But
Mhm.
just take it anyway since we've already logged it as a sample in situ.
Yep. Let's take it anyway then. Here we go in the rock box.
Side of the rock is yellow, which is known as full of crust, and that will make it easier to tell the age
Yeah.
of a rock.
It comes on. It layers on very, very slowly. So if you know how much there is, you can tell how old it might be.
Okay.
It sort of precipitates out of the water and ends up on the thing. Do you want to? All right. In about 5 minutes, we should let Alexis and Brian come up here.
No worries.
Okay.
So 022 was that sample? That?
That was 022. All right.
It looks pretty loose. No, these lock marks are very loose and copy.
So that was the second sample that was taken.
Yep, we took one. Remember, we took a coral and the oaf, and then we took this
The
rock. And the ophrite and the coral are both really two samples, but we'll separate them when we get upstairs.
Okay.
Yeah.
So, question, are we, because we haven't got the depth coming up right now, if you could announce the depth roughly where
Yep.
we are.
We are 1,870 meters below the surface of the sea right now in the Pacific, Central Pacific. We are still in international waters on an unmapped, mapped today, unknown, undived seamount. And this is the first time anybody's ever been here before. Look at that. And you're here with us. That's so cool.
That's pretty big.
Hey, Steve, let's take a look at this. That's beautiful.
I'll bring you up to the end of it.
Yeah.
Just a touch more.
Come in.
All right, and then I'll get you a
Oop.
little bit out.
Oh, I know, I am sorry.
I'll try to hold that.
Right there?
Yeah, I'll bring you in.
Okay. Ooh, let's get some focus on this.
And then another focus is I'm gonna be moving it to.
Okay. Oh, there we go. Beautiful.
Dark polyps should be able to get an idea on this. It's unbranched.
It's an unbranched bamboo coral. I could give you a clade, but I don't know if that's meaningful.
I love it.
That's so
It's-
interesting.
The back conversation back here is, hey Steve, what kind of coral is it? It's an unbranched bamboo coral.
The
I love
problem
it.
is that there aren't a lot of morphological characters that make it easy to
I
get to
know.
a finer level.
It's true. So you can tell that it's a bamboo coral because of these little black nodes that you can see. There's one about to sort of be very clear. You can kind of see them underlying a couple of them under it's kind of in the skeleton right underneath the tissue, the clear translucent tissue. And so you can see these black nodes. So that's how we know it's a bamboo coral. It's sort of segmented like bamboo. But as to which one, it's an unbranched bamboo coral. And we'll have to look at the characters. Steve, do you want some of this? Or are we good?
No, I'm good on this
Okay.
one.
All right. Thank you, pilots.
Do you want me
Do
to?
you guys want to introduce yourselves? I'm sitting next to a birthday boy, so everyone in the room should say happy birthday, J-Rod.
Happy birthday!
Happy birthday, J-Rod.
Thank y'all.
Wait,
What better way to spend your birthday than exploring in the bottom of the sea?
hand down
Yeah.
for Bob Watson.
I just got brain.
Alright, still searching.
What would you like? Coral?
I think we're gonna look for corals. Let's
Start
just keep.
working our
Let's
way
let's
up?
just work our way up. Yep. Let's move. Let's make some tracks. We'll stop along the way for some corals.
I'm coming in closer. I like the-
Unless you see another really good-looking roundy rock. Let's keep going. We haven't really moved yet. We're still sort of where we are. So let's make some tracks. Oh, look at you. You've got the birthday wishes coming in from all over the world. When you're wishing J-Rod happy birthday, tell us where in the world you're from so we can tell him, give him some stats on how many countries and how many cities are wishing J-Rod a happy birthday today. Oh, people want to know how old you are. I don't know if you want to answer that one.
41.
Woo! There you go.
So full wide, I'm going full wide.
Do you want me
Yes.
to go and call Alexis?
No, it's up to you. I think they're okay. So we are really interested in coralivory, so things that eat live corals. We're looking for patterns of who eats which corals, and whether or not that stays the same as we cross different latitudes and different depths and different features. We're also really interested. Let's keep going. We're also really interested in spongivores and trying to understand who eats sponges, because we see very little predation on live sponges, but then again, we really haven't looked very much. Look at that, you got happy birthdays from all over. Massachusetts, Texas, Canada, Berlin, Hawaii, Michigan. Nice. South Australia. This is so fun. California, Florida. That's pretty good, right? That's very good. And a special shout out to Justin McAllister, who I see in the chat. Hi, Justin. Oh, England, Lincolnshire, UK, Nevada. Nevada. All right, let's keep looking here. I'm gonna turn this over to. This is another Stichopathes, right? Yeah. Let's take a look at this. Yeah, let's keep. Unless Steve wants to take a closer look at it, but I'm gonna.
No, we got some good imagery here.
Yeah, we got some good imagery of the last one. Let's keep going.
Should grab one too, because I haven't really known last time.
Oh, focus, 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 focus. Got it.
Same thing.
Yeah, it's the same thing. Let's keep going. Okay. There's a shrimp. Ooh, there we go. Can we take a stop and take a look? Where's Tim? Is he in the room?
Right here.
What do you want?
Metallic?
Let's take a look. I think this is Metallogorgia.
It looks like Ajax. Is it possible to sample this? Would you understand?
Yeah.
Love to get a sample of this.
Okay. We're gonna get a sample with the
Not sure how big it is with the
Wi-
lasers.
with the oak? Tim?
I would put lasers on the corals so you can see how big it is.
Yeah, they're on right now.
Okay, great. It may be a biobox sample, that's what I'm thinking.
Sorry, we turned the audio off for a second as we decided to discuss our sampling strategy here. Tim Shank is really interested in the Ophiroid, and so we were just trying to discuss whether or not we thought we could take the whole Ophiroid and find a space to take this. But this particular species is of interest to almost everybody on our cruise. Ooh, let me focus this here. So, nope, wrong way. Hang on. I'm not getting this to focus. So we're trying to figure it out. Not a secret sampling strategy. We were just calling across the room and thought it would be rude.
There you go.
There we go. That's beautiful.
What the heck? So long.
We can put it in the bottle box.
Yeah, I think we probably should.
I've got good in situ, it's ready to sample.
Do you want it in the RNA later, or do you want it?
No,
No,
just
just
Biobox.
Biobox.
Right.
Okay.
So everything.
Can we try to get a full image of the whole thing before you take it?
Like all the way to the base? Like that?
Yep. And then let's get a close up of the left side of the tissue.
We don't need the base of it for sampling.
Close up of what?
I just want to get a close up of this side, and then we can go right in. Okay. Come on. There we go. There, that's exactly what I wanted. Beautiful. Thank you. All right, yeah. Let's go straight to the base. You don't have to take it all the way to the base. Sorry, I was just taking the picture all the way to the base. You can take just the top. Right? You don't need the base.
Nope.
Okay. Bless you. So you can see that this Ophiroid is hanging out right in the middle, which means that in order to sample this, which we really want, we're going to have to take most of the polyps. We're going to leave the base, and there we go. We're all going to, every single person, a member of the science team is going to be able to use this sample in a different way, which is excellent. So this is going to be put to very, very good use. And we are really excited about this sample. We've been looking for one of these. The ROV pilots are so good. They're going to put this in a bio box. This is going to be sample 23, Steve? Okay.
$5 for or we might be able to fit more smaller ones.
Yeah, we can try. If we get it inside, we'll do small.
Italicordia. Okay, got that written down. So it's going to go right, is it going to go, we'll figure out which bio box it's going to go into.
We're gonna try to fit in the smaller ones. just to
Automatic.
save the big one for you.
Sounds great.
Okay. Which one?
Hang on, we're going to wait and see. So you never know which one it's going to go into until they get it all the way in, because it
Okay.
could move over. So we're going to let them put it in, and then they'll tell us which one it's in.
Which number is the bio box of that?
Gonna be B.
That's B. All right, it's in bio box B.
So
Good.
I'm just gonna write.
You're going to write sample 023, FK023. And you write Metallogorgia plus Ophiroid.
Okay.
All right. So that will be waiting for us at midnight. Dun dun dun.
So exciting.
Yeah, it is. Cute. Couple of them. So the lasers are centered around a cool little Xenophophore right now. Yeah, our pilots are incredibly skilled to be able to pull this off, and then they're taking off from the bottom, and we're going to start flying over the seafloor again. We're about a quarter of the way from our landing point to our first waypoint. Really, it's like picking a hiking trail. So when we made the map, we got to take a look at it and pick a little teeny tiny spot on this incredible, really
Oh, those
cool spot.
little brown- Oh, cargo.
Yep. Do you want to take a look at that one? I think. Yeah, let's get a little zoom on this.
You're good to zoom.
Okay.
Put in auto.
don't know. That's got zoanthids on it. No?
Yeah.
Yeah.
Paragorgia
Paragorgia with zoanthids. So you see how there's, Anamata, you see how there's the pink ones?
Yeah.
That's the paragorgian coral. And then
And
these
the-
yellow ones, yeah, are the zoanthids that are overgrowing it. And then you have this ophiroid in the middle. And then what's this? Hang on a second. What's this green thing? Like light green?
It's a egg case, looks like.
Does look like an egg case. Can we take a look at that? Let's see. Can I zoom? Can we move around?
Let me get a little bit more stable if you want to.
Yeah.
Okay, so what kind of coral is this?
It's a paragorgia. So, what I don't know is what that. It does look like an open egg case. Of what?
That one looks very similar to the Dumbos.
Yeah, it does look like a Dumbo octopus case. What? So you see this right here, Anamata?
Yeah.
Is it already open?
Yeah, it's open. Tim, do you want it?
No, no, it's open.
It's open.
No,
So-
the oath.
So this is a Dumbo octopus egg case that's been opened. So we think Dumbo octopuses lay their eggs on these corals, attach them to these corals, and so a baby Dumbo octopus probably came out of that egg. There you go. I'm gonna see if I can get a little bit more of a scan over here and a little zoom. We'll take a look at the egg case, and then and the corals and the oaf, and we'll move on. You know the oaf already, Tim?
Yeah.
Oh. Pardon me, guys. I'm still getting good with the camera here. Here we go. There, Tim. That's about as close as I can get. That is really cool.
It's beautiful.
It is beautiful. So that's another reason why these corals are so important down here, right? They're not just a coral. They're capturing food particles. They're like this one coral base, right, is home to zoanthids, which is fighting with zoanthids. Let's be fair, but it's got this ophiroid, and an octopus laid its egg here, and it was home,
Is
a
that
safe
an
home.
octopus as well?
No. This is just a brittle star. All right, I think we're ready to move on. Thanks.
That makes sense. That one's
All right. All this, and we've moved very little, but we are about 1,850 meters below the surface of the Pacific Ocean, out in the middle of nowhere. We haven't seen a soul for days. This is, talk about the ultimate social distancing in a pandemic. It's just us out in the deep blue sea. Definitely no COVID down here. You can see some crinoids, right? Got some more brittle stars. I'm on geology watch, still looking for a really good spot. And looking for some corals as we start to climb. We're going to climb up a ridge here, over a couple of waypoints, and then find ourselves, and then hug the top of the ridge for a little bit. I'm going to see if I can see what's ahead here. This seems like Dumbo octopus habitat. Maybe we'll see some. Oh, yeah, I see it. Oh, good. All right, there's a lot of stuff up here. When we get up to the top, guys, we're going to fire a Niskin. Look at this. So you got a little elevation, and all of a sudden, all of this life likes to live up here because this is a great space for food capture and flow and current. You can tell it's a good rock because everybody wants to be on it. There's a lot of stuff going on here. We're gonna look for a while. We've got some. We've got some.
I'm just gonna do a nice little.
Yep. Oh, this is good. Got some Look at that. Is that wait what? That's cool. See the Metallogorgia, and then there's like this sort of one that's kind of moving
What
up
is
along the
that?
way.
Brown thing.
That is this right here is an anemone, and then this is a black coral, and again in the Antipitherians, it's kind of a double branch, and then we have what looks like another zoanthid, maybe paragorgid coral over here. And then this looks like it might be Bathypathes to the left, but I can't get a good look at it just now.
This is a decent photo mosaic target.
You want to photo mosaic this?
This is a decent target. We can
Kind of working
later
on it
do
for
this
you
one.
right now.
It's a decent layout, but it's more. This has a scientific analogy to me here. So I don't see any coral livery. I don't know if it's the best target. But
Well, yeah, there's some ophs in that primnoid, but I don't. I don't think it's coralivery. But
so you want to wait for
yeah. But let's fire a Niskin here.
In terms of him being able to go around this for a photo mosaic, this is a good spot.
I mean, hang on, Brian. Oh, he just left. One of the things that we're going to try is photo mosaicing in the deep sea. We were thinking about this as a good candidate. It doesn't. This would be a good test of the technology. Yeah, we can give it a shot. Let me go get. Can you go get Brian?
Yeah, sure.
Thank you. Tell him yeah. I changed my mind. This is great. Oh, so the long thin one that you guys are talking about is Metallogorgia. We've got a primnoid right in front of me. Wait a minute. Can we take a look at?
You can see.
That is coralivery, my friends. Hey, all right.
Let me rotate around for you. You talking about those two in front of us?
No, yeah, there's one right here. That is a sea star, a coralivorous sea star. It's a little one. Look at it. It's chomping away. I was thinking about the fact that it looked like it had been denuded, and I was wondering who did it. And now we know who done it. Mystery solved. All right, Brian, you want to take over so you can photo mosaic? Right. Anamare, I'll come in back in a little bit. I'm just gonna hang out. Okay.
Can you guys grab the size reference?
Go back up a little bit. Hold it in now.
All right, hello everyone. I'm Brian Kennedy. I'm a graduate student in Randy's lab, and what we're going to attempt to do here is we're going to take a bunch of video and use a procedure called structure from motion video to make a three-dimensional mosaic of the corals and the rock they're living on. That'll let us take a better look at them once we're back in the lab, but also let us get a scale of how close these animals are living to each other, how many of them may have been predated, and give us a spatial reference to understand kind of the spatial ecology across this rock of how this community may interact with the coralivorous. The coralivores may move around, and kind of ask some questions about the spatial extent of the coral predation within this one small community. So what the ROV pilot's doing now is putting out this size reference, which is literally a wiffle ball with a metal plate on the bottom and then what's called a monkey's fist up at the top to let the manipulator arm
Just anywhere.
anywhere is fine with me.
You know,
And so-
Brian, I think this is the first coralivorous photo mosaic in the deep sea.
that is very likely. And so before we put this down, I measured the diameter and distances on this. So we know the metal plate's 120 millimeters and the wiffle ball is 88.2 millimeters across. So we'll put this into the frame, we'll take all of our images, and then once I have this in three-dimensional virtual space, I'll tell the computer the shape and the diameter of these two things on the size reference, and that will allow me to scale the entire mosaic, which will give us a nice reference to be able to measure these, calculate with some level of accuracy the amount of space that's been eaten on each of these corals and their distance from each other.
And when you're done, could you collect the star and some of that primnoid, please?
Sure. All right. So, Steve, would you make a note to start mosaic? So, J-Rod, the idea here is to basically not touch the camera and then fly all the angles. So if you,
Just have the camera set up.
perfect, and we'll use the SciCam.
Oh, you're using that side?
Yeah,
cam,
we'll
not
use
the GoPro?
we'll use SciCam.
Okay. Then let me change that just a little bit.
And I'm just taking a quick look at the chat, and I want to say a really special hello to Lisa and Alan. dear friends of ours who my very
Down
first
about a
big
45 is
adventure in the
probably,
Pacific was with Alan. We went and
accurate,
spent
but
had a
it's kind of,
month-long scuba diving
care exactly what
expedition
the,
to the Phoenix
angle
Islands
is,
protected
as
area,
much
which
as
is
it
in the
doesn't
Republic of
change
Kiribati,
too
which
much.
we're
So
hoping to
whatever
get
you
to,
think is
but
ideal for
borders are
flying
still
and
closed to
writing,
COVID.
I'm
So we're
happy
going to be working in the northern part of the Phoenix Archipelago for sure, in the U.S. waters of the
no.
Pacific Remote Islands Marine
Really
National
from-
Monument.
I'm looking at the
So
things
Alan,
that
I'll be
from
thinking
your
about
head
you as we head towards the equator and
across
think about our
here,
old stomping grounds.
I'm pretty
Love
good
seeing
camera.
that you online here.
it's
And it's so fun. We get to
Yeah, so
join
you
us
can-
now on this
pilot's choice, you can fly
unknown
over it,
seamount.
turn
This is the
around,
very
come back
first
and
time
forth
anybody's
two
ever
lines, or
seen
you
this.
can
We
do
mapped it
concentric
for the
kind of
first time
circles
last
around
night.
it.
This is
Do
the
this.
first
Absolutely.
dive ever on this seamount. And what we're about to do is what we
Yeah, if you just
think
wanna
is
veer
the
left back and
first
forth,
coral
that should be
livery
totally fine.
photo mosaic ever attempted in the deep sea. Not the first photo mosaic, but the first one focused on coral livery, which is the consumption of live corals. Something I've been interested in for a long time, and you can see this coralivorous sea star on the upper left on this primnoid coral, and you can see all of the denuded spines from where the sea star really chowed down. Or a little later, but at least.
So what we do after the dive here is I'll take all this video and extract still images out of it, probably several per second. I've been using about 10 or 15 frames per second recently, and those will give us individual things. And then the program will actually find points in the images that can recognize and tie those together and infer the motion of the camera from the changing of the angles, and then it can do a whole bunch of trigonometry and high-level math and build the 3D reconstruction out of the points it recognizes in the image. It's a really impressive suite of technology that's come a long way in the last 5 to 10 years, going from research lab to commercially available software.
You can also tell that they're juveniles usually when they have polyps along the main axis.
When they move up, that's how you get all the different little tufts. That makes sense.
Steve, do you want me to see log for you?
Sorry, let's do what you're supposed to.
That's fine. Do you want to use your own computer or do you want to log in?
I'll use that.
It is a good candidate, Brian.
So, what I have
Nice shot.
done so far, I've stopped with
I
photo
like-
mosaic, starting photo mosaic.
Okay.
I'm not going to log anything about the photo mosaic unless Brian says something
Yeah.
that's important. But yeah, just making some boundaries.
We can include this coral over here on the far left, J-Rod, too. It's close enough.
The next sample is 24.
You wanted to come under the lip too?
If you can, I'll take it, but
How
it's,
close in do you want to be?
if you can take me as far left as you can get without stirring up the sediment too much.
Yeah, I'm getting there right
Yep.
now.
So
But,
do you
I mean,
wanna
how
include
close
this-
in do you want to be?
Oh, close. I don't need to get particularly close.
Are you trying to include this left
I am.
rock as well?
I'm trying to include that black.
That black coral?
Yeah.
Yeah. Okay.
This,
Yeah, we saw that.
it's a branch in brown.
We saw that last time. It's heteropathes or hexapathes.
I'm just going slow over here.
Yep. So no hurry.
No, that branching is sort of part of the.
And
There's
then
another different branch one.
You might want to go down and
If you
move
can give-
hand back right.
if you can give us one low pass all the way back around and then one over the top, and we'll angle down and get kind of a down looking view as you cut across the top, would be, I think we can call it good.
So what's cool is we have the software on board to be able to try to process this mosaic on ship and take a look at it and know pretty soon whether or not this technology is going to work for us. One of the things we're really interested in is quantifying the amount of coralivory by area removed, and then trying to contextualize that too into how far, sort of what the spatial maneuvering of the coralivores and the corals are, especially in certain areas. And so while there's only one coral that looks like it's really been eaten here, it's a good test of the technology to see if it's going to work for us in later situations.
So it's tough to tell, but
Really different than doing this in the water in Belize, hey?
Yeah.
the one on the left-hand side looks exactly what we call corticotrophic.
In some ways, this is more comfortable sitting here in my nice, comfy chair in my air-conditioned control room as opposed to free diving down, snorkeling.
I can dump a bucket of salt water in your head if you'd like. Bring back old times.
Any subsequent branch.
I'm not sure if advisors are allowed to do that anymore these days. But but I miss being in the water too. It's a cool ledge.
This is unusual.
Just go see before you change it.
The white Corallia, I don't think it's this is the Corallia, Paragorgia branch.
Perfect. Now, if you just want to rise up, we'll tilt the camera angle down and just take a straight pass right over the top. Would be perfect.
I could do another pirouette like that angle.
Up high, that works.
Like this?
Yeah, if we just make sure to get the top of that Metallagorgia in there.
So about like this angle?
Yep, and if you want to back the vehicle back up just a touch.
Get there.
Yep.
Oh my god.
No
this-
hurry.
this-
Just
So about like this, right?
yep, sounds perfect. If you want to pirouette back around, that would be awesome.
This is fancy
Can
flying.
you turn the lasers off for this
Great
pass?
job.
You make it or you got it?
And can you iris up just a touch?
Open
Or open it?
increase the ISO. Yeah, increase the ISO.
Perfect.
Too much, or you have enough?
Increase after that.
You don't have to get closer in.
So audio in there,
It's okay.
video?
You want to be closer to that Metallagorgia? You want to be
If you
closer?
can come in a little lower, that one. If you can lower the altitude a touch.
Coming down slowly right now? Like about there?
Even if you can get even a little closer, it'd be fine. So the cool thing about this is the more overlap we get and the more angles it works. So these wide ones are great, but the resolution on the individual polyps will be improved if we get a little closer. You can just lat left and get that black from this angle too.
I don't know if you have a pretty good. You'll have to get the snap zooms of the Coralivore too.
Yep. Or you want to collect that, right?
I do, but
Yeah.
I don't know if you'll be able to have the.
So like this right here for that glass?
How
Yep.
much of the spatial relationship you have.
Yeah, if you were just, I know you're running the wall left, but if you want a pirouette left on that from this altitude.
I got plenty of room.
Okay.
It's working. Touch wood. Only you. This determines kindly once you code jail.
Oh my goodness! There's a wiffle ball in the bottom of the sea.
Ta-da!
Wiffle ball of science.
Wiffle ball
I
of
like that,
science.
that's good.
I like it.
Any other shots you like?
Oh, I think that concludes the photo mosaic portion of this dive.
That was not very long.
Nope,
That was
it
good.
wasn't. It was fairly small. Alexis, can you log stop photo mosaic? Cool. All right, let's go collect some stuff.
All right, what would you like to zoom in on first?
I want a good zoom on that this primnoid right here. This baby.
The one that's getting eaten?
Yep. And then we're gonna take a little bit that little baby coralivore and some branches.
I was asking what software you're using for photo mosaic.
So we've got a couple questions about what the software is and that we're using. It's a commercially available product called Metashape, produced by a company called Agisoft. And I've used it a few times and been really quite happy with the results from it.
It was really interesting, actually. Brian and I taught a class together at BU this year. And because we couldn't go to the fields due to COVID, we had to pivot and use photomosaics from the shallow waters of Belize to teach the class about coral reef dynamics. It was really helpful to have students who've never seen photomosaics before, or sorry, never been snorkeling in that part of the world before, to be able to use these photomosaics and literally fly through the reef. It really gave us the perspective that we needed. It was a really fun class to teach, even in COVID.
Yeah, it worked pretty well. I mean, we jokingly call these things e-reefs because they are remarkably accurate, and we can get, share them, move them around the world at the speed of light, which is really nice, especially in a year where all of our field work was canceled.
Can you zoom in, Brian, and really scan and get a good focus on this? I'll get the focus, but
Is that going into RNA later?
no.
Here, switch it in. So you can drive the camera and get what you want.
Wait, what is? Hang on. What species is that?
I don't know, yeah.
Okay.
Let's get, zoom in a little bit.
I'm trying. Oh. That's not what I thought it was. Now that we're closer.
Stairs?
I don't know.
Depth is 39. This is really small.
Yeah, it's really small.
It looks like Hipposteris.
Mm-hmm.
Sea star.
Hipposteris, Tim thinks.
Yep.
Look at that margin. I'm hoping it'll kind of wave around and we'll get a little glimpse of it holding on.
See, it's on a bare branch, right?
Well, yeah, but I think it's eating. You can see the tissue, right? Sloughing right from where it was. To the right?
Part of it missed.
All right. Wow, that's cool. Okay, oops, sorry. I went the wrong way. Let me zoom out here. Yeah, let's take some of that. I wonder how it'll hold up, but I guess we'll try it.
What do you mean?
I'm just thinking about in the respirometer overnight.
I mean, it's a promoter.
Yeah,
so
I know.
That's what you're thinking works best.
I know. The polyp structure is really different. Let's give it a shot. Okay, I think what we're gonna do is we're gonna take the branch with the sea star and a couple of the other branches, maybe just 2 or 3. That might take too much. So if we could set up for a collection, that would be really helpful. Thanks. What are you looking at? Fish. Here we go. Hang on. Fish zooms on request. All right, yep. Thank you. Halosaur. Okay. Let's go coraling. All right. I'm going to let you and Alexis do your thing. After this, yeah.
So why don't you
I'll
stay
finish it.
until you get exactly what you want. It's going to be in Q14.
I've got a question about how long it takes a sea star to eat one polyp.
We don't know how long it takes one sea star to eat one polyp. That is one of the things that we are interested in knowing, is with the rate of corallivery. I think it could stay on this coral for days, weeks, maybe even months. So we'll have to see. What are we doing? And get the thing in focus.
You make it? She got it.
I think I'm working on it.
You're going the wrong way.
Perfect. It's little.
What number are we on?
This is going to be sample 024.
You want to image it real quick?
Ye- I think we got some good images. We're good.
Thank you, Sarah.
Gorgeous. You know what? Before you drop it, I want to image the underside.
Gotcha.
I can't do that at the same time.
Focus. Wait. Wait. Perfect. That's what I wanted. But just hold it still for a minute. Perfect. Thank you.
Oh, you're good now.
You can let it go. Yep. And if we could just grab another couple of
In the same.
Yep, same coral, please.
In the same jar.
Yeah, in the same jar is great. Thank you.
Two okay?
You're gonna get a bonus little crinoid in there. Yeah, it's great.
Want the crinoid?
Sure.
Do you want this to have the same sample number?
Yeah, it can. It can, because it'll go in the same bio box. Doesn't matter. Tim, do you want that O farm?
If you can get it, but
If you can get that O farm, Tim will love it.
Oh, you got one on the close.
Oh, it just one jumped into your sample. Nah.
yeah, so the little green there.
You're talking about
E-
the,
yeah, if you can get a little bit of that O farm while you're picking it off.
you're talking about the birth thing?
The brittle star, yep. So, sorry.
Grab the whole sample to grab it.
Yeah,
Then
you might actually.
no, then don't.
You might snip the whole thing.
Okay. Then don't take the whole thing. Just grab
I would-
grab an arm.
With the little snip up halfway up there.
Yeah, exactly. If you can give me I'm gonna ask you though for two or three branches though.
Alright, you need to get this one. That's it. That's the one.
Perfect. Perfect. And maybe one more branch?
Sure.
It's long, or two.
So I guess you can get that down.
Yeah.
Right.
Yes.
you can get both of those, great.
I think that one is cement.
If not, don't worry about it.
Just pull that one. Close it, sir. Put it on that. And bring the torch up just this one. And then rotate clockwise just a touch.
Real fun.
After this, what do you wanna go do?
After this, we're gonna move.
Just another part on this rock?
No, I think we're gonna move.
But we're gonna recover the size reference and then move on.
Yeah, we gotta pick up the wiffle bar. Nice job. We even got the time.
Randy,
Yeah.
Steve wants to make sure you get enough for him to have some too.
Oh,
Do you need more than,
how much does he need?
he needs enough for barcoding, this much?
Yeah, that's fine.
Okay.
Good?
Yep.
Okay, good.
We've got 3 branches. I think that's fine. We only need 5 polyps per sample. Look at that.
That's the bonus sample.
Okay.
The bulk will dull the.
Do you want a water sample here?
Yeah. We're gonna pop a Niskin. Good call. I said that in the beginning and then forgot I said it.
Hey, Randy, before we go, there's a juvenile Metallogorgia, the poodle.
Uh-huh.
On the second poodle, there's a, oh, if you go, if I can just get a zoom on that.
Mm-hmm.
He thought it looked different.
Well, we just gotta grab that.
Yes,
It's
please.
right there to your right.
We just added 2 things to our to-do list on this route.
On that road.
We're gonna image. We're gonna pick this up. We're gonna image the Metallogorgia, the baby one that's got the 4 little poops. It's got a brittle star on one.
Like
So let's take
super
a look
zoom or
at it.
free fly?
Super. Free fly is fine, I think, as long as we get a good look. Otherwise, we'll sit down again. And then we're gonna pop a Niskin right about right around the rock. Thank you. All right, should we go get the wiffle ball signs?
I don't care what order to do, as long as I don't forget it.
I think it's got to be called the wiffle ball of science.
So this one right here, right, the one that's just above the ball?
I can't quite. Nope. No. It's to the right.
Yep, right to that.
It's right next to it to the right.
Yep. It's here's the top of it.
I'm just
It's that little
going right
wispy.
in real quick.
There we go. We just want to take a look at the brittle star that's right on that. So I'm going to zoom in if you're ready.
Good to go.
Okay.
All right, I'll get you a little bit closer.
So
So that's the species that we are supposed to see on wait,
Camera there?
On what? Tim?
wait one, I just wanna see it focus.
I'm working on it.
Right, so that's the one that is supposed to be known worldwide. It's not what we have on the other Metallogorgia, I don't think. So, any objections to sampling this one?
No. If you want it, take
that
it.
possible?
This is a high sample dive.
Just if you clip it right below it, I don't wanna,
Do you want it in RNA later? Do you want it in
no,
a?
just a bio box, I think.
Okay.
If you don't mind.
Do you want another sample? All right.
Yeah, so that's true Ophiuchus right there.
Okay, so Tim Shank is really interested in the ophroid on this. It's a different. We want to image the oph on the full-grown Metallogorgia just for comparison.
It'd be good if we're,
Yep.
if we're gonna sit there,
Mm-hmm.
we're gonna collect the ball anyway, right?
Yep.
You're gonna want that in the RNA?
Nope. Just straight in a bio box, please.
Vamos?
Yeah.
You wanna say some real estate and put it in the same one?
No. Same one as what?
The
Oh. No. No. But thank you. While we're here, we're also going to ask you to image the brittle star that's on this guy
Oh, yeah, I guess so. Yeah.
after you sample, probably.
I mean, this is,
Yeah. Especially when you have things that like around the shred.
When I found the other day, I was like sliding down things.
That
Yeah.
camera's yours. You wanna?
Yeah, Tim, I'm going to zoom in for you.
Yep.
Give you a snap zoom before we. Hold on.
yeah, so
Are you ready?
This may be two different Ophiuchus right here.
He's flying. Stop.
Yeah, so that's the different one, I think.
No, I'm just trying to land out.
But
He's not, yeah.
I'm talking about the opioids, right?
All right, should be good as camera.
Thank you. Here you go, Tim.
Yeah,
It is different for sure.
I think it is. Excellent, excellent.
Okay, let me get you focused. All right, Tim.
Excellent. Great.
Great. Good shot.
All right, thank you.
All right, so yes, collect the other one then?
Yes, please.
All right, we'll collect the one to the right. Thank you.
That's answers. Okay. And this guy here. That one.
You just below the brittle star.
So do,
We don't put it in there.
So you'll.
Yeah, I guess. If we snip it, we could put it.
good question.
Yeah, if we snipped it to quiver it.
I was gonna,
Yeah.
take the top off.
Take top,
Yeah.
Yeah,
Yeah.
okay.
You good with that?
Yeah.
Perfect. All right, that goes to
I think it's too long. See? See?
See
See that over?
Can we pull it down a
It's a nice crown work. See it? It's beautiful.
Yeah, I'll go for 15, so it's going down.
Yeah.
Mm-hmm.
That's not it, is it?
Yeah, it's the, yeah, exactly. That is it.
Is it missing the top of it now?
It's just out of.
No,
there
it's
to
just
go
see
to the
how
frame.
it's
Oh,
wisping
okay.
around.
Yeah. Mine said it. See, must have.
Perfect.
It's escaping.
It's just right. You know how it's showing through these things. Yeah, I'm doing it two pieces. Make sure it fits enough.
Perfect.
It looked like it might have fit, but.
Okay.
Just a bit far out.
That's cool. 50.
About there.
Yeah, that's perfect.
Drawing. Is that cool? 50.
I believe so, but let me double check. Yes.
You want one sample number or two?
One.
Okay. I got it.
Alright, what was the next one we were gonna do?
Um,
Wasn't there one more thing?
Spire Niskin, please. Team. All right, we just have two things left to do with this rock, and then we will move on and make some tracks. We are going to collect a water sample because we will be looking at some eDNA, and this is a really nice high biodiversity rock, and so we'll get a sense of what we can see in the water column contextualized to what we see in the benthos. So that's what we're going to do right now is by punching one of these levers, we can collect a water sample. It'll trigger the release and fill
What volume do
on
you
this
want
skin.
here?
I think we probably want 5 liters for this.
A 5 liter?
5 liter, yep.
So that's,
Number 1, 2, 3. I'm watching. Number 3.
Nice skin number 3.
And I'll let you know if it's higher in just a second. Right, yeah. Three is fire.
That was Michigan
Yep,
3.
and it's sample 26, FKO26. Thank you very much.
Just the one,
Um,
correct? That was 3 that you had.
Okay, we just need to.
So you're in one of them.
oh, right, yeah.
Okay, I'm gonna get up after this, Alexis, and you can come in the hot seat. All right, we're just gonna pick up the wiffle ball of science and call it a day.
Call it a day?
No, call it a rock, and keep moving. Call it a day on this rock. I feel like we've done all the things we could possibly do on this rock.
I'm sure we can figure it out.
I'm sure we could do some more things. Well, we haven't moved very far, but we've done a lot.
Yeah.
So if you are just joining us, we are in the Pacific Ocean. We are, let's see, 1,840 meters below the surface of the sea on a seamount that is unnamed. That this is the very first dive ever done on this seamount, and seamount was totally unknown and to science until this very moment. So we mapped it a couple of hours ago doing the first dive on it, and we're on. This feature is very cool. It actually looks like a sea star, and we're on one of the arms on a teeny tiny piece of one of the ridges. So a really small part of the seamount that we'll get a chance to see today. But even though we haven't traveled very far, we've already seen a lot. So this is the second dive of our expedition aboard Falkor with the Schmidt Ocean Institute. This is dive 420 of Sebastian. I'm Randy Rogen. I'm sitting here with Brian Kennedy. We're from Boston University. And we are headed towards the Phoenix Archipelago, but along the way we're stopping at these really cool, unknown, unexplored high seas seamounts in international waters. Which are really underexplored, and just studying them and looking at them gives us really context for thinking about their potential conservation, as well as what lies beneath in most of these unexplored seamounts, of which there are 18,000 of them in the high seas, ish.
That number, I've seen a number as low as 33,000, and that's kind of a low estimate.
That's
I would
total
say.
seamounts, but 18,000 in the high seas, I think. Right? No, go ahead. What do you think?
Total seamounts are more than 200,000.
Yeah.
Yeah, more. It's got to be more than 200,000.
Those numbers are
They're
lower
wrong.
estimates than I'm used to.
Fair enough. But so even at the low end, that's a ton of seamounts.
Yeah.
But you're right. It has got to be. Tim showed me the map that he's been working with. All right, there's a fish. Should we take a look at that fish? Resting on the seafloor.
snowy here.
Beautiful.
It's a Custeel, I believe.
That's not a Custeel, is it?
I think so.
I don't know. It,
Oh, your ass.
I don't know. It does. It doesn't. I think it. All right.
Yeah, no,
Thank you. Ooh, wrong way. All right. Hey, what's in here?
Yeah, that's a cat stuff.
Is that just nothing? That's just some rock. Okay.
That's not what a geologist would say.
Well,
But it's interesting.
I think that's actually where you're seeing a break in the manganese and crust. So the sediment has been pulled
Exactly.
away more recently. So you're seeing the underlying rock, whereas the black rock is the manganese crust. And so that's an area that was protected, and something has disturbed it, exposing new rock to the seawater without it being encrusted
Yet.
yet.
We got some xenophinophores and a little sponge and another ophoroid and a bunch of stuff right here too. Urchin. Okay. Thank you. Let me get out of here. And there's looks like some kind of primnoid from afar. Do you want to check out the associate on that, Tim?
The opioid?
Yep.
It's just a snap zoom will be fine.
Working on it. Come on. Oof. Maybe. I can't seem to grab focus. Okay. I got it.
It's a spiny arm.
There you go. There's a little crinoid on it too, Tim.
See it?
Ah.
Alright.
All right.
Okay.
Sorry. All right. Alexis, you want to come up?
Brian, does that mean you're logging?
I will start logging.
Fun.
Squimps.
Zeno. Yeah, it is. Cool. We can just make some tracks, yeah.
We don't have a rock yet, do
We don't have a rock?
I don't believe so. See a rock in the rock box?
do have a rock.
We do have a rock, okay.
Cody. Up top. Bridge. Correct. Yeah, bridge.
What does this say? Fish.
Another halosaur.
Oh, I guess. Can I take a snap zoom on its head?
I'm coming here.
Okay. Yeah, I can't focus it.
Looks like a Stickopathes, yeah, black coral.
No associates.
Slider folks are things that not fine.
Another little urchin. Another Stickopathes, black coral.
I don't see any associates, do you?
Nope.
Almost.
Woohoo!
Big
Well-
rock.
That looks like a. Can I take a look at this coral right here? I think it's a. Let's see. Oh, I'm sorry. Okay. Yes, sir.
Yeah, I don't know what that is.
It's a bamboo.
It is a bamboo? Okay.
Yeah, so
I'm just
see
saying just the
right-
polyps. I see that kind of saw the nodes, but the polyps were so big and fleshy.
So it's unbranched. I think that's the first bamboo we've seen. Look at that little chrysogorgia too, right next to it, behind it. There we go.
And then what's this whip right here?
Looks like a black coral.
Black? Okay.
Okay, that's good. Thank you. So first bamboo of the dive. Small but mighty.
Just a little Anthomastus.
Oh yeah. Little soft coral. So Anthomastus' common name is mushroom coral. So if we got really close to it, it would pull its polyps in. We don't need to do that, and it would look just like a little mushroom sitting on the rock. There's also one called Pseudoanthomastus where they have slightly different morphology, and that's hard to tell which one it actually is.
Looks like is that a Syngid?
I think it's a crinoid or a Syngid sea star. Yeah.
Yeah, it looks like
Where?
a chryso right here.
Oh my gosh, it is almost. It's very dainty.
Keep going, or you want me to zoom in on it?
Can we take a zoom on it? Looks like a little more like bottle brush style, but chrysogorgia. Chrysogorgid, shouldn't I say.
Anybody home?
No, it doesn't look like it has any associates. And that Xenophi 4 behind it. God, it like blends in, camouflage. That's good.
Abby?
Yeah.
So we're making our way up a track line that's on a seamount in international waters, also known as the high seas, also known as an area beyond national jurisdiction, in that it's not within any country's exclusive economic zone, so nobody owns this space. There has not been, historically, there's not been a lot of exploration around these types of sites, just because most people are interested in finding out what is in their own waters, and so there's not always the same amount of funding to come and do this really international type of exploration. And so we are at about 1,800 meters, and we're going to work our way up to, what's our final waypoint, do you know?
Waypoint 7 is
1488.
Up to 1488, so covering about 400 meters in depth, over probably around 8 to 12 hours, 8 to 10, yeah. It kind of depends on what we see and how these, oh yeah, stock crinoid coming up.
And then there's a sponge over there. You wanna look at either one or just?
Let's look at the crinoid right now, since it's coming into view, and then we can look at the sponge, because we haven't really seen sponges yet.
I saw one real quick, but it
Okay.
was one stalk sponge. But yeah, I'd like to take a look at the sponge.
So this is we call it a crinoid. It's also commonly known as a feather star. These are echinoderms, so they're related to things like urchins or sand dollars, and they have a very ancient lineage in that they have evolved very, very long ago, and we find them both growing on these stalks like this, and also free swimming. So when they're not stalked, they have the ability to move their arms independently, and they can swim through the water column.
Stalked crinoids are really well represented in the fossil record, and they were actually thought extinct for a very long time until we started exploring the deep sea, and then discovered that these stalked crinoids are very much alive and well in the deep sea. They were just so far away from terrestrial animals that we are humans that we assumed that they were something that had gone extinct hundreds of millions of years ago.
And there's a number of different kinds, so we see, I don't know the taxonomy of crinoids very well, but we see this one, and there's also ones that have almost not feet, if you will, but they have the ability to actually move their stalk on the rock, so to reposition themselves if they need to.
I think we're probably done with this.
Yeah, we can go on to the sponge. I know.
Can you turn that off?
And we have a glass sponge. So these are made of silica, hence why we call them glass sponges, because it's essentially made out of glass. And there is often shrimp that live inside these sponges, and they feed off of what the sponge is also feeding off of, which is what's coming in through the water filtration.
Can we get a tight zoom just on the
Yeah.
structure of it?
Yep.
Can we get a second?
Go ahead.
Just zoom in and I'll center it up.
Okay. Where do you want Brian?
Just a good tight on the structure so
Okay.
we can see
Yep.
how it's woven. You know, amphipod running across the seafloor behind it.
Try manual focus or levels.
I'm trying.
I just switched it
Oh,
over.
yeah, okay, go the other direction.
Okay.
There you go.
There you go. So,
I think that's good enough.
okay.
That's good enough, thanks.
So you can see how intricate the lattice is of the sponges as they build their what's a sponge? It's not a skeleton. What's a sponge
I think it's, I mean,
structure?
the
Is it
spicules are the skeletal
Okay.
components, but I still think I'd call it a skeleton.
the sponge body?
So guys, we're hunting for another rock here at some point that we can pick up.
Okay.
Another black coral, sea cucumber coming up here.
Mhm.
It's pretty much a vertical.
Okay.
Yeah, it doesn't have to be right here. Just if you see
No,
a,
I don't know that any of these would actually be.
I think they're all cemented in, so it definitely doesn't have to be right now. But just to, if you can help us look for something that looks
Really?
collectible.
What about rope? Like, do you want to break off a rock anywhere, or?
If that's possible.
If
yeah.
that's possible,
Oh, yeah.
we're happy to. But that is, I realize that is much harder to do than it would sound like.
You're giving him a challenge.
No, okay. Every time I've ever tried to do it, you end up
We have a-
shaking the vehicle around with the arm more than you end
We've
up
had a lot of
breaking the
people
rock
who
off.
don't want loose rocks, they need one.
Yes.
Yeah.
Yes.
Because they know it came
Yes.
from that area. So
Right.
you can break them with a rock.
Yes, if that is possible, that's what
Yeah,
we
we
would
broke one
prefer to
earlier,
do.
so I think this stuff is.
Okay.
Yeah, it's not super.
We just need a big enough chunk to get the
There's a
crust
small
with the
little-
basic, with the kind of underlying rock underneath it.
Okay. And so when we're, if you're watching at home, watching on YouTube or Facebook, you're seeing one screen, so one viewpoint from the ROV. In the control room, we have quite a few other camera angles, so the ROV pilots have another camera that they're able to see further in the distance. So when they're flying, you're not just seeing what's directly in front of you, but you're seeing what's coming up also.
That's how we always end up on things like this.
There's another crinoid. We don't.
Fuzzy ones.
It is. It has some fuzzy stuff on it.
Probably hydroid or something. I'm assuming.
Maybe hydroids. Yeah.
I might actually get a better image of this.
Oh yeah, there we go. Around the front.
This one's photogenic.
That is nice. Yes, please. Somebody on one of the chats asked what's the temperature down here, and we're
2.6.
currently at 2.6 degrees Celsius. So it is really cold, but lots of life still lives in this space. Most of the deep sea is kind of between 2 and 6-7 degrees Celsius. Every once in a while you have places that have, there's a
Another one of those things.
primnoid, yeah, with a little tiny oaf. You see the tiny oaf?
Keep going, or?
Yeah, keep going. There are places that have more circulation with surface water, and so you get kind of peaks in temperature, like anything that's under the Gulf Stream on the eastern coast of the U.S. The temperature is much more variable in those spaces than out here in the Pacific, where you have pretty homogeneous water at certain depths. These are very bubbly rocks.
Well, pretty,
Yeah,
Is
it
that
looks
what
like there's
we're-
a lot of. Oh yeah, can we go take a? Is that too far out to check
Do-
out
do you
that
wanna look at that?
sponge?
The sponge on the little bottle sponge.
It's like a Walteria sponge up right too.
I missed that one.
there's another big sponge out in the distance too.
And a demo spot. Remember,
Yeah, looks like a.
That's probably if we can just get a good focus on it. I think that's probably all we need.
Okay.
I'll get it a little closer for you.
Yeah. yeah. It's interesting because the stalk looks like not as healthy, right, as the ball of the sponge.
Alright, I'm happy.
Okay, we're good. Oh yeah, another big.
There's another something right there. Bamboo maybe?
Yeah, looks like a, or maybe an primnoid actually.
Okay, keep going.
Keep coming.
You want to tell you one of those?
I'm sorry.
Yeah, one of those. Cool. I'll get you over
Okay.
Bamboo.
I'll bet.
Nice suited.
You're in manual focus, huh?
What?
That
Oh yeah, bamboo. Very fleshy polyps. Looks like it doesn't have any branches, so an unstalked bamboo.
With no associates.
No associates.
Oftentimes we'll see a crab.
It is, yeah, they get out of sync. That's all. Keep looking at it.
I think we're good to go.
Yep.
Come over to spin real quick. Take a nice big in.
Oh, a little
Let's see
frogfish.
Little Chonix.
They're awesome. Give me just a straight zoom.
Whether you can tell it to.
Okay. He's so cute.
So these are in the anglerfish family. And if we could see his face, which we don't need to maneuver for or anything, you'll actually see, a little scale worm too.
Oh yeah.
They have a little angler, little lure that pops out in between their eyes when they're fishing. You might get to see it here if he keeps turning.
And they have a very interesting fin orientation where it always looks like they're perching themselves on top of their fins, as opposed to most other fish that have their fins more on the, that don't use them as walking mechanisms.
Yeah. I think we've probably got what we need from him.
Yeah. There's the sponge.
Could be the boomers.
Another nice glass sponge. Do you want to zoom on this, or do
I
you?
think that's enough.
Okay.
We got enough there.
Alright, we're just gonna be turning some lights off.
Okay.
It's not happening.
There's a shrimp.
Yeah, I'm just gonna keep warming them up. It's 3 also get a bit of toasting or something. Smell.
Two shrimps.
Boomers getting everything done.
Yeah, so it just kept up everything, even though I lost forward like 3. It just, I'm just
I realized we were so zoomed in. Another long stalked crinoid. I don't think we need to spend time on it. Just cruising along, looking for So we're mostly curious about deep sea coral and sponge communities, and then animals that live on the corals and sponges. So those are known as associates. There's a little urchin on the seafloor, and you got a big sea cucumber. Oh yeah, that is a big sea cucumber.
So these guys are basically tridivores, so they just make their living walking across the seafloor, sucking up the sand, digesting the biological material, whether that be, you know, kind of dead biological material that's rained down, or meiofauna, small invertebrates that live in the sediment, and then they just pass the substrate right back out.
That's good. So when we plan these dives, on a seamount like this that we've never been before, we spend the first couple hours, once we arrive on site, mapping the seamount or the feature. So that means essentially sending sound waves from the hull of the ship down to the seafloor, and those bounce back up and then essentially get processed, and are able to tell us what the topography is, or the terrain, likely what the terrain is. And so once we have a map of the seafloor, we can look at what the feature looks like, and if there are interesting slopes, if there's ridges, if there's scarps. We're looking for features that have shown in the past to be where corals and sponges like to live, so where they can get up in the water column, they can filter feed, and not get covered in sediment. So we're seeing a lot of exposed rock here, but can we take a look at that sea star on the right? We have a different sea star that we haven't seen. That's not an ophoroid, is it?
No,
Okay.
it's Neo something, I can't remember.
Okay.
Yeah, but it's a system.
I'm not used to seeing the long and skinny ones down
Yeah.
here.
No, we heard.
Neophthalmus or something.
So what we're talking about is whether this is a sea star or a brittle star. And brittle stars have a distinct central disk with arms, whereas sea stars kind of blur that line. And with these long-armed sea stars, sometimes it's a little hard to tell whether it's actually got a distinct central disk or just the base of the arms.
And one thing that over many years of people studying these systems has been found that there are certain types of ophoroids that live almost exclusively on corals, and we don't see them on the seabed. And so one of the questions that one of our researchers out here, Tim Schenk, is interested in is kind of why and how those relationships happen. Why are the ophoroids on the corals, and then is there a specific benefit for either of those organisms to having that relationship, or is it all just about having that ability to be off the seafloor? I would be really curious do you think that the ophoroids eat what the coral is trying to catch for itself?
Yes, we've looked at that. We
Oh,
found
really?
They eat the same carbon source,
Okay,
right? I think sometimes they pick it off the coral, so they may.
that's what I'm
They
asking.
may nip
Yeah.
the coral once in a while.
Do you guys want to go eat dinner?
Sure.
Sure.
When I can stay up and switch?
Roger that.
Does that sound good?
What's for dinner?
Yep. The chefs on the ship are outrageously good. But we all have to eat dinner at mealtime so we don't burden our our staff to,
So we've been making good tracks.
yep, it looks like it. They are doing well.
Yep.
You got two fish, you got that Alice one.
Did you guys pick up a rock yet?
No. No.
Ooh,
Didn't
another
receive
one
it
of
in
those.
place.
Nothing what?
Didn't see one really in place. Not sure, you know, you
Mhm.
want one.
You guys want to see some fish? Let's take some look at some fish. Fishy dive so far. I saw a really cute one.
Yeah. More like a rat tail there.
That does look like a rat tail. And that looked like a halosaur below us.
Mm.
Okay. There we go. Swim in a frame. There he goes. Okay. There, yep, there it is. Oh. There, it certainly has been. Just looking to see if there's a coral over on it.
I don't see anybody. It's a Primnoid.
No, I think it's a. No, I think it's a bamboo.
Bamboo. Bamboo.
Yeah, it's definitely bamboo.
Yeah, so 244.
Reach any closer?
No, I'm good. Beautiful. Yeah, there we go. Great. Get a good image of that. Okay, thank you. That's great. Mhm. Keep moving on.
And I said Primnoid.
Alright, we're gonna
What?
kill some lights. Just in there.
Perfect.
Another pro isocrinus crinoid.
Yes, it is. It's a big stalked crinoid. It's my favorite one.
Big red one.
Big red one.
Yeah.
No reason really.
Pretty
Except
impressive.
there we go. It's another
Oh, we're
shot of the rat tail.
Getting followed.
Likes us. What do we have here?
I don't know. Cucumber.
It's cute. So it could be a cucumber. Oops. Ah, wrong way, wrong way. Hey, rat tail. Let me back out. I don't know. We're flying too fast. That's why I can't focus.
Am I flying too fast or am I keeping up with it?
You're keeping up. It's okay. Don't worry. Let's. I'm trying to zoom out though, so I can get a wider view. Oh, there you go. This is a neat habitat though. All right.
Another stalk, something to the left there.
Yep.
Not
Let's
sure
see.
what that is. We're gonna look at it. Another,
It's just a stalk.
another stalk.
No. It's got a crinoid.
Another stalk bamboo over there that had nothing on it.
Keep going up?
Yep. Keep moving, please. We are being followed. He likes the lights.
Yeah.
Get out early and go
Could
eat
be.
or stay in for another 15.
Someone asked what's for dinner.
Mm-hmm.
What was for dinner? I haven't.
You want me to say it out here?
Sure.
It's
It's okay.
okay.
People are asking about it.
It's good. It was mac and cheese, and there was like this dill salmon that was really good, and there was like green beans and broccoli and salad.
Right.
The food here is
always
outrageously
really
good.
good,
It's
yeah.
amazing. Peter and Bert, best chefs, so good.
For sure.
I have to say, I usually really miss cooking because I cook all the time at home, but after COVID, cooking 3 meals a day every single day for everybody, it's really nice to
Yeah.
just. We're only a week in, so I'll probably still miss cooking after this, but it's really nice. Look at all these rocks. Yep. Happy birthday, Dad. I'm looking for a round rock. Oops, they're still switch shifting.
Well, we're just switching out seats here for dinner hour.
What'd you say? Yes,
Switching
exactly.
out seats. Don't even know what's going on. We're off the bottom now a little bit.
When you go downstairs, it just the smell of dill hits you. It's so good. I love it.
It was garlic that hit me in the hallway. Passage
Oh,
earlier,
yeah.
but
There's some like it's like a Moroccan fish thing. I don't know. It's really good.
you know, you would think there's so much habitat. You would think here, all this rock, all this hard bottom. You
Totally.
would think it'd be covered with sponges and things. But you know, the current's got to be right too. The depth has to be right, and so not seeing a lot of scouring here. I'm not seeing, you know, I think there was some ripples earlier on, but not seeing a lot of in the way of current here.
No.
So
You know, and there's an urchin. It also just looks like, I don't know, everything, maybe it's just the ROV, but it does look like there's a light drape.
right.
And so it might be,
This could be a sedated bamboo.
yeah, I think it is. Let's go take a quick look here. Yeah. I think so. Can you turn the lights up a little bit?
Yes.
Thank you. Okay. It's hard to tell. It's so washed out. I don't know.
Yeah, I think so.
Yeah, it is.
I
It
think
is.
so.
It's a bamboo.
Yeah.
Thank you. Let's keep moving. So I see some of you talking about, you know, doing exploration and the different kinds of technology. And the tools that we're using here are absolutely outstanding. And just to be clear, the Schmidt Ocean Institute is so incredibly generous with making this ship and and Sebastian, our ROV, and the pilots available to scientists. But you know, we have to write competitive proposals to get here, and so it took us a couple years to get this expedition supported and in action. And we're all really lucky to be here. But there are a lot of technologies. Let's take a look at that. There are a lot of technologies that people are starting to use to explore on their own. So there's some deep sea drop cams, and actually Brennan Phillips, who's going to be on Falkor in a month, is an awesome engineer and scientist who's been building some of those. This is great, good imaging, and there's a bunch of other stuff that's out there, like the Open ROV Trident. I think they're just now called SOFAR Technologies. Can we turn the light down a little, actually? Thank you. Just for imaging. So there's a lot of, you know, and there's a bunch of sort of open source ROV plans that you can look at and build yourself. So there's a lot of ways to explore the deep sea, but basically having a camera like this and a sub like this and a ship like this is pretty much the Cadillac of deep sea exploration. It's pretty much amazing. And of course, having Cody and Jared. I almost said the wrong name. I just looked at you and had a blank right in front of me. It's pretty amazing. This is really, really beautiful. Yeah, it's gorgeous. Okay, I think this is a good. No associates though. I'm looking
Nope.
for a little jelly. Actually, it's kind of what I've been thinking about. Instead, this perfectly clean.
We've seen no associated fauna with these bamboo whips today. Oftentimes there's a crab called Europtychus that lives on this, or Heteroptychus now. They changed the genus. But this is no one home here.
No one home. Just the coral. Is gorgeous. You know, I love corals. They're in the class Anthozoa, which translates to flower animal. And I think when you get these close-up looks, oh, there's something. See, there was.
Yeah,
Can
I
we
saw it.
can we scroll down just a little? Let's see. Wrong way. There we go. Let's see who that is. Oof. Who put me in charge of the camera? That's a bad decision, Tim. You should take over.
No,
Look at
you
it.
got it. It's a dance.
But yeah, flower animal. So clear. Whatever it is, it has those two little wispy tentacles.
Sometimes.
I don't know. Do you want to
No,
care that much?
I'm okay with it. No, I think it's just a.
It's an
I
amphipod?
think it's just a polyp, actually.
No, I don't think so. There was
You
something
don't?
brighter in red. Yeah, let's go around the other side. just take a look?
Yep.
Might as well make sure.
Good, sure.
See that? See, it's got that red little.
I do see amphipods swimming around it. I don't see it on it though.
I mean, just thinking about the skill of you guys, it's unbelievable. Where we're like, can you please spin around this tiny little wispy thing that's, probably, I don't even know how long it is, but, it's super, super narrow. And can you do it at 1,730 meters below the surface of the Pacific on an unknown seamount, please? And you maneuver around this as if we're just spinning around and get us exactly where we want to go. Really, the pilots on the ship are, ooh, here we go, scale worm, are amazing.
Yeah.
All right, let's see. It's up. Let's look at the scale worm for a minute while it's here. It's beautiful. Where'd you go?
Right towards us. So if you
Okay.
come down a little on your camera, we'll be right.
There we go.
There it goes. That's a polychaete worm. Pelagic polychaete. Spends a lot of time swimming.
I think it heard me speaking
Pretty-
about like
pretty
delicate
big.
dancing, and it decided to demonstrate.
There you go.
This is really an appreciation dance for you pilots. All right, let's go take a look at that coral. Let's go see if we can find that little creature, that little associate. I know exactly where it is. Let me see if I can find it.
Think it was a worm?
No,
Oh,
I
okay.
think it was
It's
that.
amphipod.
It's that.
You know,
I don't know what that is.
when I see that at a juncture like that, where it sort of alters the. Do you see it?
I do.
I often think that the coral sustained some sort of injury or something, and it just modified its growth a little bit.
I don't know. It's a different color, which is what's.
That dark thing you mean?
Yeah.
Oh, I
Is
don't
that
know.
just a polyp? Or is it something on it?
Or a piece in the
I know.
polyp.
Did you
Help.
go over there?
Help.
You know, the great news is that we have these frame grabs that are being taken every second,
Oh, it's
so we
true.
can go back to it and look at things.
I know, but they might be blurry. All right. No, come on. This is really hard to do.
You think
I'm
it's
sorry,
one of
I'm
those?
making everyone seasick here.
Sometimes it's just let it go, and let the pilot
All right.
be the zoom or be the zoom anyway.
Can you help me?
Yes.
Save me from myself. Thank you. I just want to see if it's a polyp, and
It's a
what
really
I'm
long
what I'm
coral.
looking for is to see whether or not it's a polyp that's been damaged, like you said, and whether or not it's pigmented, or whether it's something else on that polyp. And I really want to know. My new postdoc, Stephanie, is really interested in. She's a joint between my lab and the Kagan lab, and she's really interested in pigmentation and the role between
There we go.
pigmentation and immunity.
So the goal of these polyps are really to be able to capture particles out of the water column, and sometimes they do trap
All right,
things.
here we
like
go. Here's our look. What are you? What are you? It's a pigmented polyp.
It's a polyp. That's what
It's
I
a
thought.
polyp.
Yeah.
And look at that.
And I'll just cough it up just a little more, and there was
Yeah.
another one.
Dan.
Pretty good.
Wow. That's awesome.
Yeah, it's a good shot.
Can we
Thanks.
get, we
Thanks,
can't
guys.
get like a careful snap zoom, can we?
You want?
Oh my
yeah, go
God,
ahead.
you're so good. Look at that.
You did?
Did it eat
But
something
it's in there,
red,
isn't it?
or is it just? No, I think it's just damaged. Well, I don't actually. Tim, take a look at this.
What?
I disagree. I think it's a worm or something on top
Nope,
of it.
that is a collapsed benthic ctenophore.
Is that what that is?
Oh, I guess it is.
I
That's
believe
why you
it
see
is.
that,
That's that
thing
that's
coming off
what-
of it.
Yep.
Yeah. I agree.
Tentacle, yeah.
Collapsed benthic ctenophore.
Yep. So they can
Oh,
suck
you're
up
right.
and puff up, or they can kind of suck their tissue in and get small, and that is the small version with one tentacle out.
That's why we see
That makes much
those
more
tendrils
sense.
coming off of it.
Yep.
Part of
That makes much more sense. Predator?
Poor polyp.
Don't think so. I
Don't
mean,
think
that's
so.
its feeding tentacle right
Right.
there.
Nice shot, Greg.
Off and
Just resting.
Really
It's just
nice.
a resting space.
There
I
you go.
love that. Well, mystery solved, but holy cow, look at the shot. That is beautiful.
I'll go down to the other one. That's the one at the top of the stall. And then I'll just run down here. There's another one.
Thanks for walking in at exactly the right moment, Brian.
Yeah, I know.
I love those things. They're one of my favorite deep sea organisms.
So it's a tuniform.
Collapsed benthic tunicorn, no less.
Tiny.
Teeny
Yeah,
tiny.
that's tiny.
That was.
It was
the
hard to-
smaller
Pretty
side of
big,
it.
right?
They can,
do.
yeah.
Yeah, for sure.
But
You know,
the
fist-
big
fist
ones
size.
are benthic, generally living on their own on the seafloor, and the smaller ones land or grow. I don't quite know
I've never
their
seen him
life
collapse
history.
like that though. I just don't. That's cool.
But I've seen chrysogorgias with dozens of them, and just the tentacles just radiating out of the chrysogorgias before.
Well, that's pretty cool. There it is. Yep. You get me in? Tight, tight. I don't see the tentacle on this one, so let's see what this is. Is the polyp just closed?
Yeah, I think that might just be a closed polyp.
I agree. All right. Oh my God! Great shot, Cody. Yeah, I agree.
Looks
Great. Beauty shot.
Got it.
All right, let's move on. But thanks, that was really productive, and what a gorgeous shot.
How long do you think that one was?
I don't
Makes
know.
sense.
You always want to estimate the size.
lasers are on. See them right there? Right by the base.
Mhm.
There's the base. It's 10 centimeters.
Wow, the perspective.
That's like a meter and a half, 2 meters.
Oh, you can't even see the end of it.
I can't see the end.
And it goes.
Oh my God, that's easily.
That thing's about.
Wait, what?
They're distancing to the rock on the sonar. It wasn't really a strong scientific interest. It was just curiosity. So we don't need to worry too much about it.
4 meters?
Yep. Almost. On the end of that where the sonar is to that rock is around 5 meters. Curves up. About 4 meters.
All right.
Is that an encrusting sponge? What's that white lower on the rock?
I think
To the left?
Yeah, on the left side. Yep.
I think it looks like an old base or something
That
that
might,
broke
yeah,
off.
that might be a base.
I think that's what it is.
Shrimps.
Cute. Charismatic.
Yep,
Yeah,
that's
the
a
old
base.
base.
All right, that's good with that one. Thanks.
Here we go. Plexaurid. Hard to see. There's a Metallogorgia too, with a shrimp in it. I think. Steve, you want to get a close look at this?
No, that's Acanthogorgia. We collected that.
Oh, that is Acanthogorgia. Hang on, I need to get a close look at it so I can tell the difference. And I need to zoom. Yeah, you're right. Okay, great. There's something behind it. You see that?
Yep, there's another little crest-looking thing.
It is a chryso with a little crinoid on it. And there was a Metallogorgia to the left, which we should take a look at and just image the O for Tim. It was kind of in the left and down. Yeah. I know it had a shrimp on it. That's what it was. Crab, something.
Shrimps.
You think? Are you sure?
Yes.
Okay. You only see one. Do you see two?
No, just one. One
Oh,
shrimp.
Okay. You said shrimps, so I was looking for the second one.
Sorry.
Can you give me focus, guys? There we go. Perfect. All right, thank you. And let's keep moving. Keep on, keep it on.
The Jedi is going to eat also.
Cucumber. I can't. Sorry.
When did we get the status of the fatal
When?
So that's just Facebook, isn't it?
Yeah, the reason we use that is because it's much better indicator
Than the YouTube.
yeah, because the YouTube is almost useless.
I still feel like we should pick up a round rock. If you see a good one, let me know.
Yep. Got a black coral off in the top left.
I thought that was a stocked crinoid.
That could have been. It's possible.
Let's take a look.
Oh, you're right. That's a crinoid with hydroids on it.
Yeah. Little cucumber to the
size
left of it.
that head
It's so
is
little.
disproportionately compared to thing. I wonder if it's been bitten off and then is regrowing.
Regrowing. There's another little crinoid in the base of it.
Okay, don't worry about it.
Cute. Wait, there's something on the crinoid too. Like a little barnacle. See it?
Yep.
Life upon life upon life. All right.
Okay.
This is one of them.
Cody, do any of these rocks look pickable?
Wait a minute. Something definitely took a munch out of that.
This stuff's pretty angular.
Can we take a close look at the head of this crinoid, please? Guys? Thank you. Rhinoidophore? Who is eating you? Anybody see a culprit? Fish, maybe?
I've seen
Swim
this
by?
fairly commonly seen one or two arms missing or what looks like a head regrowing. I have no idea what's doing it, but I don't feel like it's all that uncommon to see an arm missing.
This one's relatively recent. I think the other ones in the bottom are regrowing, but here you can see the open wound, which is what I'm really interested in looking at here.
You might have a little more image.
I do. A lot more zoomage. Wow.
Shot there.
Yeah, when you get done with that, come out and just frame the whole shot and we'll get a pretty shot of it.
I'm looking at the wounded area on the right in particular, just so you guys know I actually have a target here, and then I'll zoom out and get the glory shots. Look at that. Looks like there's some snipping on these too. See?
Yep. This is a little
And who's
little
this?
amphipod or polycute or something.
Yeah, exactly. Who are you? Wow. I love using this camera as a microscope. It's amazing. Oh, focus, focus, focus. Come on. Ah, I was so close. Can you help me focus, guys?
Of course.
Thank you. What is that, Brian?
Can't tell.
Look at the wounding. Okay, well, shall we get our glory shot? Since we're here? This might be our glory shot. This is beautiful. Oh, you know what? Hang on. Cody, Jared, there's another little one of these.
There's two.
And there's two. There's another one here.
Okay.
I'm gonna take a close look at them while we're just all the way zoomed in. That's all I've got. I've got no more zoomage. Oh my God, it's beautiful. Well, I don't think they're eating it, but or at least not. They're not eating. They're not the ones making the large wounds. Here we go. Oops. Let me go the other way. Actually, here. I think this is them. That's the shot.
I'd hold there for a second. Just let it float.
Beautiful.
So what's that? That's still crinoid.
It is a stalked crinoid. Crinoid.
Tomato.
Tomato.
Potato.
We have decided that red crinoids, Jimbo, who's in the room, who's our fabulous marine technician, thinks that they're pronounced crinoid, but he's wrong. It's crinoid. He also said tomatoes or tomatoes, but he's wrong.
That is tomato.
We could call this a red tomato crinoid just for you. What do you think? All right, let's move on. But this was great. Thank you very much, guys.
Dropping them over. Alright. So you're still looking for a rock,
Still looking for a rock.
right?
These are a bit angular, as Steve pointed out, but
You want a circular one?
We want a
square
round
one?
rock. We're very picky.
Fine. We'll find one. Make sure it's over.
Or you can just keep moving. But if you happen to see a round rock. Here, like there are a couple. There's one.
it doesn't. The roundness is. It's more the texture of what's
But
on
some of
it.
these are like bumpy and
Yeah,
like that one, right? Isn't that the right thing?
that could work.
Can you put like any of these? This one to the right or the left?
But how much vertical have we moved?
Can you see,
Not
can we put
much.
lasers on to
It's our
see how big
last
they are?
rock sample.
Yeah,
They're 100 meters.
So,
There was a conversation in Slack that the first rock sample wasn't all that awesome.
Yeah.
So
It wasn't.
I didn't see it.
It wasn't.
Okay.
It was crumbly crust without a rock underneath it.
Okay.
we'll take one for each.
I don't see the lasers. Why am I blind?
I'm just tumbling on the mountain here.
Don't fall.
I'm not touching. Shoot you right.
All right, Cody. I like this rock right here, or this rock right here. Your choice. Captain's choice. Pilot's choice.
So that is
Too big?
it's pretty big.
Pretty big.
What about the one to the right? There you go. Right there. This one.
Which one?
This one right here. That's
That looks pretty well attached.
I don't think so.
It goes into
Maybe I'm wrong.
this right there.
Oh, maybe not.
So those rocks are on top of it.
And then would this suit our needs, or is this too angular?
No, I mean with all that bumpiness, but I think that's about the same size. I don't think that's much smaller.
I know, but if it's unattached and that size, it's perfect.
Okay.
Or this one. You have lots of options right here. This one also works.
That one, I can mess with that.
This is the rock. So I see some of you asking whether or not it's rolled down, and It is possible,
Give it a little push.
probable even, but it's still, I think, kind of it tells us something about the age for the area, which is sufficient for our needs.
Yeah, I mean, knowing where it came from is good, and so these talus slopes, it definitely came from higher, and so this is not perfect. But we also don't know exactly how old this talus is, and so if it's been here a long time, because the crust comes, grows in the order of millennia, many millennia, but you can get things.
Will you give it its next
Let's
sample
get it.
number
Yep.
in the sequence?
Yep. Kind of take a look. Ooh,
Broke a bike jar.
all of them have been so flimsy.
It's not brittle anymore.
Yeah, why not?
You pluck off in, kind of swim by.
It's really not good. I don't like this.
Ooh,
thick
yeah, that's
area at
perfect.
all.
All right, I think since that fell apart that much, I'm not.
You just had that little very tip of it.
Yeah.
Right.
We'll get a nice zoom in on it. Take a look at it. Give it the next sample number.
This is gonna be.
So we have a question. Is it important to determine if the rock-
26. Sorry, 27. 27. What?
Is it important to determine if the rock came from this depth or if it might have rolled down?
Yeah, we just chatted about that. It's a really good question. We think this probably did roll down,
Sure, crust.
but we're still, we're on a very steep slope, and it's hard to find rocks that are unattached. So we're working with what we've got for now. All right, that's great. Thank you.
Still want that one?
Do you want it, Steve?
I'd have that.
Yeah, let's grab it.
Happy with that?
Yep.
Take it.
You're consent student.
Yeah, want the sample ID number one more time, 27?
Randy, what number is this?
27.
This one had a Xeon 5.4 on it, so we can distinguish them, right?
Yep. It also has a really different shape.
Fortunate.
Okay, wiffle ball of science. Just saying hello.
Hopefully it survives. It's a trip in the rock box.
Doesn't look like that heavy of a rock. All right, well, Alexis is back, so I am going to turn it back over to you two.
But if you guys want to start heading on to the next waypoint while we watch change, that's fine.
Alrighty. I'm on a cruise.
Okay.
If I find something cool, I'm gonna stop.
All right. Happy diving. Did you eat, Steve?
I saw an attempt at a rock.
We got it, but
Okay.
it's a mac rock.
It's a man rock.
We'll let the geologist sort it out.
Are we? So did we collect the rock or not?
We did collect the rock.
Okay.
We made rock 20 sample collection 27.
Okay. Where are we? We're almost to waypoint 2.
We're moving.
Arr.
Cruise.
Very angular rocks.
Yeah.
Earlier we had very round rocks.
Question, why do you want a round rock?
So, what you end up seeing is, as the crust builds, you get what the geologists call a botryoidal texture to it, and so it's the edges, the angles, the sharp lines kind of dissipate as the crust builds on it. So you get these old crusts that have kind of rounded edges, or old rocks with rounded edges generally mean they have more crust built up on them. Is kind of the idea.
So think round equals oval. So that's
A little bit. That's the very broad kind of generalization.
It's a sponge.
anybody home? Hello?
see anybody living in there.
It's got a couple of smaller sponges. Looks like it's growing off the base.
Yep.
I think we've got what we need on that one though.
Is that the only other collection since we were gone? Is
As
the
far
rock?
as I'm aware,
Okay. That's
yes.
what it looks like.
Found it.
How steep is it?
Pretty steep.
Oh, there's a little Anthomassus.
Yep. See, Jimbo, how this rock right here has got this more rounded texture with the little knobs on it. That's kind of more what we've been ideally looking for.
Is that what botryoidal means?
I think so.
Okay. That's what I've always interpreted it as.
Likewise.
Okay.
I don't think I can give you the textbook geological definition, but that's how I've always kind of interpreted it.
Oh, that's an interesting fracture.
Looks like we've got, is that a Persimmon?
Oh, a nice
and
big
here we go.
Iridogorgia. This is a.
Surprise.
Thanks, Cody. Did you know it was coming? You saw it in the sonar.
I saw it.
Oh, cool. This is a type of Chrysogorgid octocoral. So, the common name for these is a golden coral because the skeleton has kind of a golden sheen to it. This is the genus Iridogorgia. Steve, do you have a species for us? Iridogorgia magnispiralis, which we see pretty commonly along these seamounts in the Pacific. So it's beautiful, but we don't have to spend too much time on it.
Get a nice shot at it.
Yeah. We have another couple stopped crinoids. Definitely. I think we've seen at least half a dozen of these today. It's interesting how you have different classes of organisms that use the same mechanisms for. Oh, can we zoom in on that sponge? I think you got spongivory.
Spongivery?
Spongivory.
Is that a scientific word?
It is.
We're making it one.
Yeah, sponge that's being eaten.
Oh yeah. This we want to spend some time on.
We want to spend some time on this sponge.
Yeah, probably run one.
This is the first instance of spongivory we've seen. We've seen a couple situations where corals are being eaten by other organisms, but actually we can't conclude that it's eating it yet, can we?
No, but
Likely. Oh, is that its stomach?
I think we can conclude it's eating it.
That's a great
That's
photo.
really.
I'll conclude if y'all won't.
And so we have a couple ophiroids, which are the brittle stars with this more stringy legs, and then we have this sea star that definitely seems to be eating the sponge or attempting to, where it looks like it has its stomach extruded out of its body to essentially get its digestive juices onto the sponge to start breaking it down before it can use those as nutrients.
You wanna take a look from the other side?
Or can we just take a full shot, like a come out and I can do that. There you go. You got lasers on. Is that wisteria?
Walteria?
Walteria.
I think it might be, yes.
Wisteria is a plant, isn't it?
It's gonna be a collection target.
I don't know.
I'm tempted,
You want to?
kinda.
Okay.
This is the first real spongivorous and spongivore we've seen in the deep sea.
Yeah, that's a
so
good-
I think we probably should. Do we have room in a bio? Yeah, we have 2 empty big bio boxes.
Yeah. I feel like it's also not. So if we took from right below the star, could that fit in the bio box?
Oh, it might be able to put it.
No.
Might be able to. See, how big is it? 10 centimeters? No, I'll fit in the footer. Yeah.
Plan a depth for it?
Oh, sure. Can we put it in the bio box?
Yeah, sure.
Thanks. Let's do that.
One letter for shells.
So, and that's good because there's also ophoroids on there, so we'll get those in the collection as well. I'm really interested. That'll be a good candidate for Anna to do the gut of
Yep. Yeah.
in comparison to the sponge, the ones that eat corals. I mean,
Right underneath, right?
yeah.
Yep.
I'm not sure if it's gonna fall over. I'm concerned to cut it and fall over.
It's possible.
Let's see.
Alright, this is 28.
Mhm. Yes. Oh, where's the running?
I think they've fallen apart on it. Yep, it came off the base, so just grab the whole. No, just it came off the rock, so you can just grab the whole thing. Yep.
Sample number 28.
Yes.
Yep.
Bio box.
Which bio box you want?
What do we have?
Only the middle one has anything, so
Far left good?
Far
pilot's
left is
choice
good.
for.
Okay.
Box A.
Box A. There we go. Got it.
Yeah, you might want to put that stars in, which is good. It is a Metallogorgia. Nicely done.
Great job, guys. Thanks.
How do you spell that sponge name?
W-A something?
Walter.
W-A-L-T.
Walter?
IA.
Okay.
What is it, Jumbo?
Sea
Good
cucumber.
God, Jumbo.
We can move on when you're ready.
Do a screenshot, Jumbo? Yeah. No, you like sea cucumbers. Really? What did they ever do to you? What's sorry? What did they ever do to you?
This
Huh?
kind of the stuff on the left is what I was thinking about for the future. Yeah, if you can break stuff off of that, that's better than picking up the talus stuff. See how this is very much textured on the
Yeah,
left?
so Steve is saying that you hear him. The stuff on the left is what
Okay.
we're looking for, rock wise.
Do you wanna see if we can break something off?
Sure, let's try. Maybe like this knob.
We just got, so if we can't get to anything, that's fine, but
I know, but we might as well.
that's kind of the, you know, what we're going for, big picture.
And then take them over the head. And then that part's gonna come off. But maybe one of these nuts might. Do you wanna get another rock sample right now?
I'm fine with that, I was just making it,
I
Yes.
would.
wait a little bit. But it might be interesting to see if you can actually move this stuff. I kind of agree with you, but we're gonna try.
So we're gonna quickly attempt to see if any of these rocks come unlodged, because we're looking for specific geology samples that will allow us to best date the seamount and do other mineral analysis.
Yeah, that's the ones that are left. Cause that stuff looks kind of rusty. Something that looks like it might have a rock inside of it.
I think that,
Oh, they're pretty in place. Oh, yeah, they're in place. I mean, some of the smaller bits and it's just looks pressed, but
Yeah, I think we can move. I think we can move on and see something a little bit shallower depth.
Okay. Worth a
Thanks
try.
for thanks for trying.
good to know. This is like beautiful, crusty stuff. If you could break it. What's the current? Is there current here?
How much current are you guys feeling flying?
Not too bad. It's coming towards me.
Okay.
But
Just
nothing too-
another,
not
is
too
that
bad.
another Vertigorgia?
It's to southwest. Get a shot of it.
Just a quick, we can, or, let's take a look at that core look there,
Top right?
actually, yeah.
Okay. Get ready for screenshot.
Right into the,
It's a spiral, not a wave.
It's a spiral. Take a quick. Oh yeah, we got possibly a type of Paragorgia with zoanthids on it again,
And the oath,
and an oaf. Yeah, that's the same.
got a good disc.
So what's interesting to me too, Tim, is that the ophoroid, that the zoanthids don't seem to have an issue with the ophoroid and vice versa. Yeah, so you can see how it's
No.
moving the colony at the top, the tips, so we can know that that's a Paragorgia and not a Hemicorallium based on the.
Beautiful. Great.
Right there.
Oh,
Frank, yeah.
You good?
excellent.
Okay.
Was that the shot, Tim?
Very nice.
That's it.
That was beautiful.
National Geographic, here we come. Keep on moving,
Yeah, let's keep on moving.
moving and grooving.
We're about at waypoint 2, yeah?
Hey, I don't.
Yeah, let's do a quick, I'm gonna do a quick, or you can fly in, whatever you want. Okay. Just to look at the sea star on the, or the ophoroid in the middle.
I have not looked at any of those ones from 2017. And,
Nice.
Is that the same type of ophoroid? Give me the simple answer.
I think that's Ajax. That's not. I don't
Okay,
know.
okay. Is that good for you?
Yeah, it's great.
Okay.
I like Ajax disc and Ophiuchus arms. It's getting crazy.
Frank and
Getting
It's
crazy.
getting crazy, Tom. In the oaf world. Oaf.
Yeah, looks like another sponge. It's kind of a strange, bulging
Yeah.
morphology. Do you want to take a look, Brian?
Just a quick look, just a
Quick look.
flyby.
Quick look. Has something on it.
Pretty sure
It
it's
does.
Bacteria.
Yeah.
got two something. It's got a Walter.
I saw it. I got the whole thing. But
A little shrimp
I
up
love
there.
The white oats and it would be important.
Got one oaf.
Like a group of oaks?
And what is going on with?
I think it's just two or more oaks having a party.
Take another one out.
What's the depth of waypoint 6?
We're just above 1,700 meters at
6 is
1,690 meters. We started today's dive at roughly 1,870 meters. We've gone up about 200 meters so far. We're going to attempt to make it up to 1488 today.
Do it.
We're gonna do it?
Oh, yeah.
So much confidence over there.
Close your eyes for like 5 minutes.
Y'all eating yet?
Y'all need snacks?
Yeah, you looks like you got a couple of hanging out.
Yeah. All right, I think I'm good. I got what I
Alright.
need here.
Got it?
We got
Yep.
what we need here.
Big old rocks.
The crusts are coming off my sheets here. Crusted. That's probably why a bunch of this stuff is kind of.
Do you like climb up this ridge or do you want more gravel?
Us?
Yeah.
I think we're into the ridge.
You like the ridge?
Yeah.
Super crusty.
There's a small chrysogorgia. We don't need to stop for it. Lots of little.
Something's coming to your frame in a few seconds.
Okay. Oh yeah. Looks like
Hmm.
a primnoid.
Randy wanted another primnoid at some point.
Do we know? Does she want a Norella or does she
She
want?
said, "I'd
Oh,
like
there's a tiny
one."
sea star on there. Hold on. Am I right? Am I wrong?
I see an oath.
Maybe I'm wrong.
See it? I see something.
So this has
Sorry.
library, dichotomous, dichotomous.
Yeah, there's the oath. The little star there.
So
Trying to figure out what that
this.
spot
Narrow it
is.
down, 'cause I think this was the earlier one.
So, Randy's note said she'd like one more primnoid from a different colony.
Okay. Let's just take a look at this real quick because I want to know.
Rochesville.
Oh,
You want me to come out?
You're fine. I'll
Okay.
get you back there.
Okay.
Rochesville. Yeah, come on, just a little.
Yeah.
I want to set up somewhere different.
Let's do. Steve, what'd you say about what this was?
It's Calotropha, either Angularis or Bayeri probably, but Calotropha definitely.
Okay, Calyptrophora.
Or Calyptrophora. What is it? So I started saying it the other way because I don't like it. It's a harsh syllable. Calyptrophora. Calyptrophora.
there are rules.
Oh yeah.
You're good to do it.
Okay. I mostly want to see what this little chunk over here is.
But I'm still saying Primnoid. I'll never say Primnoid.
Yeah.
That one's hard for me.
Uh-oh.
You lost on it?
Yeah.
Okay. See if I can get it.
Do I need to come out?
It's not just a-
There we go. There we go. See all those Steve and Tim? Do you see that group of polyps in the back?
Yeah.
What do you think that is? Something that the coral grew over? No comment? Okay.
I don't, I think it's like an issue.
You think I'm right?
I think it might be. Like a
Coral issue.
it's a
Coral issue, okay.
Injury, overrun,
Not over a little
all.
bit.
What do you mean?
See how all of the branches are going almost
There is
across,
growth,
and then
yeah.
that much just comes to the back a little bit?
Yeah.
Is that branch? Do you think it might have got hit when it started hurt though?
Yeah.
Maybe.
Or are we doing
We
something
can,
with this?
no, we can head on. I mean, do you want to collect this for Randy?
At some point, we need to get one more primnoid, so we can. Steve, is this at all interesting to you, or we can get something else later that might be interesting to you?
I mean, they're pretty sparse.
Okay, so let's
I'm
go
worried
ahead
about
and
Okay.
not getting something.
Let's grab.
collection, please.
This one is not personally interesting, but I'll be able to use it.
I think if we do like here, is that quiver length?
Make it fit. That branch.
One that gets
Or
the
just
oath.
Yeah, that's what I was saying
Yeah,
too.
I agree.
Okay.
That gets the gall too, or whatever.
Yeah, and it should, at least a couple of branch points would be helpful.
Okay, so Steve, you want it below that hinge, right?
Yeah, that's fine.
Okay. So like here.
Okay.
Frustrates me to not be able to see it on the plane a little bit. But that's okay.
Do we have something on 15?
Yes.
I think so.
What is it?
A metallic gorgia. The coral. Is that so?
Thank you. Corral.
We have something in 14.
What's in
too.
14? You have coral as well?
Coral as well. And then in 16 we have a coral.
Okay. So all the other ones are open?
Yes. We don't want to use 19 through 24 though.
Alright, so let's go for. Yep. 13.
Oh, this is.
Are you squiggling the collection or squiggling the collection or no?
Doesn't matter. Your call.
Wait, you take it over.
Okay.
Just don't want to enter two samples.
Yeah.
Yep. And this is 29.
Correct.
Pretty sure this is Angularis or Bayeri, very, very similar. Just the teeth on one of the scales varies by a fraction of a millimeter.
Oh my gosh. Genetically different.
I don't have much Angularis and Bayeri, but the fella, let me see.
Just curious, right? Nice. That's good.
We'll have to cut it.
Mm-hmm.
Angularis is up here. What do you know? The depth range for that one is only 1,500 to 2,000.
Bingo.
I guess there's something to this whole research thing after all.
FK029 going in Quiver 13.
Correct. Yeah.
Or the distribution found up in California. I think Angularis is more Central Pacific. But one of the things that I'm interested in resolving more is this whole Electrophora clade is just you see you've got these two I think are gonna be I think Clark Eye and Attila, they're all
28 or 29?
29.
Well, select.
That's great. That's the
Great.
That's the problem with so many things, like crabs or something, leprosy spreads, very, let's say, like on the calcium,
Nicely done.
But not even just, not even that, but like the shape, whether it's concave
Yes.
or flat.
Yes.
And I've seen both in the same colony.
Moving on up.
Moving
Nicely
on
done.
up. Another crinoid, stocked crinoid.
Oh, yeah, is it
The species that,
no,
yeah, right.
yeah. But the bamboo is, I like getting up sometimes.
It's helpful.
Man, I'd rather look at the books over there. So it's like, no one else knows that.
CRTC. Power line shaker.
Yeah.
So here's the full phylogeny for most recent Crinoid family. So this is based on both colony shape
Man, we saw that one rock earlier on with all those, the one we mosaicked, and then like,
circular
Pretty, pretty sparse since then.
but
Have
it's crazy how many species were on that one.
We'll
Or
have to-
what's not.
yeah.
Sorry, I keep
really interesting.
say what you want to say.
Phylogenetics.
No, I'm just a little surprised given that the additional marine snow we saw on the way down. I expected this to be
Yeah.
a more abundant community.
Which maybe it is somewhere, right next door. That's
See
also
that as we
what's.
get in this little crinoid field.
Yeah, I know. Lots
This
of
little
crinoids.
pocket of
There's another
crinoids and
Rhodogorgia.
a rhodogorgia.
It's okay. We can take a quick look at it.
Anybody home?
Emphasis on quiz.
Nobody home. Which, is it normal to see associates on a Rhodogorgia?
Sometimes there's
Really?
crabs and shrimp that will get on them.
Okay.
These have had none, right?
Yeah, none.
Yeah, I haven't seen him yet.
That's a lot.
Yeah, there's a coral over to the left.
Some kind of like sword
Or it looks like another
or another
paragorgid
paragorgia.
with those zoanthids. So every single one we've seen so far.
It's quite an association, isn't it? It's in the Atlantic too. It's
The zoanthids? Or you mean the.
Paragorgia with zoanthids.
Yeah.
I feel like it's gotta be like a perfect balance.
Right, does it? It never seems fully overgrown.
Very rarely, but
Right.
yeah. it's-
Sorry, I'm.
No, you're fine. That was me.
Does that mean you have a susceptibility to?
Oh, let me just go in real quick. We'll do it.
Yeah,
See
well,
while we're
there
here.
is a Paragorgia called Paragorgia coralloides that has a very characteristic symbiotic.
What we were saying as we're approaching this is that we often see this association
always
with
called
the
the
pink coral being a paragorgid, so the primary coral that's built the base, but you have that yellow, the yellow polyps are a zoanthid, which is another type of coral that's overgrowing. But you don't really see this yellow coral fully overgrow the pink coral very often. It seems that both of them still have polyps present, whereas other times we see zoanthids completely overgrow like a bamboo coral or a different type of coral.
Is that all with eggs?
I don't. Oh yeah, look.
There's a little green one out there.
It's almost the same exact like orientation as before too. That's as far in as we can go. So it looks like this is possibly some type of egg casing, and it doesn't look very large, so maybe it already hatched. But in the past, there has been found to be a Dumbo octopus will lay its eggs on corals. Tim was part of a team that actually saw that for the first time, and when they brought it up to the surface, they opened up the egg case. They found the baby octopus in there.
Oh, that's cool.
And there's another one down here.
I was wondering about that.
Oh no, I lost it.
Oh, click it again.
Click it again,
Yep, so now you're in manual.
and it'll do itself.
Oh, no, you're in
No,
auto.
yeah.
It just jumped back. Now you're in manual.
Okay, there we go. So another possible old egg casing, and there's another little green bit right at the bottom too. But we're not seeing those on other corals, so maybe it is again some type of very specific association. That's interesting. Okay, I think we're good to
In
go.
the Atlantic, there were 4 corals that would have these kinds of octopus egg bones.
Say that again.
In the New England seamounts, there were 4 different coral types that had octopus type eggs in them.
What were they? Do you remember?
Yeah, I have it in a PowerPoint
Okay.
somewhere. Give me a minute.
No, it's okay.
They were octocorals.
Okay. I mean, we see that too with certain sharks. Sharks will lay their eggs on corals. And it's probably a similar, I mean, we don't know, but it makes sense to think about it as a similar thing of getting that egg out of the sediment. So it's up on some type of substrate that allows it to not be completely covered in sediment, and for that egg to mature and then eventually hatch. And there's been, I mean, work in the deep sea has been historically sparse, so we don't have a lot of evidence for those types of life history, that type of life history information where really you would want to study something over its whole life cycle, but we just don't have the capacity. There's a big Brisingid sea star, and another big, or not too big, but another Iridogorgia.
Can you tilt up the camera?
Yeah. Is that better?
Yeah.
Looks like probably some old bases of some kind.
Yeah, it looks like this rock at one point had a bunch of old somethings on it.
Yeah. And when we say old, we really don't know how old that is. We just know that based on there being something left on the rock at some point, maybe even, thousands of years ago, there was something living there. Well, very tall, yeah. Looks like we have a bamboo whip.
Found out what your bases were made out of.
Maybe. Oh, hello. Even longer bamboo.
Those are called basil fruits, right?
Called what?
Basil
There's the fruits.
Is that
Yeah.
brancher
Right?
a bamboo, a branching bamboo, or is that no?
Sorry, Cody. I don't know what you're saying. Oh, the basal plate.
Basil.
Okay.
That's what they call it, right?
Yeah, or it's like a hold fast.
Well, look at the
we call
big
it.
star on the other
Yeah,
side.
we gotta- Okay, well, hold on. Let's get in a little closer to this. Has like kind of a hitch in it, right? This is another little hot spot, it looks like. We've got a couple different bamboos.
Yeah, can you take a water sample?
You want to take a water sample?
Yeah.
Okay.
It says.
Um, can we take a look at this bamboo over here, and then we'll come around and look at that sea star, and then we'll take a water sample. How's that?
That sounds wonderful.
Okay, Steve, what clade of octocorals, or, bamboo coral?
I-4.
I4. Eloquently named.
Or Isodella. Same thing. Oh, she's-
Oh, they're all facing the other way. See that?
So, some sort of, probably a hymenaster.
Salt water.
Rotate the other way so you can see 'em.
Steve, do you want to see the front of the polyps?
No, that's all right.
Okay.
I can identify.
Then let's go ahead and we can look at that big pink sea star.
So distinct.
Oh yeah?
Not the strong.
But more than you before?
It just might have changed.
Okay.
It's not firm. It was coming from me earlier.
Is it a hymenaster?
It's like a,
It's a sponge too.
super spiral.
Well, yeah, you get closer. It could just be another pterosaur. I'm not sure. It's been a while.
What I have seen before in the Atlantic is like these guys. These types of sea stars are something similar, having like a smorgasbord on a dead sponge. Like absolutely like 6 of them just going to town. Oops.
I found one of those at 7,000 meters once. It was
One of
respiring
these?
like crazy, yeah.
How do you know it was respiring like crazy?
I'll show you the footage. It just opens its the dorsal side there in the center.
Yeah.
That opens up. Where's the stick? Sorry. Sorry about it, yeah. So yeah, that was respiring like crazy. If you want it, you know, pushing water through anyway.
Okay. And now this one's just sitting there.
Yeah.
Interesting.
Not breathing heavy at all.
Yeah. Are they scavengers? Very interesting. Are they scavengers? Do they just eat detritus?
I assume so. Yeah.
Okay.
Just roll over the bottom.
Is right here okay for a water sample?
Yeah.
Okay. Then yeah, let's pop in this in a 5 liter, please.
5 liter?
You zoomed on. Oh.
Yes, sir.
What do
What'd
you
you
want to
say?
see, Dennis?
They got the camera right there.
I'm sorry.
Nope. Just curious. Steve saw an oaf
Oh.
parachuting.
Oh.
I wanted to just tilt up and look and see what coral was there.
This should be sample 30, correct?
Correct.
Keep it right here for you. Pretty clear.
What did we see? We had bamboos.
Bamboos.
Iridogorgia.
Paragorgia.
Yeah.
Iridogorgia.
Got it here. Vitagorgia, Paragorgia, Vitagorgia, bamboo web. Yeah.
Zoanthids.
Cigars.
This one over here. Plus one's this.
What was it? Astroschema was there too.
That's one card.
Cool. Thank you.
Sorry, it was Niskin 1?
Niskin 1 is 0601. Great.
I just wanted to zoom into that. Whatever that is.
You want to know what that
Sorry.
coral up there is?
Just to zoom up here while we're sitting,
I
and
think
then
it's just
and
a
on
dead
that.
base.
Yeah. So
I saw it earlier.
fair enough. And then that thing right there, whatever that is.
Okay.
It's probably a non-living coral associate.
This is a, yeah, look.
Looks
So,
like a
That is a dead.
Gorgia.
a Iridogorgia with no plume. And then
That's all, yep. How close you can get in on that.
That's maxing.
Roger that. Okay, fair enough.
Okay.
Yep.
Okay, are we ready to carry on? Anything else?
Let's take a quick zoom on that
This-
little thing. It's
Okay.
probably a crinoid, but
Yeah, I think so.
it's a different morph than we've seen, I feel like.
Thanks, sorry. Didn't need that after all, did I? Sorry, Brian.
Yeah, has like a purple and a dark purple base, or like at the base of the arms.
Alright, I'm good.
Okay. All right, gentlemen, if you carry on.
Have we gotten any sediment samples anywhere?
We haven't. That's a good. Next time we sit down.
Sounds good.
Okay. Does she have does it need to be from around somewhere that's diverse?
I think she is more interested in areas that have more diversity, so yeah.
Okay. Somebody was asking, it looks like they're saying the picture looks really great. They're wondering if there's been any recent updates to anything. Nope. Just great piloting and expert focusing on my part, I would say.
That is it.
It's all you, Alexis.
Which isn't true. Sorry, everybody.
All you. Bridge Army.
So when we're sampling, you can kind of hear us chatter amongst ourselves about sample. We'll say, is that sample number this or that? And that's because as we do this, we take pretty detailed notes about what we see, but also what. Oh, can we go to the left? There was like
Yes.
that's a Metallogorgia. Got a couple Anthomastus and some type of Chrysogorgid, I think. Let's see.
Is that anemone on there?
On the
The bottom branch? On the Lexard?
yeah.
Tuna.
I wanted to get this one first.
There's still different stuff too.
Yeah. It's tough. So I'll just set it right here because this is a good position. for
Okay.
All of it, right?
Yeah.
Okay.
got kind of a damaged Chrysogorgia. You can see that bare skeleton right by where that brittle star is. That's really golden. So that's where they get the common name of golden coral. No.
Can you get all the way on the central disc, or is that you got full zoom already?
You can't actually see the central disc is on the other side. But that's full zoom.
No.
And there's a barnacle up there.
And,
But I'm just curious about the damaged
well, yeah,
space.
just noticing that the mouth is more or less right on the
Yeah.
the tissueless section.
And we don't see any chirostyloid crabs, which are more common to find in those types of Chrysogorgia corals. We have a couple of these Anthomastus, so mushroom corals. And then up top, we have a nice big is this Canthogorgia again? Yeah.
Yeah, looks the same.
So what makes you say Canthogorgia versus a different type of plexaurid?
It looks the same as the Acanthogorgia we sampled. The
Okay.
Acanthidide.
For those of us who aren't as good at coral taxonomy, what does look the same mean? What are some of your diagnostic criteria here?
Really, you don't know the difference until you see the polyps retract. So I'm
Okay.
primarily looking at the retracted polyps to make the
Like this little one?
distinguishing character Acanthogorgia.
Like these ones over here on the left?
Yeah.
Okay. Is it like the shape of the polyp when it's retracted?
Yes.
Okay. And in a different plexaurid, would it be pulled in all the way?
Maybe.
Maybe. Okay. Great.
Usually. Yes.
That helps.
I didn't even hear that. Can you do a snap on anemone quickly? 'Cause I'm not sure what that is.
Okay.
And so while she's zooming in, we got a question about the sediment sample.
Yeah,
So
it is.
when we take the sediment sample, we're actually going to be subsampling the sediment sample for the microbial community there, looking at the
Thank you.
bacteria
Think it is.
communities there.
Huh?
What do you think that is?
An anemone, but it's facing the other way, so it's closed. I think we're good here.
Carry on.
Carry on.
Interesting factoid shared by Jeremy in the chat about the origin of brittle stars potentially coming from the deep sea and moving into shallow water versus the opposite, which is what we think a lot of these evolutionary histories come from shallow water that move deep.
Another Anthomastus. Iridogorgia. Almost a waypoint. We have 7 waypoints.
6 or 7.
1, 2, 3, 4,
Looks like we're on a ridge, doesn't it?
5. Another
Almost there.
Metallogorgia.
Roisopranus rubrinus.
Somebody had fun naming that.
Yeah. No, that's one of the it's the same.
Keep on moving. Yeah.
It's the brewery Christmas. Those get some guys forget about that name. Rubber.
It's an interesting rock formation.
That's a tall one.
What's the hard bottom on this?
A tall one, what?
Kreiner. It's
Oh,
another
yeah.
tall Kreiner.
There
Alan
was
Rickman.
at the beginning.
Yep.
Yep.
Many stocked crinoids on this dive.
I still think we're kind of in a gully, like for the current, like once you get on top,
Yeah.
it's gonna get a little faster. Although surprisingly, we haven't seen a lot of the fallen debris. So maybe, we'll wait to get up there.
Another Metallogorgia. So we're seeing a lot of the same types of corals along this dive. Metallogorgia, Iridogorgia, wow, one very long bamboo coral.
Any guess on how big that thing is?
What do you think?
I don't know how big this is.
Do you know how big?
Yeah.
2 meters? Is that too tall? 1 meter.
Too short.
Too short.
That was at least 3.
3 meters.
Almost there. It is total. The exact 5 meters.
5 meters. Wow. You
Exact
said exact
rough-
roughly.
Roughly exact. Roughly exactly. Yeah, 5 meters.
Wow, that is very, very tall, long. I don't know how you would categorize coral length. It's like the base of one of these corals. Now that looks more like that.
Rally
Yeah.
type base, yeah.
We don't need to. Sorry. Thanks.
Say pineapple if somebody really wants.
Okay.
Checking with Randy.
We'll take a quick look at it. We don't need to spend too long.
I'll get a face on for you.
Cool. That's perfect.
Yeah, you're going for the yellow Luxord or whatever it is.
Stuff's not easy to find.
No.
It does have an oaf and a crinoid. So this is the other type of crinoid. So we've been seeing a bunch of stocked ones, but there's also unstocked crinoids that just kind of cling on, and then they'll swim away if they're disturbed enough. Looks like the same Acanthogorgia.
This one looks a little bit different.
You can't win.
No.
This is why nobody works on them, because they're looking frustrated.
They are frustrating. So there are many species, there are many different genuses within the Plexaurids, so this type of octocoral, and it can be really difficult as we're sitting here struggling to identify them from just video. So that's where the specimen collections of a couple of them throughout the dive are important, so that you can actually take them back to the lab and analyze them using morphology but also genetics. I think we're good. Lots of particulate.
Stir up the bottom, feed everything for 2 months.
Yeah, right. There's another Plexaurid, yeah. Do you guys want to zoom on this or you want to keep going?
Keep going.
Okay, keep going.
Keep going.
Great, Johnny. Yeah, see that, man?
Do you guys know if anybody has ever tried to date crinoid, like not
Living crinoid?
yes, living crinoids? I'd be curious how old they get. There's another metallic gorgia.
Yeah.
Tim, do you want us to zoom or keep going?
You can keep going.
Okay. Whole bunch of ophroids on that dead thing. Man, so many stock crinoids.
Everywhere.
Should have kept count.
Cucumber.
Cute.
25. 26. They found berries.
Yeah.
Officially passed 2023.
Woohoo! Writing that down. Let's see, so it took us 45 minutes.
Wasn't that bad. That's actually good time. That's a big drop off.
Just cruising along. Oh, there's a umbellula,
Oh,
right?
yeah.
Oh, can we take a look at
That
that?
one, I've never heard someone say that, so I don't know.
Umbellula? Yes, you have.
Big,
We were here in 2017.
like, yeah.
Sticopathes. Sticopathes.
Yeah. Is that what you're saying?
Umbellula.
Umbellula. Sounds like some Cajun name.
Did you want to stick a patties?
Oh, do you want to? Oh, yeah, Steve, you wanted.
Yeah,
I want to the umbellula a second. It's got some weird, gross bottom of the polyp.
She's Sprague's.
Whoa.
Yeah, those are pretty yoky.
That's having a hard time with all that particulate.
Very interesting pigments on the polyp.
Very interesting.
Very interesting. I've never seen it so dark.
And this one's, look, you have like a regrowth polyp on the bottom.
Any closer? We'll zoom on the yoky masses.
Trying, I'm sorry. We're getting there, we're getting there. Wow.
I've never seen one so full of eggs before.
That's what you would think they are, is eggs, right?
Yeah.
That's pretty neat.
That is.
Great, it's ready.
What's, Tim?
Nothing. It, yeah.
I wonder what
It's loaded.
these are. It's orange.
Those go like come out or something?
The eggs. So it's probably in some part of its life cycle where it will at some point release those eggs. But it's not normal to see it quite so packed with eggs in the center. So Is the color more like brown, Steve? Is that what is
Yeah,
different?
it's dark.
Okay. Cool.
Well, if, while we're parked, if the Stickopathes is within range, we can,
Okay. So right over
I
here,
can just be able to slurp it up.
Cody,
The Stickopathes.
is this coral
Barn
something we can
Stickopathes.
reach?
That one?
The stick spiral.
The big orange shift.
Why don't we do that? Let's shift a little bit and we'll sample that coral because that's something of interest. I think that
I was thinking it might be nice.
on blue is really cool. I've
Yeah.
never seen it
I've never
look
seen
like that
like
before.
that. One like that either. That was definitely nifty.
That is. And there's a shrimp. So Steve, there's two different Stickopathes that we've been seeing. Is that right?
Yeah, this is the larger, darker of the two.
Okay. Do you have samples of these from before?
I do not.
Okay.
If possible, can we slurp it by dislodging the base and just slurping it up, the whole thing?
Did we get that?
Suction sample?
So if we grabbed it from the base and then you section the whole sample. Is that what you're asking?
Yeah.
Yeah.
It's small enough, right? It's not gonna get too tangled.
Yes, this one. Say that again, Steve. Why do you want to suction it?
But I thought it would be easier. If they can grab it, that's fine too.
Oh.
Yeah, no.
I think they can grab it. I think it'll go into a quiver. You just kind of shove it. Okay.
Want the whole thing, or do you want to take some good pictures of it first?
Yes, let's take some. I'm gonna. Yeah, I'm sorry. Thanks, Cody, for knowing how to do my job.
I snagged a couple.
I know. I just want to get really
wide.
close
I was
in.
thinking all the way in, right?
They're gonna put it in there.
Alexis, I thought I saw something toward the tip that was in the article or something. Maybe I was,
Okay.
there's something behind it. Just
Never seen it associated with these.
only if they've been damaged or something.
Okay, that's Maxu. So
Not too crazy
we
large.
have good images of the tentacles. Tim, I don't see anything.
Me either.
Okay. All right. Ready to grab.
Ready?
Yes.
We are gonna try the suction sample.
Yeah, we're gonna
Okay.
try suction.
Okay.
So we're gonna make sure it works smooth.
Make sure it's happy.
Yeah, that works.
Yeah. Fill it up.
So Jeremy, who I know studies black corals, Steve said, when possible, check the spines. They could have small conical tubercles near the apex of the spines. If they have those, they should be in the family Aphanopathidae. Okay. Can we? Is that something you could do? Do you have to do that with the surface, or is that?
Oh, we should be able to look at it
Oh, well,
here.
We're
On the
sampling.
ship.
okay. On the ship, you mean? Thanks, Jeremy. This will be sample 031.
Agreed.
pilots are in the process of setting up so that we'll use the suction sampler to sample this coral versus using the jaw of the arm like we have been so far.
So going into jar one.
Okay.
As well attached.
Well, doesn't all stuff do that real fast.
Coming outside the base.
That's our time. Bring it in again.
Switching around.
Steve, do you want to switch to just picking it up, or keep trying with the suction sampler? Do you want to switch to grabbing it, or?
That's their call.
We're happy to use the manipulator arm too, and just grab it if that's easier. If it's too well attached for the suction sampler.
As low as you can get, that's fine.
It's fine if you have to cut it. Nice.
Okay, what y'all we looking for? So you have more rooms to move around.
It's like turning this stuff and undraw Slinky into it. Two.
That's
That- that's where-
there's not helping anyway.
Yeah, that's where the slope is nice.
Cut it to start.
We're gonna try to put in that suction hose,
Oh, okay.
and then we have it off.
Yeah.
So what they're working on right now, since it wouldn't come with suction sampler, they clipped it, and now but it's really hard to put it in the quiver because it's such a flexible skeleton. So they're using the manipulator arm to put the sample near the suction hose, and then use the suction sampler to put it in the jar.
Hose out before.
Sure.
There it goes.
Ready?
And got it.
Beauty.
Well, yeah. You with the
All right, FK 21. Third 031, correct? It's 31, right?
Yeah, but it's not in the jar yet.
Okay.
It's not tangled up in there.
So while they're working on that, let's see a question about our next dive. So we've got about a 200-mile transit after this dive, plus we'll have to do some mapping. So it'll probably be somewhere between 22 to 28 hours after we recover the vehicle tonight before our next dive.
Keep it on, keep that flow going through.
Okay. Yeah,
It might be,
I'm sure it is. They
intertwined.
produce a lot of mucus,
uh,
so it's possible that it's just kind of sticking to a wall in there.
It's kind of,
okay.
cause that clean, but.
You can see a little bit of flush out.
Look at that, you see that white arrow?
Yes.
You can see how the
Number 2 jumps up.
Yes.
So right there, I can grab it, some bullets of
Oh.
slime
Yes.
come out the top.
Yes. That's from the coral. So.
So I'll just keep the suction on.
Yeah.
And, turn around.
All right, let's go
Yeah.
ahead and just call it 31, and
Yes. We'll call it
It's gonna
31.
be either in 1 or 2.
Okay.
it could be 1 or 2.
Yeah.
There was a Caligorgia back.
Really?
Yeah, right here.
Where?
I think it's back behind us. No,
Down and
left.
left.
Okay, we're gonna look down on the left, please.
Back further.
You think it's back further or
I think it's back
Yeah.
further.
back further?
We passed it a little bit.
Was it when I was right up on the
Have
wall
we-
just a couple minutes ago?
These are flip-flopped.
No,
Came-
it's.
We came
Oh.
back down after,
Okay, gotcha.
troubleshooting.
You are right.
Right?
Yeah, right, yeah, okay.
I would just, my suggestion would just cross the top off and just relabel the columns.
They're different numbers though. This has 8. This
Oh,
is
fair.
6.
Okay.
This could fire. Yeah.
Match up rocks.
So what are we looking for? Taligorgia?
Huh?
What are we looking for?
A Caligorgia. That's from noted. Caligorgia.
Taligorgia.
Sorry.
Yeah. Maybe, at least we got a good photo of it. Well, a photo of it.
Do you want to keep doing this? Looking for it?
I think we should go.
Okay.
Alright, let's go.
we can go ahead.
We can go ahead and roll.
Yeah. Keep rolling. I wonder if we can make it to 8.4.
I'll Google it. Was it?
Sunfest down to 1,700 meters. I thought that was sense.
that last collection was of a black coral, which they can be kind of tricky to collect because they are positively buoyant. So when you try and put them into a bio box or a quiver, oftentimes they start to float. So that causes an issue. And then also when you try to slurp them like we did, they are very sticky and they produce a lot of mucus, so they can get caught up in things. And if you saw that one, it's just essentially a straight line with very thin polyps, so there's not a lot of area for the suction to actually catch onto, and so those ones are particularly difficult to get into a sampling container of any kind. But it's somewhere in there, so we'll bring it up with us.
In there like swimwear.
It's in there like swimwear. I didn't know that was a phrase. Is that
Yeah,
a phrase?
that's a new
Cody?
one. I've never heard that one.
More stocked crinoids. What are we up to? Oh, we're up to 1576. We've vertically come up about another 100 meters since Brian and I have been on. Temperature still at about 2.8 degrees Celsius. Earlier it was at 2.6. Not displayed. I don't think. There's not oxygen displayed anywhere, is there?
They can show it. O2 stats.
Oxygen, 100.
About the micromoles. Oh, here we have a coming up. Nice colony of coral. Oh, can we take a look at that?
We go zoom on that?
Tim sees a crab. Tim must zoom.
Well, it is the first
From
one.
Bamboo.
Okay.
See
Oh yeah,
the-
that, I
yeah.
see it now. Yeah.
Sorry, I don't mean that was not meant to sound rude, Tim.
No.
No, I don't take it as
What's the associate, Tim?
it looks like a Europtychus,
Okay.
chirostylid crab.
Your style is what I want to know. Eurotypus.
Europtychus, yeah.
Need a zoom.
We will.
I'm assuming this is like an Isodon. A very large one.
Yeah. Very pink
So that's
polyps.
the bare skeleton.
Okay.
Is this with the associate? Is this a an RNA later? No.
Europtychus.
Yeah, no. I'm asking if this has got one associate and
Oh, look
then
at
a
it.
bunch of not associate things. Is it a target for your RNA later?
Negative.
Okay. You only want oaks or?
Not necessarily.
Okay.
This is great. So this,
Is this a Europtychus?
yeah. So
Okay.
recently we described it in that genus.
Okay.
But historically, this Gastrotychus iaspis looks like. Oh. I'm refrain grabbing or something. It's really great.
I'll get it back in center for you.
Okay.
That's all right. That's
Come back around.
you rarely get to see this kind of a shot. What's our depth?
We're at 1569.
It's an iaspis.
Do you want me to come out a little bit?
Yeah, just come on. Just right there. I'm gonna get some good shots on the ground.
Okay.
Shot
Oh, wow,
real
look at
fast.
that. Yeah.
Took a right turn or whatever, took an elbow when it was growing.
Or I have a feeling that, at some point that's why you get those bases, right? This has become so heavy that it's just being pulled down.
Yeah.
And eventually that colony will probably dislodge at that node.
So the reason why the imagery is so critical is because those spines on the back are somewhat diagnostic. The genus and the species. The real characters are underneath the crab.
This is looking like. Still see it.
You guys,
That's fully.
That's full away.
Yeah.
Okay.
It just comes in like this, so we can't let anything around. No, that's it.
I'm sure if there's a frame grab in there, I've got somewhere.
Okay.
If you guys can't hold it, I totally get it.
Look at him hanging on like that. He's got his legs spread.
The tips of his legs are made for wrapping around corals and hanging on to little claspers.
Okay. There you go.
Nice.
Wow. He's like,
Nice shot. There you go. Nicely done.
okay, you good?
Thank you.
Great little animal.
Isn't he?
Isn't it great?
Isn't he so charismatic too?
So,
Those crabs, I just can't take it.
just an Australian to say there's no cower salads on
Each
this
one
seamount.
has personality.
Huh?
Each one of them has
They
personality.
do. They're each special in their own way.
Thank you so much.
Let's take a look at this real quick.
That one that's broken down over there.
Yeah,
Dr.
that
Seuss
real
doll.
scraggly little. That looks like maybe one of these primnoids that you guys thought you saw earlier, Tim.
What's that?
This right here
Yeah.
looks like it might be a
It is
Caligorgia.
Caligorgia, right?
So it looks like to me.
Well, that does me too.
It's very
It's a bit hacked up. though.
yeah, it's a bit.
Something's going on.
It's been through some tough times. A lot of
Chock full of associates,
sticopathes,
though.
and look at those sticopathes all over the place behind.
Right, so what's the family?
This is a primnoid.
Primnoid.
You can say in the comments possibly Caligorgia. Oh, we got a floater. The
Thank you.
jump shipped Ophiroid.
Where would you want? You want to take a look at it on this side?
Sure.
The size of that colony compared to the size of that face.
I don't know.
It's not hard life.
It has had a hard life. And these are not the. Oh yeah, the ones at the top are. Yeah.
Oh, there's two different types of oafs in there.
Yeah.
Yeah. Just
I wouldn't. Yeah, it's so interesting because the bamboo is right next door,
like it.
but everybody except that Kairos Island
I'm pretty sure I know what it is, but
Yeah. Can we
is hanging
can we
out.
sample?
Would you like to sample
we got,
this?
Yeah, we got a
Is
Coral
that
and
possible?
Associate desire to sample.
Is it possible to sample that for
What
us?
part would you want?
looks like everybody, you don't care.
Okay.
I don't know if this is intrigue me. It's the Ophiroid part that we'd like to get.
Okay, they're gonna try and set up, and we'll see if it works.
It's either good for synthesis or for
I'll go ahead and set
I
it.
need to check the depths on Formosa. Yeah.
This is not being a daily story.
So interesting for a little bit.
You can't work in your branch.
No, you pick wherever, I mean, a few pieces I should be able to identify, a few branchlets.
All the way up.
There we go. Yes, sir.
I don't see how.
Is it?
That's the only one.
So Tim, you've got Ophiocanthids
Yeah.
and another type of brittle star.
There's chronoids, Ophiocanthids.
See this?
That's to the left side. You mean?
Yeah.
Yeah.
You got another one.
Yeah. I bet that's natural schema. So let's.
Oh, there's a snail.
Oh, yeah.
Oh, I see it.
Gotta get this now.
Well, let's just clip it right there.
Right below the snail.
No, right. Well,
Oh,
It's gonna be a fine box.
I think so. It's got maybe things.
That's a big
Is that a
piece.
big piece?
yeah.
Shucks.
We've got BioBox C open still.
I would still not do any. This is too much.
Okay, so where would you do it?
I would
Below
do
the
it
sure.
here.
Fine. That's
Right?
beautiful. Yeah.
Okay.
Totally.
Gets the snail, gets the crinoids,
Yep.
gets, well, we'll see if the crinoids stay.
Lots of.
That'd be great.
I saw.
Well, I mean, I think it was published, with just the paper list, but I know from Stevenson and Winthrop, but are also further south of these islands. So this is, actually it's a northern most record for,
It's 17, 16, yeah, right.
this is the northernmost record for this.
There you go. Range expansion.
Range expansion.
Let's get it.
We're gonna sample this, right, so
feeling
you
it.
see this snail right here? So we wanna sample right below that.
We want to get the snail.
In the bio box. Does that sound good? We have bio
It's
box,
just almost certain.
we have it
that I
as
can
bio
check
box
the
C,
big one.
but the one on the right.
That's a California, just so widespread. Genius? Sir.
Oh, yeah. Oh, yeah, all over it. Yeah, it's all over. Yeah, for most of it.
Is this one right here, okay?
Yeah, sure, just so, there's the snail, so,
So can we go down lower, it'd be easier.
yeah, I just don't want it to be too big for the bio box, that's it.
Yeah, I think. Right here?
Okay.
So I have it?
Yep. You wanna get it? You got it, yeah.
Got it.
Depending on what kind of insurance he has, I might, just use the book.
$32, correct?
Yep. It would be interesting to look closely at where that snail is on there because it doesn't look like there's a lot of that skeleton that's bare.
This is for most of it. Look at this. This is just for show. It's for your vendors. Yeah.
Oops.
Yeah. This is from the ISO camera. It's the best shot.
Yeah, it's cool. But
If you look closely, there's a
multiplayer.
Great.
all of these parasites, gastropods and barnacles.
Yeah.
Beautiful collection.
And it is mutid, mutid.
Yeah.
Maybe not one. It's crazy.
That is great.
I don't remember if we got any of this or not.
Nice
I
job.
feel like we did.
Yeah, that's a good sample. That's a great picture from Giza.
No, they,
Same camera.
some of the best imagery.
Well, when you get it out front, right? Close.
The lake, that was pretty good too.
Well, that's really nice.
This was decent also.
Give you a rundown. So
Yeah.
Here's the sheet so far.
Yeah.
These are the Niskins. That's Sticopathes. They said it's hopefully going to be in one or two. So
right.
we only have 4 things in quiver. The bio boxes are full.
Right. So go quiver.
This is so we have sampled through 032. So your
Yeah.
first sample will be 033.
Yeah.
And then,
Hello.
yeah.
Are you gonna use that?
No constants needs a pen though, whatever the notebook is.
Yeah, because that's his computer.
Who's
so.
taking his? So
I got my computer for C-Lock, she needs the pen for the actual physical notebook. It looks like that's a pen already.
oh.
Oh, that's definitely you who writes in that.
Yeah.
Yeah.
So okay. So have you heard about an off bottom time? We're looking at. We have 2 hours, I think. Let's get C-Log straightened out off here.
Here. Randy said Constance can do this, so you don't
No.
have to and you can talk. No? You want this?
Yeah.
Okay.
Yeah, I'm supposed to.
Okay, I didn't know if you'd want to.
Sorry. Yeah, I think I'm supposed to do this.
Well, I asked Randy and she said you didn't have to, so whatever you want.
Who said that?
Randy.
Probably
Because we have 3 people.
here, we'll go for that.
Okay. Well, I would keep this. If she wants to do that, I guess. It's fine. I have this one too.
Okay.
Have you looked at this before? Do you know? Okay. So this is basically our notebook. Sorry, folks. I'll be right with you. You can get a sense of that the time and the activity that we're doing.
Is it? Okay.
That reminds me of really gorgeous.
So not just
sampling.
sampling,
Not just
It has this
sampling goes in here, but
like corkscrew.
also other things that we're
That's very wavy.
decide of any notable things that we see. So in other words, this is our first one of these kind of corals, our first one of these attempted to sample rocks. So we're at waypoint 2, so we're sort of at that rock of what we did. So maybe you should read through it while we start. So right now, we've been climbing uphill.
Morphology in the bark.
We're headed to the next waypoint. We're heading up slope through 4 waypoints. And the goal is to go on top of this ridge and then turn left and right up along this ridge. See how far we get. There's a rumor that we're coming off the bottom at 10 o'clock. So 2 hours. So
This species Chrysanthus.
do you need a pen? So, I have the basket sheet here. You got the C-Log, so when you see stuff, best we can.
No. I'm also going to be listening to you.
And we'll just
Your expertise, Tim Shank.
You're gonna listen to me. It's clearly a team effort here. All right. So, the next sample will be 33, just so you know.
Yeah, we were waiting to come in until you guys finished 32 because we didn't want to disturb you. It's tense when you're taking a sample sometimes. That was a great sample.
So, I think what we're gonna do is we're gonna proceed on to the next waypoint, noting things that we see, but not really stopping unless it's something different. We've seen a lot of. We just saw Caligorgia for the first time, but we want to continue. If we see something new, we'll stop. We've seen it before. I think we should keep going. If we only have 2 hours left in this dial, if that's what it is, I want to make tracks. Thank you.
So you're good with going fast, flex stops?
Yeah, I'm good with. As soon as I say pick up the speed, it's gonna be, holy cow, look at that new thing. You know how it goes. Yeah. But
We
there's
do
a.
still need, we don't have any sediment yet, so next time we
Well-
see somewhere that's really biodiverse, I think that's Anna's goal maybe.
Yeah. Well, that's fine.
Tracy Gorgid.
It's interesting.
Caligorgia. I know Whip.
Was that?
Philip
Christ
is-
Gorge, wasn't it?
It was Chrysogorgia,
Yeah, right.
but
Do y'all wanna
Nope.
see them slower than that?
No, it's
you're
not.
doing
No,
good.
no.
You're great.
You're fine. No, we will take.
I don't mind like speeding up and then slowing down a lot, like for this, like just
Right.
easily
Let's do
just
that.
pause for a little bit.
Yeah.
I'll just keep
Okay.
moving on unless y'all say
All right.
stop.
I'll try to see him early. Raise this up a little bit.
That was a tunicate. You see that?
Yeah.
We don't
Dead
need-
stalk with barnacles on it. Crinoid, Stichopathes again. So we need sediment still. We need a primnoid still. Another Stichopathes.
What do you mean a primnoid? What's that?
Didn't Randy say she wanted another
Branch
We- but we-
primnoid.
branch primnoid?
I think Brian had suggested that last collection we did of Calyptophora was for that.
Is that right?
For
Yeah.
with the
Well,
associates?
that
I
too.
had no, not that one. That was
Oh.
Caligorgia.
Right. Okay. So that satisfies that, so we're okay.
Yeah. I
And
think
we
so.
are we okay on a rock too?
Are we what?
Okay on a rock.
We do need a rock.
Okay.
That is, the whole lodged in there situation.
Right.
Another black coral. Sea star.
We have two
So we've
Niskins
just-
fired, so there's also a couple Niskins that you can.
Well, I just missed it. We passed waypoint 4. We made our turn now.
Yeah, yeah.
Oh, cool. So we start. This should be us going up on the ridge now.
Okay.
We'll see. So Constance, can you note that we've passed waypoint 4 and we're headed along the ridge?
Another Metallogorgia baby.
Control room, can you guys hear me?
That looked like a metallic gorgia
Yes.
with only the poodle there.
Yeah.
One poodle there and an Ajax in it.
That
That's
means
her
it's a
schema.
Maybe
Maybe. Or
That's
I
what-
don't know.
or less mature.
Yeah. I'm gonna keep my notes.
Bridge, are we?
All of these black corals.
Yeah.
Yeah, at least change
A lot of Stichopathes
Yeah. Lots of Stichopathes.
throughout the entire dive.
Lots of stocked crinoids.
Even when we landed at 1870 meters, there was a Stichopathes there. Something with a crinoid on it, an old stalk of some sort.
Looks like a Chrysogorgia at the bottom.
So I think there's a dip into our last waypoint.
Is there?
I feel like the second to the last waypoint was.
Ooh, look at that little cave under there. See it?
Mm-hmm.
Yeah.
Ah, it's a Stichopathes. Is that alive or dead? That's dead.
That's an old. The old fast is let go on us. It's on its side now.
Ah, another crinoid. Yeah, you've seen a lot of
Dead
these, huh? These
skeleton.
stocked crinoids.
Yeah. There we go.
Yes,
Yeah,
I should.
1530.
No, so we're at 1530 now. We're gonna come to 1470.
1430. Yeah.
1473, I think I heard.
1473 is a shout. Yeah, you're right. It's like a dip and then.
1473. So I was hoping we'd see more diversity on top of this ridge. Can we do a quick zoom on that right there, please?
Oh, there's a black coral over there too.
Over there?
Yeah, I mean this is fine.
Yeah. A lot of dead skeletons here. So this looks like Talagorgia with Ophiuchreus in it. This is what's been known globally. Can I get a little focus there? There we go. Tightly wound arms, really thin, very, very long arms. It's known in the Atlantic, all over the world. except for in this area of Pippa and now this new seamount we've been diving on. We've got this other species on it too, Astraschema ajax. So great, thank you.
I mean, we haven't seen any. We haven't even seen cup corals for sclerotinians.
No sclerotinians, no hard corals.
I feel like we had pretty
Right.
common cup corals last time, at least in our dive. We at least saw some on
Yeah.
a dive. I wonder what makes it so conducive to black corals. Something here. Oh, got an oaf on a crinoid. What's in the crinoid?
So this, a lot more, I don't know, dead skeletons. Not seeing a lot of coral rubble though either. You know,
No.
sometimes you come up and you'll see.
It's pretty vertical.
Sclerotinians.
It's popular, so if you look at this
Yeah.
camera.
Oh yeah.
That is vertical, yeah. Well, some of those cup corals love vertical walls. You know, it's
Oh, yeah.
you know, so it's funny not to see any of them.
Another sea cucumber. They have a very nice color about them.
Marco Taviani sends his best wishes to everyone on the Falcor team.
Hey,
From
Michael.
Italy?
Thank
Hey
you.
Marco.
J-Ron, you got a happy birthday up there.
Where is that?
Casey Hanley?
What scientist from Australia?
Okay.
Oh, look at there's an Iridogorgia on the left there.
Yeah, anything on it? Another Stichopathes.
I don't know.
Iridogorgia at 15, 20 meters. We've only got 50 meters to go
20?
toward to the top of this thing. Steve, is this kind of like a transect, Steve?
Yeah, we.
I know, yeah. Could
I
we?
mean, if we don't stop, we're trying not to stop, right? So,
Yeah, that's alright.
I mean, you know,
Yeah, yeah, yeah.
you know,
As long as the lasers are on, it's all that matters.
are the lasers on?
Yeah, they are.
Try to see. Ah, yeah. A lot of these stalks are dead skeletons.
Steve asked if you can tilt the camera up a little bit more.
Sure.
Just a smidge.
Sure.
Just a smidge.
How about that?
Good.
I'm all the way out.
That's good.
We can, I don't know if we hit the transect on button.
Do you want to record the transaction?
You know what I mean?
Or?
Let's do a transect on. So it's.
Oh, he just did it.
Oh, he did. He just did it.
Thanks, Steve.
Never mind. I forget Steve's logging back there, so
Starting at 7.
you can just chillax.
Kind of. You know, I'm trying to do my part here. I definitely am better than I was 4 years ago in terms of my
Yeah.
ability to identify, but a lot of that is just learning as you go.
Base up here.
And we have a really good team here who sits in the library and teaches me.
Yeah.
So
Yeah, it's a big, no,
a
big hold fast. Yeah, right.
There was a big blow there.
The skeleton's actually gone.
Oh yeah, see that sea star down there?
Yeah.
Transit.
I know.
Or trans-
Or transit, thanks.
I know.
No, we're joking. We're not.
Is it possible to tell when we've gone 100 meters about roughly?
From here, 100 meters from
Yeah.
here.
I'm just
Yeah,
curious,
we-
yeah, how
we
long
can
it
watch it.
takes.
That's to the,
100 meters like over ground.
just like
or
you're doing,
vertical?
just like,
Over,
just like you're doing.
Yeah.
over ground.
Yeah, it's almost to the next waypoint, isn't it? 20, 30, 40, 50, 60, 70. It's pretty close.
It's a good speed.
If we could sort of hold that bottom bottom following altitude like this, then we can look at densities and whatnot, so we're trying to, without stopping.
I'll do what we normally do.
Which is awesome. You've done all this stuff before, you didn't tell me anything. I don't tell you anything, it's amazing. It's like an autonomous vehicle.
Except they're doing all the work.
I know,
So-
right.
There's another Iridogorgia.
Yeah,
They're just so pretty.
just see no associates on Iridogorgia or Sticopathes. They're so, I mean, there's so few. It's pretty steep.
Oh, there's the big ones.
That's a big old song.
Covered in a lot of something.
I'm gonna have to.
We're gonna run right into it.
You're gonna have to come right up.
Look at this.
It's a big crinoid.
That's a monster crinoid.
That's not correct.
Yeah,
Looks like it.
Yeah.
It's branching like.
It is.
that's a different kind,
I
yeah.
know crinoids branch.
Oh, that has
Learn
a
something
oh.
every day.
There's a lot of stuff on that crinoid too, by the way.
That one's different, that's
It is different.
hold on.
That's not the.
Pause for a sec.
Are we pausing?
No.
Keep going. Keep going.
No,
I'm slowly moving.
Yeah, you got it.
I'm just gonna-
You got it.
Get a name for that one.
Yep.
I know I have it somewhere.
Branching crinoid.
Super Sinjin Sea Star coming up.
I think it's a telecrinus or something like
This is a sunstar?
Yes, it is. You know your stuff, huh?
Trying to pick it up.
This is like
Oh,
a
that's
Friella,
like
the
huge.
genus Friella, perhaps.
That's a huge one.
That is big. You have the lasers on it too, right? So it's like
Yeah, oh my gosh.
10, 20, 30, 40,
60, 70.
almost a half a meter across.
Yeah.
I think it might have been 14 arms on it too. Half a meter across. That's a massive percentage. Yeah. Wow. Bit of a whirl. This is just.
Yeah,
It's like the number of Sticopathes are picking up.
yeah, I'm noticing.
You know?
They're getting bigger too.
When you're recording these, are you just hitting Sticopathes or
Yeah.
black coral as much as you can?
Approximately. It's not quantitative.
Okay.
Don't try to do it.
It's just a pointer to where the
Every minute or
data
so,
are.
just write, what's in the
Yeah.
brain.
Okay.
Look at these. 1, 2, 3, 4, 5, 6, 7.
Crinocrinus.
Crinocrinus?
Crinocrinus.
I like it.
Was it crino or?
Crinocrinus, that previous.
That's amazing.
Crinoid.
I don't think I've ever seen that one
with
before.
that was branched.
A lot of ophryoids on that stalk over there. Another what?
Crinocrinid.
Crinocrinus. Says there's several of them here.
These have lots of little predators and things. And
They do.
parasites.
Parasites.
Tons.
Tons. I mean, there could be,
You see a lot of just
Half
the
a dozen.
stalks too.
half a dozen species on these crinoids.
Oh, look at that. The big Sticopathes in the background.
Where? Looks like a Iridogorgia.
Oh.
I think.
Yeah.
Yeah, the world's where they are. Once was. You feeling any current now? Nothing. Yeah. Interesting. Yeah.
Anthoptylum rock fan.
What's that?
Anthoptylum sea rock
Oh
fan.
yeah.
All right. Do you want to stop?
No.
Okay. All right. Keep going. That's a good enough shot of it.
Yeah, no, that's plenty fine.
The terrain's changed too, hasn't it?
Yeah.
Little angular blocks. It's a good run.
There's not a lot of opportunity, not a lot of angle options
No.
here. Let's see, you can crouch down in that corner, that's a shot. And then, well, there's that corner, you can crouch there.
Allosaur, yeah. Some sediment here that's got holothurians. Sea cucumbers.
We're in a black coral forest right now,
Look
basically.
at that. It's a crinocrinus forest.
Yeah, Anacrinus forest.
Holy cow. See it here coming up shortly. We've got about 10 more meters, according to our map, to get to the top of this ridge. And then it should dip down.
There's such a deep orange. It's beautiful.
Similar one. So, slim pickings here if you're an associate. You wonder, you know,
Big fish come up.
$1478 meters.
What's that off to the left there?
Where?
The coral at the,
Is that a coral?
I can't tell.
We gotta keep
That's
going.
fine.
What we got? Is that a coral?
It looks like it could be something.
Talking about that thing off to the left a little bit?
Yeah.
Got a, that guy right there. No.
You see that, Steve?
Yeah, that looks like sponge.
Yeah, it was really fleshy.
So the uphill seems to keep going this direction?
Yeah,
Which is like,
I guess.
you want me to go up?
Yeah, I did, yeah. You know, those, you can look at those chronoids and try to figure out which way the currents are going. They project into the current over long periods. So I've watched them, and they don't seem to be in any particular orientation. So I can't say there's much current here.
Can they move them?
No, not really.
No.
So, yeah. I imagine they can, when they're growing, they can turn a little bit. Well,
Reach the top.
is this the top of the mountain here?
Push.
More stickopathes. This, you guys see a rock here? She said it's kind of in place.
Yeah, I think we might have quite a bit up here.
I'm gonna stop the transect now.
Yeah.
Thanks,
Okay.
Steve.
Why don't
Ooh,
we-
I like the looks
yeah,
of
this
that
rock on here.
sponge. Do
Looks
we
like
have
it's
do we have a sponge yet?
pavement? We do.
Okay.
We got a couple, I think.
Alright, well, there's still more to go. Now we're just gonna kind of cruise
We are going up still.
and then go up on top of this mountain. This is kind of.
You're saying you're up till like?
I'm trying to look for a lip or something that's.
Like this. And then
Break
we're
it
up
off?
here.
You wanna
Yeah,
break something off?
that's the goal is to break something off, of course, but something in place.
That may or may not be possible.
You know, just some slight overhang somewhere or something.
I think that's called a ficus. A sponge. A stock.
So we're at 1469.
Here's a bridge for you.
I'll make
You got something? Yeah. A lip or a knob. that sponge there, Steve? Is that what you're talking about?
Yeah, Caulophacus.
Caulophacus. Remember that one. Caulophacus. Yeah,
Is that a hex?
Yes.
it's a glass sponge.
Yeah.
These little
Are you
wh-
sure we have
these little white things
sponges?
on the rock, are those just bases?
Oh.
Where's that?
One here.
Those aren't cuts, right? Just little white spots, are those just old bases that are broken?
Oh yeah, right. That's just hold fast. Yeah. Where sponges or corals used to be.
Oh, here's another
Are you-
one. I want at least one more. I don't care. But
Another sponge.
yeah. Whatever looks, maybe one with a stem that looks more conducive. That looks like it might be going.
More with a stem and a tulip-like sponge.
Yeah.
Look at them. Like the Caulophacus.
Yeah,
Another
that
name-
one was pretty big though, I think.
another
They had
neo-
those
another
cattail looking
neo-season
ones
one.
yesterday that I think would have been a good size.
Oh, Steve, do you see any rocks you think are interesting?
It's all pretty, and I think, we left the crust,
I know.
so I don't think so.
Well, we're still coming up slope. Yeah, I don't know.
I had a question. What allows these plant-like organisms to survive in these depths? I'm guessing they're referring to the crinoids that almost look like flowers.
You want to answer that one?
It's just nutrient availability and the currents. Just like corals.
There's some more like basement rock here, right? Nothing you.
Yes, but
Yeah.
it looks pretty cracked and angular and not crusty.
Yeah. More crusty rock. Crusties.
We haven't had kind of any sediment yet. Is that of interest still?
Yeah, I'd like some sediment.
Let's get. How about this stuff?
Sure.
Well, I mean this rock stuff here. What do you think? Is there some? Is this not in place?
I mean, it's in place.
Yeah.
It's not
It doesn't look crusty
not
enough.
crusty. It's pretty angular.
Yeah. I didn't know if it was better just to go in just to get something.
Yeah, we have two rocks already.
Yeah. All right. Good enough.
I think we can
I'm
drive
fine
for one
with it.
more towards the end. Yeah.
Okay.
Okay, to get some images of one of these.
Sure. Document this guy here. Again, these crinoids are known in the Pacific, and they have lots of animals that live, very small animals that live on them. The little polychaetes to parasites. We're at 1457, so we're almost 20 meters above where we thought the top was.
What's that? Yeah, for a little bit. Do you need one of those tulip sponges?
Yeah, got one coming.
Yeah, I got one right here.
All right, how about this sponge here?
Great.
You want a sample?
Yes, we do. And how much do you need, do you think? Do you want the whole thing?
Is it possible to only get a piece of the top, or do you think we should take the whole thing for?
I think if the whole thing fits in a quiver, I would take the whole thing.
Okay.
Nice little one.
The laser's on. Can we see how big it is? I mean,
They are, see?
oh yeah.
There.
It's
So, you know,
it's a good.
it's gonna be quiver sized, right?
Yeah, 20 centimeters. About double that.
We'll make it fit.
We can go for a smaller one if we don't think it will fit in the quiver. I'm sure we'll.
So that might be a Rittigorgia bella up there. You see it?
Oh Oh, over there.
Here, over here.
Oh yeah, it might be actually.
Yep.
A different.
We should go.
species.
We should go look at that too after this sponge. There it is.
Yeah, it might be.
It's a little bit bigger than the quiver.
I think you can get away with a chunk of that.
I think so. You could.
Like
Yeah.
snipping it.
I
That's
think
all
yeah,
I need.
I think you
so.
can get away with a chunk of it.
I think.
I mean, we could snip it.
That's what I mean,
It'll
yeah.
fit in because it's a sponge. It's up to you, which one?
Your choice.
It doesn't.
What, is it going to crumble if we take a piece?
No, it's pretty spongy.
Okay.
You just want a piece of that.
Yeah, I'll be using it for culturing sponge microbiota
Want to zoom out? You good?
in the beauty shop.
Okay.
So we're only going to take a piece of this sponge because it is about the size of the quiver.
You take it in situ.
Mm-hmm. Perfect.
And what number is this? Just make sure we got, it's 33? How does that look?
That's fine. I'm just worried that it's gonna.
What? It's not gonna fall apart, is it? He's just trying to cut it, that's all. Don't know if it's gonna float, that's the other thing.
Do you have a holder? Is it still attached?
Check
You should be able to run out a little bit.
So you should check that.
I'm not sure that is it might be the backside of it.
It's very spongy.
Mm-hmm.
It didn't even do anything. I wonder if we should just try to shove it in the quiver.
Well, yeah. Well, my thought was to just snip it at the base and squish it in. Right.
You can tell them if that's it.
So they're gonna get it here shortly, so.
It's color free because it's the backside now. The bank
Beautiful.
Right?
Beautiful.
Yeah,
Let's see if it gets, see if it gets in there first.
Yeah.
Can you guys look at the other side of that sampled sponge and see what the inside of that sponge looks like?
Good
Just
idea.
for the snapseed. Just to see if there's anything living inside of it.
The one here on the stalk?
Yeah, yeah. Just
Okay.
like just take a snapshot for the other side. Spongivery, huh? Who found that?
No, it's.
No, I know a different one.
Oh, okay. I was like, no, I don't think it had a.
This is great. I'm gonna get a couple copies. I'm gonna wait to in the library. You want it?
I'll get it after.
Color ficus. Yeah, the family is Rosalia. But yeah, it's color ficus. I think it's the size of the bell, yeah, the size of the mushroom capping there. But yeah. It's
That's what I was working. Yes, do. We wanna look into it if we can. I mean, on the backside, it's open. Yeah. I think that's often diagnostic, right? To look at the,
Backside of it?
yeah,
Yeah.
that's what I always thought.
Completely from the backside or top side down?
Well, I think it's on the other, I think it's,
I was just wondering that. I'll check it out.
yeah.
It may be.
Yeah, I'm assuming she meant look in the back of it to look for animals inside.
Yeah,
Okay, but it's also, yeah, the whole thing is bent over.
it's okay if we,
Yeah.
just. That's probably a good angle, like, on the other side.
No, I didn't like the frames there.
Never known of associates to be with these. Let's see.
Yeah, I think we're okay. Too much.
Like right there?
Yeah. Yeah, I don't see much yet, but we'll get close.
You got it? You good? Sorry, Eustace.
There we go, nice job.
Right? I mean, I don't see any amphipods or anything like that in there. So,
Move
I don't
on.
think there's any coral livery going on here. So,
Spongivery.
right,
Yep.
right, good enough shot. Great. So the other coral we wanna look at too, the,
Yeah, going up the hill.
yeah. Oh, it's just up there. It's just this, here it is. You got it?
Yeah. That's what we're thinking. It's certainly more rare to Megasporellus. When you see it, it's fairly clear.
It's got some tight curls.
Yeah, well, unlike the other species we've been looking at, this has all the whorls up really compacted tightly at the end here. Never known much of any associates in these either. 1454 liters.
Beauty.
Do you want some of this, Steve?
No, I'm okay with this.
You are?
Yeah.
Get a nice beauty shot.
Don't have any hours control. Yeah, I don't see anything else here living here.
Yeah.
Oh wait, yes I do.
There's something in
There's
there.
a shrimp in there, yeah. Any chance we can image that guy in there? Do you see him? Great. Oh, it's gonna be the.
What is this? Chrysogorgia?
What do you want?
Just going background, just
Okay,
looking over.
okay.
Thank you.
Wow. Hold on, can I zoom?
Yeah, just zoom, yeah, no pan it over.
Okay, I'll just zoom. Other way, other way, other way. Sorry, gang.
he's moving, isn't he? If you don't mind, if you be better. I can zoom out a little bit. There we go. So I think that's Bathypalmanella shrimp. Maybe Serrata palma. There's a couple species of them, but they have a massive forearm. It's like Popeye. One arm is, or leg is normal, and the other leg is like this massively long thing. Well adapted for living, but I don't remember them being on Bella, on this parent species. Any chance of suction set? Like getting the shrimp? Would that be a crazy idea? Worth a shot? I don't know. It's just hard to get these things. You got the camera? Go. Yeah, I got the camera. Okay. Wow. We often see this shrimp with Chrysogorgids and a couple of other species, but I'd like to confirm who this is. It's a sort of a rare sighting.
Are we good with jar 2, or do you want me to
Index, please. We've got a stick of patties in 1 or 2. We'll
You
go
don't
for
know which one.
we'll go for 3. Well, it wasn't. You couldn't see it visually.
No, I know. So it's pretty sizable. I mean, if you look at the lasers,
Yeah.
it's at least triple that. At least.
So there you go, right there, right? So it's at least 30 centimeters across.
Yep.
And you needed some sediment too, right?
It's a
In the slurp cup.
Hmm?
You need the sediment in the slurp cup. No way. No way. There he is.
We can call that a successful sample.
That's 34. Yeah. Nice.
What did you call this shrimp exactly? The-
Bathypalmanella.
Do you want to have a look at them?
Yeah. Can you? The little camera there. So. Nice job. Well done.
That was amazing.
That was amazing.
Good job, guys.
That was in 3. Yeah.
34, yeah?
Yes.
34.
And that was on an Iridogorgia bella.
There's your buddy, right?
That's it. Can you see the massive
Look at that arm
See the arm?
on the right side.
Yeah. The leg. Yes. Nicely done. Did you just add that camera?
Hmm?
Did you add that camera recently?
Camera.
That camera.
The jars.
That's the same camera.
It was.
Yeah, I
Five years
just
ago, isn't it?
rotated the 4K one.
Right. Okay. All right. Fabulous.
Ah.
All right.
That was trying to get at.
Good job. Good job. Good job. Okay. We should continue on. That's great. Okay. We're on. All right.
That got me excited. That was cool.
That was cool. So can you look at
Excellent.
where the next waypoint is? I don't know how to. Yeah. It's this little jog coming up, right? Or something. Not really.
Yeah, so waypoint 6 is.
Yeah. Just gonna head that
So
way.
start heading that way.
Yeah, I think so.
Look at Angel's comments up there.
What does that mean? Say something. What does that mean? Say something.
Oh, that's where you can type if you're Schmidt Ocean. It's
Oh.
just part of the
Oh,
program.
thanks.
It's not a problem.
Am I supposed to? Should I tweet that now or what?
Yeah,
All right.
along with a no.
Yeah, right. All right, here we go. All right.
So we're on our way to
So-
the next waypoint.
Yeah. We're now 1453 meters heading to our waypoint 6.
What's that? Is that another one of those sun stars?
Don't know.
No, that looks.
Over here? What is
Oh,
that?
yeah,
it
it
is.
is.
Massive.
You want to have a better look at
Yeah, let's have a look at that for a second. I want to see how big it is.
New waypoint, thank you.
Look at that here.
That is unbelievable. And also look at this rock here too. Got the camera. Sorry, hang on. I got it. You got it.
And then I'll get the rest of it.
All right. I can tell you like to fly it in, and I'll just sit back and watch.
Get better light.
Yeah, I know. This is good
That's the best
TV
part.
right here.
Right? So how many arms? How many arms you count? 15? This is a Precision Sea Star.
Come on.
Basically a grazer. We see it sometimes on deepwater corals.
Yeah,
And
you're
now
right.
it's hard corals, now it's salmon and other ones like that. How many did you get?
15.
15 arms. It's a lot. It's a big guy. Look at that. The lasers are on it there, so you know it's a half meter across. Right?
Wow.
I don't
Yeah.
think I've ever seen one that big actually. The size we've seen on these guys. These are huge. All right. Thank you. That's great. That's ridiculous, isn't it? You got it? All right.
And so you want to have a look at this rock right here?
Steve, I'm going to ask you, what do you think? Is this part you can break off? This is sort of
Yeah.
pavement-like,
Where are you looking?
straight everywhere here, right? Straight ahead. I mean,
There's like this shelf.
if there's a piece, an edge.
What's going on right here? No, that little critter.
What do you see?
That's what lobsters.
Oh, look at that.
It is.
It's a homolodon.
It is a homolid, isn't it?
Yeah.
Can we zoom in? Yeah. That's a good
Has-
catch.
Has an anemone on its back.
Yep.
Oh, that's
I totally missed that.
awesome.
Nice catch. It does. It's hard to see, but that is like a decorated crab. Here it comes. See, the back legs are modified to carry something.
Hobo crab.
Homolid crab.
It's
Oh my goodness.
Freaking out.
modified to hold things.
Yeah.
He doesn't like
So
the
this-
fact that we're here.
The back legs are modified to hold things. It often cuts off a piece of sponge or a piece of coral. Here it's an anemone, and it holds it over its head like it's hiding. and trying to fool things. And so we see them on corals all the time. That's a great catch. Here's an anemone. And we have not seen that anemone as far as I know on this dive. So
Orthodox
yeah,
in Brooklyn.
No, nowhere near here anyways.
that's fabulous.
But
It's
yeah,
just
they carry
fabulous.
pretty
Check 'em out,
genetic
check 'em out.
fish.
He's on the move.
In this part of the world, you see with the
Let me
anemones.
zoom in.
It's
Get a
like
nice
hat.
beauty shot. There we go.
He's got
Beautiful.
it. I mean, no, J-Rod's got it. He's good on it.
That's
Perfect.
Just wild.
That is wild. Isn't that great? If we get the shot of the head, we could probably get an ID on that thing.
I mean, they never see light ever, right? Never.
Oh yeah, they're probably
He must be
freaking out.
completely freaking out.
I wonder if it's something like
Well,
it
Do
could be
they-
some sort of
Do we
biofilm.
know if
They're
Hamala
using
crabs
the eye
see
or
visually with their eyes, or if it's
so even though you may see eyes on animals down here like fish and crabs and shrimp, they're really not likely functional. Oftentimes they have larvae that feed in the water column, up much shallower than we are now, like maybe 500 meters, even 200 meters depth. And they'll feed there and they'll use their eyes. There's functional eyes as larvae and juveniles. Juveniles, when they come down here, they lose their ability to see, their vision, and they're probably blind. And they just haven't lost their eyes because of evolution hasn't required them to lose it.
Is that like
There's no heavy selection to lose your eye. It costs evolutionary energy to lose something. Like we still have an
But is it
appendix.
bioluminescent
Yep.
or is
Right.
it
So that's why.
iridescent?
Good job. It's awesome. Thank you.
I think he's
Look
It's
at
pretty-
that
yeah,
shot.
let's go.
No.
It's iridescent.
Yeah.
It's
Great.
seeing is the refraction.
That was fun. That was
Yeah.
I
cool.
love seeing those hamalids. They're just so much fun.
You did
They
a lot of
often-
theorizing
I mean,
about why they have
well-
that, the chemical cues or disguise.
To disguise. Yeah. There you go.
Cody thinks he can get this off because it looks like it's almost ready to break off.
All right.
Get that one.
And if it does break off, is it gonna fit something? Yeah.
Yeah, right.
Cody.
You like it?
This is
What?
old basalt.
He likes it.
You like
Basalt and
So.
I like it.
Yeah. Yeah, that's great. Oh, sorry.
Yeah.
Yes, absolutely. Yes.
How far away you are.
Steve, what do you think of that one?
Yeah, looks good.
Yeah.
Before you get any sample buttons, just poke it, see if it.
You want to poke it? It may break if you just poke it.
You know what type of crab it is? Someone's asking.
It's a hamalid crab.
Hobo crab.
Hobo crab, no, hamalid. From the family Hamalidae. It's kind of like a decorator crab in the deep sea. But they'll often cut like pieces of sponges off and then they'll go into a coral. So it's not truly camouflage. Sometimes they actually grab
Oh,
a
you
piece.
did it.
Wow, that's a great one.
He knew it.
I can't believe you got it.
Oh, look at the.
Can we image it first?
Oh yeah.
What I'll do is I'll grab it
They hold it up for a
and I'll
little
hold
bit.
it up for you with good images. So it's only about 10 centimeters across. Is that? It's only about that
Cross,
big?
yeah. It was longer, a little bit longer. So important to note on the sample sheet that this one has reddish orange stain because the others.
Good rock, guys.
Yep.
Thanks.
It's oxidized on some part.
that God?
It's a good sign.
So.
Did you hear something? That's the voice I hear all the time. Right? You get
This is a rock.
Snap.
Flat
Snap.
rock.
That's
That's white.
Yeah, I did.
Oxidized on the back.
Or some kind of an algae.
Where
The
you-
orange.
No, that's called chemical alteration.
Yeah, that's good.
When
So
water makes contact
going rock
with a lot
box?
of minerals in the rock.
it's a close-up
This
shot.
is gonna be number 35.
I like it when J-Rod does the camera. What?
I know, me too.
35.
Hold on, let me get a beauty shot. Beauty shot.
Too easy. This could be yours.
Low,
With a
please.
grant to the Army Town.
That was good.
Yeah.
We gotta put you guys on the mics more.
Flights later.
What did it
It's
go
a
up?
multi-payment right there.
It looks like somebody plugged in a Raspberry Pi over here. Look at that. Look. Zoom, zoom, zoom.
Oh, that,
What?
that's that, what's
Is
it
it
called?
going?
Oh, in the
There you go.
back.
What's that?
Well,
So
you're
4 amps?
waiting for it. It looks like it's
Just oxidation,
maybe
just
someone
chemical
unplugged
alteration of the rock.
or
That's
maybe
why it's
this
so rusty
Raspberry Pi is
and
running
looks
all the
white.
screens.
But that was a great spot.
But it used to be that image. I would love to have it back. I don't know. Oh, that's just an experiment.
Rock box. That was 35. Happy birthday. You got 3 more hours. All right.
Just the discoloration, like those
Don't you love it?
orange, red color.
Okay.
So we can identify and differentiate it.
Is that a crinoid?
Yeah. Keep it on. Keep on keeping on. See if the coral communities change the composition.
Did that go in the rock
The
box
Almala crab
successfully?
was right there.
To avoid predation, or
Did the rock go in the box already?
stuff from the.
Well, we're not going to fit in the suction set.
So it's not eating with the little spines.
Pretty strong.
No, but it can capture.
Yeah, right.
Did that go in?
Right.
That went in the rock
Bring
box,
it-
and I should
bring
write that in right here.
Okay. No, I just want to make sure I turned around and asked you a question.
Yeah, so that was FK
Crinoids, the ones that swim, like the swimming star.
35.
35.
Do they have a sense of direction? Or do they, are they just randomly moving and the current takes them wherever, they're not making
No.
a decision, a nervous system
Yeah,
based
anticipated
decision
action descending a little bit.
to go in
And then
direction.
going back up again.
Right, they're not that smart.
Thank you.
Because they just
He just wanted to note the color.
fly up and they drift down like
Discoloration.
the correct Mary Poppins style.
Do they sense pressure? Because we've noticed when they come up to us, they'll flip and go our way, like immediately.
Who does that?
Yeah.
Crinoids?
The crinoids? Is it
Ah.
crinoid?
Oh, we got another Viticordia.
As soon as they see us, feel us, I guess you could say.
Yeah.
That's a shrimp? Yeah.
It's like a
He's
Blesioptenaeus or
You got your close up. You get a beauty shot.
That's fantastic.
That's cool.
That's what they do all day. Those swimmerettes are saying
Is that
it's
what's
built
like
for swimming. What?
is on their legs? The translucent.
Yes, it's like a brush, so that it hits the water with more surface area, shrimp, and pushes water back. That's why it's a really efficient swimmer with not a lot of energy expended. So it's great. Still seeing crinoids.
I know, so many stocked crinoids too.
We're
Oh, and there's-
1,453 meters, so we're still 20 meters further than we thought, further shallower than we thought we'd be. Vitagorgia,
Doesn't look like it has any associates. I already noted it.
massive Brisingid sea stars. Those things are massive. You see their arms are outreached right there because
Are they like reaching out into the current?
they are trying to gather organic particles. Clearly, they look well fed, don't they?
Yes.
There's a whole thing. There is some sediment here. There's a sediment interest.
Yeah, why don't we go ahead and get some from here?
You do?
Yeah.
And you want to get a house. Sit right there.
Yeah, that nice little pillow.
That pillow of sediment?
Yeah, just a little bit.
Pillow? You wanna try the scoop thing again, or
What do you think, suction or scoop? It's up to you guys. I trust your expertise.
Well, we've got the poker doker thing. We'll poke into the ground and see how deep we can go.
Okay.
You don't think?
Or do you want to do something? Thank you.
I just need the sediment, whatever you think. Whatever you want to try this
If
round.
we do the suction sample, will you,
Clean it out.
yeah, will it clean the sediment out too much for you? Or, I don't know what you're getting out of the sediment. How
I am
small?
going to culture bacteria from the sediment.
Yeah.
I mean, we can get a whole bunch of sediment with the suction sample.
I mean, whole lot. I'll pack in.
Okay?
It's up to you.
Try that?
Yeah. Like the suction sampler.
This is probably pretty shallow one.
On the back here.
Just a nice little graze of the pillow.
No, this is the most sediment I've seen so far, really. So
No, thank you for saying something. That's great. So
Yeah.
sample in situ. And this is gonna go into jar 4, I'm assuming.
Jar 4. Yeah.
We are jar 4, correct, page 3, that section is on.
We want 4? Going to 4, guy.
Roger that.
It's number 36, I believe.
Yeah. When it happens. You good?
Yeah, I was just,
Right, just
memory.
the
I was
top.
just checking myself.
You want just the top, or you want?
All of it.
Oh, like this, any from anywhere?
Do you care?
No, I don't care.
Fill it
Yeah, a
up.
little, yeah, there we go. Very nice.
Look at that. There's some rock.
Sediment is incredibly microbial rich. It has 10 to the 9th microbes compared to the water column here, which might be 10 to the 4th.
You can see it filling up the jar up there.
Where do you see that
See
now?
that- this
Oh,
jar cam up there.
okay.
Pretty effective. You can watch it going, so it's just gonna sit there and churn in that
Yeah.
jar.
Maybe we'll clear off a rock for
You think that's
coral.
enough?
Where?
I think that's plenty, thanks.
Do you want that Xeno? Do you want it? Do you want the Xeno, Steve?
Sure.
Is that bother
Sure,
you?
no, I'm not
It's
at
all
all.
part of the sediment.
Yep.
Code, you got it. You got this.
Angles.
Angles.
No, I think that thing's full.
I was gonna say,
Might end up in section jar number 8.
Oh, okay, great. Well, that makes it easy.
FK 21.
Jar's full. Alright, you count.
30, that's 36.
On to 8.
Sediment's
It went into
36.
4 and 8,
then,
correct?
And the
Oh,
xeno.
no, you- eight's a flush.
Eight's a flush.
Yeah, it doesn't
That's
matter.
what I do know.
That's why it should be there.
Okay, so that's backwards, right? Ready?
Go.
So we got the xenophyophore? Almost. Yep,
Don't know.
it's in.
Yep.
And what one did that go into?
The 8 has your, some sediment, well, and the xeno. 4 is just nothing but sediment.
4 is just sediment, and 8 is xeno and sediment.
So do we
Okay.
call them the same number? Anybody?
Yeah, that's fine.
Same sample.
I think it's fine.
Yeah.
Fortunately, I can only sediment plus Z. How do you spell xenophyophore?
Just say Xeno.
Z or X?
X.
Cameras are on, so you can see the recording.
Okay.
Cool.
Oh,
Thank you very much. I think that'll be a very productive sample.
Yeah, I think it's full.
Steve, do you see this coral here? What is that? Yeah, come please.
I think it's
Just
another.
at the. Oh, it's a Iridigorgia, right?
Yeah.
Sorry. It's a little funky. Okay. So what else do we need to make sure we get? We're on the cleanup crew here, right? We got less than an hour left probably.
We don't have any, obviously, stony corals, or but we haven't seen any.
Nope.
Do we have a branched primnoid for Randy and
Yeah.
I?
Yeah, we did.
Great.
We did.
Branch?
We did get it, yeah.
You got your Metallogorgia with associate.
Yeah.
Have we done
Who's the
any
one that's
RNA
inside?
later collections?
What's that?
have we done any RNA later collections?
No, we've not.
So that's a big one.
That is a big one. We haven't really seen the. Just gonna do a quick snap.
No, wait.
It always looks like they're lounging on something. Are we chillaxing? Yeah, just some rain, some Dapper Johnson.
Just make sure I get a frame grab of that. I'll get that.
No, I'll just use it on
Perfect. Good.
Here's the camera around.
Thank you. Other direction.
Wow, look at the like almost like raindrop like structure of their legs.
Okay.
Their spines?
Look like it.
Oh, sacks, that's a good word for it.
Yeah, yeah. Pretty awesome. Got it. Alright, good. Thank you.
Plexaurids are on the list. Did we get any of those?
I think we did.
Okay.
I figured that's what he was trying to do.
Right.
Get the words right, but not much else.
Right. Anything else? We got the sponges from Brian.
Plexaurids for this guy?
If we see any more, yeah.
Okay. If we see any more, he'll
Alright,
take them.
let's just keep working. Let's keep working.
Culture.
Fan corals. Plexaurids.
There is one.
On a single plane. There's another one of your sponges.
That was great.
That's a good rock sample, wasn't it? Does it feel flat here now? It's still coming up.
It's still coming
Still
up.
coming up a little bit.
Be good. You could see some pretty large stuff here.
I would think so. I was really hopeful to see a lot of stuff.
Here's
up here.
another metallic orchia. Is that what that? Oh no, I can't tell.
Yeah, there's one there. I think you're right. Talagorgia. Stichopathes is here still.
Yep.
Wow, I really thought this would be packed.
It's real sandy.
It's getting really sandy. So this is the.
Oh yeah, pecan.
Wow. Snail trail. We haven't really come down though in depth yet, but
Is that a rock? Yeah. I guess.
yeah, pretty cool up on here.
You don't want to look at whatever that was?
No,
Mm-mm.
no thanks.
That was my bad.
Just another rock. Really gorgeous skeleton on this pavement.
Yeah.
A lot more sediment here. This is really.
Do you know?
Well, see, there's more. We saw more corals on the slope than we did up here on the top of the ridge. There's a whole thurian.
There's a black coral,
Another
Stichopathes.
Stichopathes. Yeah, we're here exploring for corals up at the seamount, 1455 meters depth.
Looks like it's dark crinoid maybe.
We start heading down here hill here in a second. It should be downhill a little bit and then come back up again. There we start. 0811, 1456 meters depth. As you're on also heading down slope.
Then you slow down. I might have to slow
I
down a
know.
little bit, 'cause I'm not running much.
I know. That's a good.
Do you wanna poke over to the other side of the
Well-
ridge while we wait for the bus?
Yeah, absolutely. It's within your watch leash.
Oh, I follow Jason in Michigan on Twitter. Cool. He watches a lot of the dives, if not all of them.
Still more stickopathies here.
Yep. This has been the Stickopathies dive, at least for us.
Just jaunting off here to on
Another
the side of this
tulip
ridge.
sponge.
On the western side. Just wait for the ship to make a move.
Or is it another Metallogorgia? Or is it a double spiral?
That's a
Bella.
Iridogorgia. Bella. What's that?
Focus is kind of.
It's in autofocus.
Yeah, it was in autofocus. Now, whenever I go past it, it kind of
It clicks in.
starts finding. Well, there's something living in there.
Is it really?
Yeah, a little bit of shrimp in there. Two of them.
Oh. Same species, looks like. Let's get some in situ shots here. Come out. No. Sorry, you're good.
Those aren't the same species, are they?
Yeah, they are. They aren't.
They have the big
I think
arm?
they aren't. There's just
Tucked.
tucked into the bottom. Coming out.
Hi, Jason.
Alright.
Kind of going back now on the side of the ridge where we were.
Sorry, I'm looking at the.
I got it. I totally got it. So not seeing anything much different here on the western side of the flank. Although there's a Isidella, is that right?
No, that's a Carnoa. That's
Oh, you're right. That's
Oh,
not
a
a bamboo.
Primenoid with.
That's right.
not interesting.
You want to see it?
Yeah.
Let's take a
Mhm.
look.
Let's get some
Got a
associates.
crinoid on it.
Looks like two.
1457. Haven't changed depth much.
You should get it.
Good. How's that branching look?
Good. Push in.
Yeah, okay. Want some more?
Yeah, a little bit more.
That's it. Does that look in focus?
You want some more? Yeah.
That looks okay to me.
I was just trying to get better focus, but I think that might be it there. It's kind of funky.
Looks okay to me. No. Happy with that.
Yeah. Any thoughts? What you might?
No, not yet.
Good. The no thought coral. That's good.
Another sample?
Would you like some?
I think
Is
That one that we
Randy?
had earlier? That was bigger.
it's very
Diff-
similar.
Yeah.
Okay.
You want to get a snippet?
I'm coming.
Do you wanna get a snippet of it?
It's 1 second. Randy's
heard
gonna
it.
come in.
Yeah, if Randy wants some, then keep it for.
I mean, I'm thinking we're here. Get it.
Sorry. Hey.
We got
Okay.
space
Do
for
we
it.
How
want
many
this?
Prunoids do we already have collected?
What do you have so far? You think it's 4? No.
4 Prunoids total?
No, no.
Or just one?
We got Quintiflora. I didn't think it was that many.
California.
1, 2, 3.
No. 2.
Patella. Calyptophora. Calyptophora.
I think I'm good.
Okay.
You
Thank
good?
you.
Yep.
I think we're good too.
Stevie?
I'll pass.
You want it?
Yeah,
Look,
why don't-
I could use like a little bit more because we didn't collect enough in that other one. All right, go ahead.
It's also will be
I mean, what's
fresher.
that
too.
thing? Oh. What?
Oh.
No.
Is that a mushroom?
That's
No,
a
no,
picard.
no.
Homo crab.
It's a crab.
It's a
It's a
Hermana crab.
picard. It's a picard crab.
A bigard. Oh,
Yeah.
you're right. It is. Look at that. It's decorator.
It's the back leg.
Oh, it's
Yeah.
decorated. It's wholesome.
All right, you know what? Go get a couple of stems of that Primnoid
I
bees.
think that's a good idea.
That's a good thing to do. Let's do it.
Do you want more into it? No, we got it imaged well enough. We're ready to go.
Yep, we're ready to go.
All right. Talking about what else do you do? We're up on this ridge and we've seen the top of it for quite a few. We're seeing the same things. Not much of anything up here, so we went down to the jump off to the west to go on the flank to see if we could find anything off the side. Same as what we've been seeing.
You want to come up?
We could we could go on the other side. We can keep going along the on that between the second to last and the last waypoint. It has that dip, that depression.
Yeah, yeah.
We're starting on the dip, so we have to
So we could make tracks through it and see what we get.
Could do
And
that.
Come on the other side. Do you want to try that?
That's fine.
Let's do that.
Yep.
Okay.
I guess I heard something like 10 o'clock or something like that.
Yeah.
That's
I
up
was
to
sort
you.
of. It's 9:15. I mean, I don't have like a time off bottom. It's super strict, but I
It's
had
an hour.
originally estimated 11. But if we're done early, then we're done early. Shouldn't linger.
We can do whatever you want to do.
I say you run through the dip and see what's on the other side.
Yeah.
And then
And then see what's there.
see what's there and like don't linger. But
Right.
did you
Well,
fire?
we're
How many
not-
Niskins have you fired? 2.
2 Niskins.
When was the last
One
one?
was in a really good one, good spot. It was number 1 was the last one, which was sample 30. I don't know what. I think that was in a coral patch, right?
You might just want to fire a Niskin somewhere on the top of this ridge.
Say again.
You might want to fire a Niskin somewhere on the top of this ridge.
We can do that.
Great.
We got plenty of a big one.
Yeah.
Okay.
Nice Brazingid, by the way.
Amazing.
And that Metallogorgia, that pancake flattened, really gorgeous. Not Metallogorgia, I mean Iridogorgia, entire.
Yeah. Yeah, it was nice.
Really nice.
I know.
I am.
Haven't seen him for Arne later yet.
I keep
But-
every
but
time you
it's-
pass those Iridogorgia, just so you know, I'm like kind of looking out for those little jellies.
Oh yeah.
Okay.
Yeah. I'm
I've
always
been-
scanning. I'm always scanning.
Yeah, I know you are.
But-
You're always looking for
but-
associates.
But I hear you.
Just a little snippet.
Maybe get like 1 or 2 branches. So, let's see. From the base. Like not the base, you know what I mean? From the.
We're only here once.
Yeah, exactly. So
I would-
Tim's got it.
I would just take that or
Either
something.
that or one piece from the bottom.
Or
like
go
right on the
here.
side.
Get Brent. Yep, that's fine.
Branch.
Cody. Yep,
Yeah.
that's great.
Thanks, guys. How's it going,
Oh,
Constance?
it's orange.
Enjoying this?
What?
It just folded up. The crinoid.
What did?
The crinoid. It just went.
Oh,
Do you want the crinoid?
They're gonna get it. I think.
If you want it, you gotta tell them.
Yes. If you can you come down?
Of course.
Yeah. Great. So you probably saw we just got some sediment, so I don't know what else.
I think we've basically
Collections.
we've done well.
Yeah.
I think let's just get some terrain shots. I'm looking for a little more corallivary. Split if we get another. I kind of want to see what's on the other side of the dip. I want to get to the top and then
That's fine.
upload it.
Yep.
It's because this feature was so cool. So if you are listening to our side conversation here, we are diving on a feature that we mapped, I don't know how many hours ago, but not that many, for the first time. We're diving it for the first time. This is an seamount that has been, you were looking at a part of the planet for the first time that nobody else has ever seen. So we're here too. And the feature that we mapped is pretty darn cool. The whole entire structure, the seamount, looks a lot like a brittle star. It's got all these big arms, which are ridges, volcanic ridges, and we're sort of summiting one of those ridges at a local high, and we're in this little divot right now.
37.
As we were sort of on the top of a ridge, and then we're kind of in this little divot, and then we're going to kind of go back to the top of the ridge and all the way up to this local high. And so, kind of want to, we've set out a hiking trail. You got to make it to the summit.
Quiver 11.
So we'll do that.
All right, it's in there.
Nice job.
Sweet.
So good, guys. Thank you so much. And so we're going to check it out, but we're not going to linger too long on this because we are still, we've got a late night in the lab after we're processing all these samples after we get off the seafloor, and then we have a ton of transit left, another day or so, as we head to the Phoenix Archipelago in the Central Pacific. So we are going to be heading to the U.S. waters in the Pacific Remote Islands Marine National Monument in the Howland and Baker Unit, which is a no-take protected area in the United States. And it is the sort of mirror image in many ways, or the other half of the archipelago to the Phoenix Islands protected area in Kiribati, where we've been working for a long time. So it'll be really neat to sort of work both sides of the equator here, compare to our 2017 samples. And so everything that we do, these expeditions are, time is a zero-sum game. The time you spend here is time you're not spending somewhere else. So we're gonna keep getting to the top of the ridge and then see in a day or so on the seafloor in the Priminum, the Pacific Remote Islands Marine National Monument, which is our target area. Or actually, I think we'll be in one more ABNJC map before that. I lied. I can't remember.
Go ahead and submit that.
Great.
Do you
All
see
right.
Angel's comment up there about
Who?
the
Yeah.
Angel on Hill? The
Nice job,
crinoid
guys.
rolling up versus swimming
No.
away?
Yeah, they do that.
They do that? Okay.
All right, let's keep moving on.
All right.
The bonus content.
Let's see the top of this hiking trail.
Yeah, let's go across the pond.
My friend Jan Winning,
Impression.
who has like one of the best quotes to describe deep sea diving. You're like hiking at night in a teeny tiny trail on the side of a huge mountain with nothing but a flashlight. Let's take this flashlight and go hiking. Yeehaw.
Boogity, boogity, boogity.
Boogity boo.
Mhm.
That's no worse.
I love it.
Fuck, ESPN. That's awesome. That's it.
What about you? Here we are making our list and we're being rude. Jason, it's your birthday. Is there anything you need from the deep sea floor?
No, I'm good. Astros won today.
What'd you say?
They did?
Astros won today.
Did they?
All right.
Good one. Good one.
Stick a path these.
Yeah.
Something shit.
I'm gonna gorge you again.
Something shit.
So that was
Another tulip sponge.
Somebody's making a comment in the chat about, the map looks like the rover's exploring Mars. It's kind of exactly like that. Here we are in this totally foreign environment, but to us, but you know, it's part of our planet. But it's true, we do require and rely on technology. Like right here, we are doing this with our fabulous ROV pilots, a marine technician, and on the bridge we have the captain and the mates who are positioning the ship. You know, this requires, there are 39 people on the ship, and it really takes everybody to make this happen. This is a massive collaborative effort just to be able to show you these images in the seafloor and collect these samples. So and we're in a room that feels like a mission control room. There's, we counted it before, something like 34 or 35 screens all around us. And on a map that we made, and it's totally like, look at this lunar landscape, except that there's much more life here. Way cool. What is that?
Sponge.
No, to the left.
Oh,
There it is,
stocked
crinoid.
crinoid.
I see it. Barnacle. Yeah. What?
Party people.
Hi.
Here we go.
Somebody took a chunk out of
Down
that too.
under this depression. They go down for a while, then come back up.
Yeah, but there's a fish.
Yeah. Fish.
Woo!
Rat tail. Holothurian.
Keep going.
Keep
I said
going.
keep
Keep
going.
going.
Oh, we've got some more glass sponges over on the left there.
I don't know if you guys saw this, but you know, pandemic stories, but the Keep Going On song. Did you see this little video? It's fabulous. It's just this husband and wife. I can't remember their names.
Another Rita Gorgia.
They made like this homemade song. This original song called the Keep Going On song, about like pandemic, when you guys keep saying keep going. It just kind of pops into my head every time. But it was just very sweet, very inspiring. Like, we can do this. I'll show it to you.
Keep swimming stuff?
No, it's better than that by like a lot.
Is that how it
I mean, I think it's probably a musical masterpiece.
What'd you say?
So just keep swimming is really a musical masterpiece.
It is. It is.
Is that,
One of
that's a
Disney's
Chrysa
finest.
I
Gorgia.
don't know. Well, there's a broken hole. No, it's not.
No, it's a little Rita Gorgia or something.
Yeah, it is.
I think it is.
Yeah.
What about, is that a sea
Umbrella.
pen?
Yeah.
Yeah.
Umbrella. Lula. Lula.
Another Chrysa Gorgia over the right. That little thing just sticking
You wanna go
up.
back there?
No, no.
Really crusty rock.
Okay. There it is.
Yeah.
A lot of
Cucumber.
cucumber.
Yeah, would y'all like some crusty rock?
I think we're
Rocked out.
I think we're all rocked out.
Okay.
ready to roll. Sorry.
I like that.
I mean, like,
I like it.
nobody's
It's
done
good.
it yet. Thank
I
you,
like it.
Cody.
I like it.
Uh, oozing with cheese. Thank you very much.
Oozing with cheese.
Velveeta.
Steve, another one of those white tubes? Those U-shaped tubes? Is Steve in there? Is Steve back there?
Steve,
Steve is
it's
there.
not back there.
No, never mind. Never mind. Stickopathes,
More
Holothurian,
stickopathes.
Cucumber.
And a shrimp.
cucumber. Oh, plenty of rock here, though.
It's pretty.
More stickopathes.
You know, it's amazing. I keep thinking that, you know, I would hang out on the top of that rock if I was living down here. But clearly, that is not the place to be.
You be alone?
All by myself?
All by yourself.
Clearly
Oh,
not making
gosh.
good choices at the bottom of the sea.
Not making good choices.
It's like a little elevation. There's like a little mountain on the mountain. No one's going for it. Well, somebody's going for it. So they've stocked crinoid or something in the back.
Oh, the Holothurian, stickopathes. Oh my gosh.
The stickopathes are. This is their spot. There's a ton of them.
62, so we'll come down a few meters.
What is that? Wait, what is that?
Yeah, that's,
No, that's
yeah, that's
Numbulula.
fine. It is.
The sea pen.
Yep.
Nope, 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 no, thank you.
Another stocked crinoid.
I love these crinoids so much. And Jimbo's not in the room, so we can call them crinoids if we fear. Look at that tomato color.
Tomato.
No, 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 no.
Monsters.
They're beautiful.
They are monsters.
They're like dandelions.
Steve mentioned they're prehistoric, like they date back to.
Oh, they go
like,
way back in the fossil record.
For sure.
Absolutely.
I remember when I was a kid, I went to the
Sea star.
Smithsonian
Same
Museum of
sea
Natural
star.
History, and they had those fossil crinoids. You know what I'm talking about? Oh, captured my heart forever. They had a whole wall of them, right? Really amazing.
Is that an urchin down at the bottom, or was that
Yeah.
an enemy?
Stickos, stickos,
Gorgeous.
stickos.
Another sunstar.
Those are the stock.
It's like a crinoid forest.
Yeah. The percentage of
But they're all oriented in different directions. You notice that?
Yes, we did. We did. We mentioned that. It's just strange. Normally they would be.
Whoa, whoa, whoa. So that one looks beat up.
Yeah, the stalks had it. We've been calling out to a bunch of crinoids. Oh, if you're right.
I was saying dandelions, but whoever said this in the chat, you are exactly right. Deep sea sunflowers, almost.
Good.
But they're all turning in different directions. It's cool.
There are some Retigorgia down the slope there.
Oh, yep.
It is,
And that's
Dead.
a dead one too in the
Yeah,
front.
but it's amazing, right? Like that was clearly stocked crinoid habitat, and now we're back into a Retigorgia. Minute little
On the slope here.
changes, and all of a sudden you get a change in a shift in ecology.
I think we gotta hang here for a second before we move the ship.
Oh yeah, we're moving now.
And,
How many more waypoints do we have?
One.
One. Perfect.
We're roughly.
Al's gonna kill me. He keeps saying anytime but 10, and we're done. We're done at 10.
Well, on deck.
Yeah.
He doesn't
Oh
want to
no,
be
he doesn't want to be on deck at 10. Okay. I thought it was beginning of
No.
operations. Okay.
So like if we
We're
start
good now.
coming up at 10.
Great.
We have about 124 meters before we're at waypoint 7.
Okay. Thank you.
1, 2,
1, 2, 4. You missed 3.
1, 2, 3, 4.
Ding.
wow. Thought there'd be a lot more up here.
I know.
I thought the currents would be
You
blowing
never-
over this ridge and that the corals would be loving it.
It goes to show you never can tell, huh?
No,
We,
never
every
can
single
tell.
one of us this morning looked at that map and said, "This is the spot."
Right.
Every si-
It's
look at
birthday ridge.
This is Russell's spot, you know, guys. If you wanna
Russ?
get- Mhm.
Russ's Ridge.
This is Russ's Ridge.
Well, that's why there's no coral, because it's.
I knew that was coming.
All right. That's good.
Oh, Russell.
Oh, goodness.
It's a mix because it's, you know, it's got the curly hair too.
True.
Just a little.
Got a bunch of 'em.
I want to know where all this rubble came from.
Do you want to fill us in?
When it erupted.
Rubble, huh? No.
It just looks like it
Jason's
broke
on
off of
Frayella.
somewhere, and there's nothing above it really to break off of. So, do you understand the geology? What happened here, Tim? Could you educate me?
Not, I don't know. No. I mean, I can talk about them. I mean, I think I'm one of the few that witnessed live the volcanic eruption on the seamount before, at a mile down. And I've never, I don't know about how these were actually formed from hot spots, but I think, there are all kinds of eruptions, explosive eruptions that are pyroclastic, which, result in like fine sand, volcanic sand, and not much in the way of rocks. These rocks here have been so altered over the last, I don't know, I'll say 80 million years or so. They're more crust than rock, it looks like to me.
Well, I know we picked up a couple and they just crushed.
Yeah, and so I don't know.
There's a
It might have just been nothing but a kernel, like a little seed to start on kind of
Yeah,
thing.
yeah.
Maybe this is all just
I don't-
crust,
I don't know
precipitated crust.
How close this was to the sea level, right? Whether it was at one point
Yep.
close or not, or so that's just. I feel like I haven't seen many rocks that were in place like they were when the seamount formed.
Yeah.
They've been there since. They've all like
Well,
broken up.
we had that sort of like compacted pavement for a while,
Yeah,
but you're right. We're back in.
it's amazing terrain.
Just keep thinking about my kids. Their favorite thing to do is to find big rocks like this and scramble all over them.
I have pictures of that from when Zoe was really young.
Yeah,
actually.
they still do it.
Our first
We took
lab
them to
outing.
Arizona, and it was their favorite thing ever. Was to just jump all over the stuff. A little less hospitable here, though.
So we're still coming down the line here.
We're
Nuts,
still going into
nuts,
the
nuts.
pit. No, we're coming out.
We're headed to the waypoint now, so
Yeah, yeah.
we're still in it.
Yeah,
For
right.
sure, yeah. But still not sticking path. You still not seeing much whole thing. It's much the same fauna as it's been
I know. It's
since
interesting.
you got up on the ridge. Even before.
Landscape
There's a
shot.
big sediment pond.
You wanna go to the rock?
With the most amazing
Go
thing in the
around.
middle of it that you'll never see, Tim.
What's that?
I don't know.
You
That thing.
should go look.
There it is.
There it is.
Umbulula.
Umbelula.
Hmm.
Yeah.
They're beautiful.
And more Stickopathes. Wow. I don't know how many Stickopathes we've seen today, but
It's a lot.
Probably the most I've ever seen on a dive like this, continuously.
Do you have some underground calorie?
Yep. Well, while you say that, let me just give a shout out to the many undergrads we have in our lab that actually do go through this footage, frame by frame, line by line, and pull out some of the interesting features that we can make it easier to analyze these data. It's one thing to be exploring here live, but at the end of the day, when you've collected however many hours of video, it's a big undertaking to go back through it all and make sense of this and turn this qualitative view into a really quantitative one. And so, to all of the interns and the undergrads who cut their teeth on these kinds of data and help us out and contribute to science, they deserve a lot of credit. And it's win-win. They really learn the taxonomy and get to a level of granularity and detail just by kind of stopping and slowing down and looking through this. That it's really
Is that
incredible.
another
Actually,
sunflower?
so
Yeah.
it really, I say it takes 39 people on the ship, but it takes way more than that in reality to turn something like this into a paper or a discovery. So I think we're going to leave the seaboard.
So
Your favorite hour?
No, so it's gonna take different 45 minutes to an hour at least
Sure.
to get up to the first ones.
I thought you meant leaving the seaboard. So 4 minutes, we'll probably get up.
Yeah.
I think you like these, J-Rod.
They're pretty.
They are. They brighten your birthday.
That's gonna be very much.
Like a little firework.
We're close. We're like 40 meters away, from our last waypoint, and where we were meant to come up a depression like we're doing right now and see the top of that. And we're at one of the most shallowest parts of the seamount. It was mapped earlier this morning. 1472 meters depth. Wow. It's funny how we always search for things and want to find certain things, but it's often what's not here is what's just as interesting biogeographically. Right?
Yeah, it all fills a piece of the puzzle.
By the way, Alexis won, everyone who wanted to know.
Which one?
She won the person to guess the summit of the seamount, closest without going.
Another bit of
over.
Gorgia. Crinoid.
Yeah.
I don't know what she said it was, but she was right.
Yeah. He's,
She got dark chocolate coffee, Kona coffee macadamia nuts. I know.
Still
Press is
going
good at
up.
this ship, so. Just saying, you gotta play to win. It's like Colorado. You were always invited. We have a board going in the library, so every time we hit a seamount that we ever found before, you gotta guess the summit. Full ship activity.
You gonna bring those out?
well,
What's wrong with right now?
Gummy bears are a pretty good prize. You know, speaking of
yeah.
nothing whatsoever, I'm really
Another
glad
stick
we're not
to
seeing any
cookies.
marine debris. I mean, I'm not surprised, we're in the middle of nowhere, but
Still.
still, it's really nice.
Middle of nowhere.
Around Waypoint 7, I came to the highest peak around Waypoint.
Oh, there's another
As you was
pin.
now you're supposed to go down. That's it, right?
When we go down,
It matched the map really well, didn't it? I don't know. I mean, I haven't seen much of a change through the last
No, I
don't
think
know,
we're good.
half hour. I do too. Another stick of Pathies. You know, you talk about nowhere though. There's places 7 miles down that have microplastics in the guts of animals.
I'm sure there's microplastics
You know?
here too,
Yeah.
because they move around.
They're everywhere.
We could test for it. We could look.
Do you wanna
we
do a,
we
this
did
one?
last time.
What?
Oh yeah, we should do a Niskin.
We should pop a Niskin.
We should
Good call,
definitely pop a
Cody.
Niskin.
Out in this.
We can look for microplastics, but
Flying cucumber.
I don't want
It
to
is.
find out.
It's a pelagic sea cucumber, a holothurian,
Chilling.
I think. It's a benthic one that just left the bottom. I don't see it. It's not a swimmer. That's interesting.
Ship them dead in this one.
All right, which Niskin? You want to do a 5-liter Niskin? You wanna do a smaller?
Yeah, we've got some. Yeah, let's pop a 5-liter Niskin. And then you've got another sea. No, what is that on the bottom? See?
Pen?
No, that's a Bathy Pathies.
Here. I don't know.
Isn't it?
It's you see it? J-Rod straight ahead?
That's something different. I want to see
it's
both
a
of
C,
it.
it's a C pen.
Yeah.
Is it?
Yeah.
You can
can't
see the
see it.
peduncle, I think. I don't know.
I'll let him bring it in. Yeah, it's a question mark C pen. I forget the name.
Oh, you're right. It is a sea pen.
Congratulations. Yeah.
Can you focus on that, please? Oh, thank you.
J-Rod's all over it.
It's a nice one.
It's a
I don't know that
beautiful
one.
Yeah, I can show it to you. I just can't think
Okay,
of the name.
that's fine. I don't know my sea pens super well. That's great.
It's got a big base.
Yeah, they do.
All about that base, about that base.
All right. Well, that's great. It's a common name, and I can't think of it.
I don't know it, but it's
Sorry.
great. That's
I
all right.
apologize. Thank you.
But look at that. You see what's around it? Lots of stickopathies.
Yeah, just little ones.
we got something like Peter.
Oh, nice.
Gorgeous.
Sponsored by Louis Vuitton.
All right. It's a good shot of that camera.
It is.
That's really nice. Isn't it? That was a great mount right there.
It looks like it's our NASCAR sponsor.
Tim, did you look at the footage off of that the other day?
Yes, I did.
Was it good?
It was good. It was good. You could see the science camera.
Oh,
you know,
nice.
as part of the frame. So they built another mount for it.
Oh,
Above the
nice.
science camera. It's great.
Are we ready to take a listen?
We are.
Yeah.
Yeah.
This is like more of a landscape view, Tim.
That
Yeah.
where it is now mounted, it is.
Mm-hmm.
Yeah.
Woof.
Go for number dos.
Roger.
I think we're at 38. Correct me
I
if
got
I'm wrong.
38. Yeah.
Good.
If it works.
That's good. It'll be a late night in the lab, but it won't be like 4 a.m. It'll be a 1 a.m. That's reasonable.
You said number 2.
Green ones.
Are we going 2 or 4?
I heard 2.
Is number 2 green?
Yeah, because it was 1.
Do you
2.
see a number?
Yeah. It's 2.
What's he doing?
Remember the nickname that used to be a friend? There it is.
Two's fired.
Alright.
Okay.
Verification.
That is
We need to
38.
confirm that.
It is.
Yes, sir.
2 it is. So we just took a water sample, which we will filter for eDNA and for microbial diversity.
Do the selfie thing. Sugar and all, I'll do it.
Okay.
Selfie of what?
Think,
What?
we're taking a selfie.
Why?
You said we couldn't do it.
Remember last time we did that?
Oh,
We
good
spent a
lord.
lot of time doing that. Remember that?
Yes, I do. That was an epic.
It was good for outreach.
Selfie.
You did it. I'm so impressed.
Where?
Right there. See, smile. I love it. You know, it's a little freaky though. I'm not gonna lie.
It is so freaky.
Oh, look at that.
Alright, so we're coming up out of here.
Looks like
Or do you
Wally.
wanna search
It
around?
looks
Yeah.
like Wally.
What's that? No, I think.
That is Sebastian selfie, everyone. J-Rod manipulated. Oh no,
It's like
Cody
Wally.
manipulated. Sebastian selfie.
So we're coming up bottom here? Or do you wanna have a little poke around to it?
I think we're. You want?
It's up to you. I didn't. I don't know.
I
I,
think
didn't
we're
see anything
good.
different for the last 45 minutes.
You know.
Yep.
Since we got on top of the ridge, it never changed.
Did you want to see the ridge up there? Want to see the ridge? Try to put it-
Oh, sure. The map, yeah.
Yeah, the map
No,
would be really
thank you.
helpful.
Yeah.
We're right there.
3D model. That's kind of
It's a
nice.
small bit of
Yep.
Yeah.
I think we're good. So I think it's time for us to leave the seafloor
Yeah.
and
bridge.
So is it nothing Niskin when we get off the bottom?
yep,
Well, a small one.
yep.
Yes, but just to let you know, we'll be coming off the bottom in the next 10 to 15 minutes.
That's what I'm saying, coming off bottom in the next 10 to 15.
Yes, ma'am. Another Niskin?
We're gonna take one as we get off the seafloor. Maybe let's do a.
There's a
It's-
jelly. I just went by.
You can just like hang on. Let's take one last look at this beautiful stocked crinoid before we come up, and then we'll go, and then up we go.
Hi.
That's got
But
it.
see, look,
What? What?
ectoparasitic. Well, let's take a look for some snails while we're at it, and then we'll come up. Might as well.
The Zingo.
Can you zoom way in?
Grapes.
All right. We're out of focus here. Oh, there we go.
He's got it.
Cool.
All I wanna do is zoom, 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 zoom.
Nice. That's a really good view
Yeah, I don't see this.
into the belly of the beast. All right. Whenever you're ready.
You wanna do it about 10 meters off seafloor?
Yeah, that sounds good. All right, everyone, we are headed off the seafloor. We'll see you back in a day or so as we head towards the equatorial Pacific. Whoa.
Whoa.
Maybe not leaving yet. We're looking at one more coral. This is the problem.
What the heck?
Like,
It's an already
what?
isn't it? It's an
Oh, wait a
already
minute. Never mind. We've got a big collection.
No,
I don't know.
Maybe.
Okay, let's take a look. What do we got?
Snail, maybe down below.
Yep. And some
Ophiocanthids,
crinoids.
crinoids,
Hydroids.
barnacles. There's already been wounded. Oh, here's this hydroids right there.
There's your oafs.
It's a gum.
That's barnacle.
That's all hydroids down
Yeah,
there
I think
too.
those are hydroids.
Showing you some woundage.
Oh, smart.
Can we look at that snail, please?
Wow. There's barnacles to beat the battle. That's
oaf
a
down.
parachuter.
Look, Tim, there's an oaf on the seafloor that's not on a coral. Do you want that one?
The one that just fell.
I'm just kidding.
I know. I know you're just
is
kidding.
this Norella?
It's a primnoid.
It is, okay.
I think it's Calyptrophora. Which we have plenty of. You guys wanna?
Wow.
Ooh, yeah. Focus on that snail.
Yeah.
That's what we need. I'm sure that's a coralivore.
Are there
So a
oafs
good
on
snap
here
zoom
too?
of it would be great.
There's oafs, there's crinoids, there's a gastropod.
Okay, got
There's
it.
barnacles in the dead areas.
There's a lot going on this.
There's a Ophiocantid there. Different ones
See,
there.
they don't heal. Like they just other stuff just colonizes the dead areas.
Well,
But then again, look at the snail. It's been moving up, right? And I don't see a whole lot of dead tissue.
no.
Do you?
Maybe it's going up the bottom.
No, I don't.
Oh, you think the snail's eating the crinoid instead? It's possible.
It's a wonderful shot. Yeah, I don't know.
I'm tempted to
They,
to
yeah.
dislodge it and see what's underneath this.
Suction tank?
Well, I would,
Yeah, suction sample the.
Do you want them?
or do you want to take that? 'Cause the snail will hang on there. I think. From past experience, if you wanted to
I
snip
don't
it,
think I
take
need more coral. I think we could either suction the snail or gently relocate the snail. So I can just see what's underneath it.
relocate. All right, yeah, sounds good. Into the slurp chamber, you mean?
You don't
Yeah.
want this for RNA later?
Well, I was looking at if you could see who was.
If you want it for RNA
There's-
later, that's a different thing.
It looks like every part of this thing is occupied
Okay.
and a little crazy, but
It's all.
We-
Can we get lasers on base too? Sorry. Yep, there we go.
there.
Thank you. Yeah. Big base. Cody, now you have that song in my head.
Yeah.
First our brand.
Abandoned ship.
there they go. That's not the one we've been working with anyway.
Yeah, if we could slurp the snail. Is that another snail to the right, actually? We might not need to.
I think we should get a snippet of the coral as well, though.
Yeah, but can we take a look at the snail let's see how it's facing this way? Right there. There
That's
we
the-
go.
that's
Is
the-
that the same baby? Sure is.
That one's easy. That one's been busy.
That one has been busy.
Yeah.
But
They're
look
the
at how
they're the
long it
same.
must have taken. Because you see those hydroids are.
Well, the hydroids have stinging cells, so they're killing the coral too.
I know, but I'm wondering if I mean, this is all conjecture, right? There's no way to know without sitting here for months and watching this one coral. But,
Yeah.
I'm thinking about the initial wound that was made by the snail that allowed the hydroids to come in and colonize. I'm just trying to figure out the sequence of events here.
Yeah.
But that snail's been busy. But I wonder how long it's been on that one branch.
I don't know. Those branches are broken off too.
I know.
With, it's usually the crinoids are not on. Usually they're on the dead part of the coral, where they hold on. They're often ripped through the coral or give the coral a problem, but it's not happening there,
Yeah, you can tell.
right? So
They're attracted and kind of weird.
hopefully, anyway.
It's
I think
thorough
it's a-
though, but look, you can
I
see a
think it's
little bit of
great-
live, fluffy tissue in the bottom. You see?
Yeah.
Base of the branch.
Mm-hmm.
Yeah. Sure do. So I don't know.
Marco.
Well, I think I think we need a snail and a little piece of coral.
So
Can we-
Marco said?
No? What did Marco say?
A gastropod and
Raise
a
it back up
Darien.
slowly.
Yes, sir.
This is
Could be a
good.
fantastic.
I think we're gonna do that, but I think we're gonna do it because we want to do some stable isotopes and see,
That's right.
right? That's the
Perfect.
goal.
I mean, look at. I mean,
Confirm coral livery here.
there's damage on all sides here.
Where's the left side of this? I know it looks actually quite healthy.
There's more damage on the down below though.
Yeah.
I think, but yeah. I would definitely get the predators and the
Yeah.
coral.
Alright, do you guys want
At a minimum.
to pick a snail, any snail?
Which one is this one down below? Huh?
Yeah, I'll go right back. Snip that corner,
So we're gonna take coral as one.
little piece.
I want
Yeah.
substitute drawn number 5.
Number 5.
Such is on maximum effort.
Snail versus the vacuum. Ooh.
Nice catch. That's 39.
Yep. Got the same.
And we'll have to record the sample separately, but make sure that they get linked, for the coral.
Yeah.
We could.
So make a note that they
Do you want
They
both
work the
snails
same.
or just one?
just one, please.
Okay.
Then a small piece of that coral.
Yeah.
What jar was that?
That was
Right.
jar
Number
number 5.
5.
I think there's
Yeah.
another snail.
I
Would you
would-
like to put the coral in with that snail?
Oh, could you do that?
I could snip it and drop it into the.
That would be great.
Then we call it the same sample number.
Yeah, that's perfect.
Thank you. That's great.
Can you also just tell me if there's another snail to the left of the crinoid, or sorry, to the right of the crinoid? I don't know.
To the right of the crinoid? Oh, yeah, up yonder. Up here?
Is it just, nope, that's just what is that?
Scar tissue.
Just, I think it's just a weird, it's just an into the page coral. The shrimp
Sometimes
behind
I
it.
prefer the words
Alright,
in
thank
my
you.
mouth.
That's huge.
The other thing I'd like to look at when we're done is snap zoom on the area where we took the snail from. But let's look at the coral first, so we get
You wanna
finish
do
the
that
sampling.
first before you go to
Yeah, why don't you
construction?
go
Oh,
look
yeah.
at it first, then I'll go there.
I'm pretty sure it was right here.
Yeah.
Yeah. It's bizarre, isn't it? So you were saying it's right here?
Yes.
Yeah, definitely.
So how come it didn't damage this
Well, that's what I'm thinking.
on the way up?
Maybe.
Did it
it's
land
going to
there?
meet the other one.
Maybe it's a flying snail. It landed right there and it just started eating. No, I'm just kidding. I don't know. But there's definitely damage, right? Can you zoom in even more? Can we use this beautiful camera as a microscope again?
Yes.
Perfect. Right there.
facing downward.
I know.
It didn't. So it walked up the other side. Oh.
I don't know.
No.
That's really helpful. I'm really glad to
No,
have
it's
this
good.
imagery.
Yeah.
Thank you. Yeah, let's grab a branch and a little branch. We don't need a whole lot. It doesn't even need, it can be from the top too.
Is this brittle?
No. But it's not rubbery either.
It snaps
it's
really quick.
Snap and then yeah, you should be able to snap in just like one twist.
You might want to take it from the top so you don't have to worry about it.
Scared by a dog.
Right there, that little piece looks good.
Perfect.
So where's that gonna go?
It's gonna go in the same suction sampler, right, Cody? Nope.
Yes.
Yeah.
Oh. Yeah.
Another test size.
Yeah.
I believe it's going to jar 5 with, but I'm not sure. Yeah, it's going in the suction sampler jar 5.
Yeah.
There it goes. Alright, alright. Did that go in? Can't tell.
Mm-hmm.
Yeah, I might have to grab the other piece and go in
Yeah, I think we, so there's a request from Steve to get a bigger piece anyway. So why don't we can also just stick it in a quiver. Whatever works for you guys. So he wants basically double what you got. Double.
So now there'll be separate samples, likely.
Okay, separate samples?
Correct? We're gonna go in a quiver, or are we going in the jar still?
It's up to them. Just let them collect it and see what it ends up.
They're going for number 9.
So I'm looking at the chat and gut contents and stable isotopes are definitely our plan for this. To see if the snail is eating the.
So you said double it. Wow.
Yeah, so a
What
little.
if I just turn
We're on 40 then, right?
Yeah.
like do this off of here or something like that?
Yeah. We don't have markers developed for these corals, so or somebody might, but we don't in the lab, so we probably won't use molecular techniques to confirm that. We'll probably use
Like this, Randy?
a little more than that, please. Maybe to the left of that Y. Yeah.
Up there?
Yep. Exactly. Okay. Perfection. Perfect. James.
Wait, that was numbers 10 then?
Yes, sir, 10.
10.
Go over 10, sample 40, from Noah the Head Snail Associate, link with FK 2139.
Oh, what? Yeah.
Beautiful.
Yeah. Yeah, exactly.
There we go.
Alright, anything else you wanna look at on this before we leave bottom?
So maybe just, see where the.
No, we're good.
You like this image?
That was crazy. We're like, let's go up, and then turn around, and there's this giant from nowhere.
What, there's more to see?
No, it's just the fact that there were stigopathies and like all these, you know, they're gonna go up, and you turn around, and there's just this giant.
Yeah.
You wanna say your goodbye?
It's a beauty shot.
All right, goodbye. I
Bye,
don't like
Marco.
No, just kidding. Thank you everyone so much for tuning in. We have loved exploring the seafloor with you on this brand new, brand newly mapped, previously unexplored seamount in international waters on the high seas. We've gotten a lot of collections. This was intended to be a high collection dive, and I think we have done what we came here to do. We're going to take one more Niskin sample. Don't forget, boys. As we
Oh, there's a crazy
and we'll be on the next dive in about a day or so. And then after that, just so you all are prepared, because I know you like watching these dives, and we love having you here, we'll be in a slam. We've got basically 15 days, maybe more, of just nonstop, all the diving again and again and again. So we will be on the seafloor just about nonstop. These are our two warm-up dives. So get ready to not sleep or get anything else done as you hang out with us in the bottom of the ocean. This is great. Again, all the work that we're doing today is part of several science objectives to try to think about corallivery and spongivory, coral and sponge biodiversity and associate biodiversity, trying to understand new parts of the seafloor. We have some geology objectives to try to understand the underlying geological history of these seamounts, and we're interested in the microbial diversity, both from a culture standpoint and from a sequence standpoint to try to understand how the microbes contribute to everything, and then specifically whether or not all of this predation we're seeing, whether or not microbes colonize these open wounds. So we'll be checking it out, investigating these questions for the next month. I'll see you next